Hello everyone, and welcome back to the second day of the Medical Devices track of the 2022 Regulatory Education for Industry, or REdI, annual conference. I'm Joseph Tartle, your moderator, and we will continue where we left off yesterday talking about the post-market side of the devices track and keeping quality devices on the market. Here, our medical devices are maturing and now are either getting ready to or are on the market and impacting the public health by being safe and effective and working as intended. Today's session will consist of several medical device quality system topics that I like to refer to as the morning of medical device quality systems. Thank you again for your participation at REdI. So first, here we're going to talk about our learning objectives. We'll review a couple of housekeeping items, then we will look at today's medical device post-market agenda and meet the speakers, and last, look at your call to action. So housekeeping. Speaker and final evaluations. During each presentation, we'll have a link to a speaker evaluation under the speaker's picture or information on the right side of the screen. We'll encourage you to take the time to complete the evaluation as your feedback is much appreciated. In addition to the speaker evaluation, as we have our final evaluation, this information is important for us to determine how we are meeting your needs and in planning future educational outreach events such as REdI. Continuing education and recordings. Continuing education. CE. The full 5-day conference offers a total of 32.5 continuing education or CE credits in support of RAPS, SOCRA, SQA, and ACRP. This is for attendees who self-identified themselves as healthcare professionals such as physicians, nurses, and pharmacists during registration. You need to attend in real time and through the Adobe Connect platform. Unfortunately, CEs are not provided if you're watching on the YouTube link or after the conference ends. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of a single company. Speakers are required to disclose to the attendees when products or procedures being discussed are off-label or unlabeled, that is, not FDA approved or cleared, and any limitations on the presentation information. Note, we may reference off-label use of FDA approved products. We have no disclosures for today's program participants. To obtain CE credit, participants must attest to their attendance and complete the final activity evaluation through the CE portal, ceportal.fda.gov, within 14 days of the conference. You'll need to complete the faculty evaluation for each day. Each track will have its own survey evaluation link. When an attendee completes the survey evaluation, they can download a certificate of attendance. For any pharmacists and pharmacy tech in attendance, please be sure to provide your correct NABP and date of birth information in the required format. Pharmacists should log into the CPE monitor 8 weeks after the last session to ensure your credit is captured correctly. To simplify things, we will be emailing the claim codes each evening of the conference with the survey link, so be on the lookout for it. And recordings. So all sessions, including today's, are being recorded, and the recordings will be made available at www.fda.gov/REdI2022. So please take a look at those recordings and feel free to share those among your colleagues. So now let us get to our agenda for the medical devices track today. The morning of quality systems will cover 3 important quality system topics and then wrap up with a presentation by an Office of Regulatory Affairs, ORA, medical device investigator on remote regulatory assessments. You can see the agenda here highlighted in the green box on this slide. I cannot think of more important and timely post-market topics than what you will hear today. And the first of those is the Quality Management System Regulation Qmsr Proposed Rule Overview and Status Update. So let's start with some background. The year was 1996. There were about 36 million web users online. The flip phone and Palm Pilot were just released, as was the Nintendo 64 gaming console in late September of 96. Then on October 7, 1996, FDA released the Quality System Regulation in the Federal Registry. 30 years later, in 2016, the latest edition of the International Standards Organization Medical Devices Standard was released, ISO 1345-2016. Today, it's 2022, and currently FDA is reviewing the comments provided to the Quality Management System Regulation Qmrs proposed rule. We are lucky enough here today to hear from Melissa Torres, one of the Center's quality systems expert and a point person for updating the proposed rule, which will eventually become our current good manufacturing practice for medical devices, and Keisha Thomas, another of Cdrh's quality system experts, also leading this update, who will then answer your questions during the Q&A session. Your quality management system is a focal point in ensuring that devices are designed and manufactured appropriately, and that those devices work as intended and are safe and effective. So I'm very excited to hear about this talk. Next, we'll hear from Joseph Hillring from the Division of Industry and Consumer Education, or DICE, on purchasing controls at a glance. Supply chains, raw materials, safeguarding that your vendors will and do provide you with the quality materials and services that you need is a very important aspect, especially these days. This presentation will explain the requirements identified in the Quality System Regulation and the International Organization for Standardization ISO 1345-2016 for purchasing controls. Next, we're going to go forward with process validation. So process validation establishes by objective evidence that a process consistently produces a result or product meeting its predetermined specification. This presentation will discuss the process validation requirements of the Quality System Regulation and the requirements of ISO 1345-2016. Tanya Wilbon, our DICE Post-Market and Consumer Branch Chief, will review the similarities and differences of these requirements and also review examples on implementing them. And then last, to wrap up the track, Brittany Franklin, a medical device investigator with the Office of Regulatory Affairs, ORA, will present on remote regulatory assessments. Currently, RRAs are a voluntary program for medical device facilities that is being implemented by FDA's Office of Medical Device and Radiological Health Operations, OMDRHL. This presentation will discuss this new inspectional tool FDA will be utilizing moving forward. You will obtain insight geared towards the agency's jurisdiction to conduct a remote regulatory assessment, the process of completing an RRA, and provide additional resources to learn more about them. Again, my name is Joe Tartle, and I'll be your moderating for today's session and program. Tanya Wilbon, who is also presenting today, will be helping out to facilitate the live Q&A portion after each of these presentations. Please, during this time, ask your questions. We want to hear from you. So, how can I learn more about medical device regulation outside of this great conference? First, I recommend that you start with the available online resources that we noted yesterday, such as Cdrh Learn and Device Advice. Both of those are great, comprehensive, educational, regulatory tools that are available to you 24/7 and are at your fingertips for free. However, if you've exhausted these resources and you still have a specific question that you cannot find the answer to, please feel free to contact us at the Division of Industry and Consumer Education. We are mandated to help you understand your regulatory requirements. That is why we have our educational mission to do so. So please reach out to us. Please note, though, that any pre-market review questions about a submission that is under review or a facility that is currently going underneath an FDA inspection need to be communicated to the reviewer and to the investigator respectively. We cannot interact or do not have any aspect of interaction with regards to an active submission review or an active inspection. Also note, we are available to educate and provide you non-binding public information. In this link below, at our webpage, www.fda.gov/DICE, please click on it, and there's a 2-minute promotional video on the webpage. When you get a chance, again, outside of this conference, please click on our promotional video and take a look at what services that we can offer to you. We're here to help. So how can you contact us? Contact us by phone or email. We're available Monday through Friday, 9 to 12:30 p.m. and 1 to 4:30 p.m. Feel free to reach out to us and call. However, please, with regards to this conference, where we have over 13,000 people who have been registered and close to 3,000 people who tuned in yesterday, and it looks like several, more than 400 today in this room, probably several hundred in the other room, and those on YouTube. Wait to call, not just call directly after this conference. We're here to help, but at least during the conference, you'll get a message right at this time that you need to tune into REdI. So please reach out to us and feel free to contact us when you can. If, however, you want to text or email, feel free to email us at dice@fda.hhs.gov. And again, we are here to help you. So what does that mean for today and for what we would like you to do? This is your call to action. Enjoy the program, interact and learn as much as you can. We always try to teach new things and educate and bring the best speakers and experts in their particular areas to this program. So I know myself, I learned a few new things yesterday, and I guarantee you I'm going to learn quite a few new things today with this post-market quality system track we have going on this morning. So please interact and learn. And as I said earlier, make sure to ask your questions. Your questions are important because it helps us understand what it is that you need to know. Okay, so please ask those questions. You'll have the Q&A bubble that will appear. Put those questions in and we will get to as many of them as possible. Last, take advantage of the Fda resources that are available to you, be they Device Advice, Cdrh Learn, or any of the online materials that have been put together particularly for you so that you can understand your regulatory requirements. You know, it is a belief within our division that knowledge fosters voluntary compliance. We think that everyone wants to do the right thing and make sure that their devices are safe and effective and work as intended. As we talked yesterday at the beginning, our families, your families, ourselves, all utilize these medical devices. And we do believe that most manufacturers want to do the right thing, hence why they are participating at a conference like REdI. And then give us your feedback on what you need. So this is where we put out those evaluations. We want that information so that we can build better educational programs that meet the needs of our customers. And our customers are you. So please make sure to provide that information. And enjoy. We thank you for your participation, and we very much enjoy the fact that we're able to have this outreach, this very important outreach, so that everyone can be on the same page and understand what it is, because these are medical devices that are being designed and manufactured. They have a huge impact on all of our lives and on the public health in general. So with that, I'm going to give a little bit of an intro for our next presentation. Overview Proposed Quality Management System Regulation. And it's going to be presented by Melissa Torres. Melissa Torres is the Associate Director for International Affairs at the Center for Devices and Radiological Health, Cdrh, where she is responsible for the oversight of Cdrh's involvement in international programs such as the International Medical Device Regulators Forum, the IMDRF, which is now taken over from the Global Harmonization Task Force. It's been almost 10 years now since the change, and She oversees the Medical Device Single Audit Program, or Mdsat, a very important program in the center. She's also responsible for managing communications on technical and regulatory issues with foreign governments and international organizations, and developing policy in response to emerging international issues. Before her current position, Melissa worked in Cdrh's Office of Device Evaluation, known as ODE, and then that changed to OPAC, the Office of Product Evaluation and Quality, as a branch chief and deputy director. Melissa received a Bachelor's of Engineering in Biomedical Engineering from Vanderbilt University and a Master's of Engineering and Science degrees in Biomedical Engineering and Engineering Management from Catholic University of America. Melissa is also an ASQ certified quality auditor. So with that, let's give a warm welcome as we hand this presentation over to Melissa. Thank you.
Good morning. My name is Melissa Torres, and I am the Associate Director for International Affairs in the Office of the Center Director at Cdrh. This morning, I will be presenting an overview and background of the proposed Quality Management System regulation. This morning, I will be discussing the proposed amendment to 21 CFR 820, the Quality Management System Regulation, or Qmsr. I'll review the requirements of the standard, ISO 1345, and discuss why FDA has decided to incorporate the standard by reference into the proposed rule. And finally, we will look at some of the changes being proposed in the Qmsr. As an overview, FDA is proposing to harmonize the current QS regulation for medical devices with the standard ISO 1345, which is used by many other regulatory authorities around the world. To accomplish this, we are proposing to incorporate by reference the 2016 version of ISO 1345, because many of the requirements in the standard are substantively similar to the requirements of the current quality system regulation. The proposed Qmsr was open for a period of 90 days for public comment and just closed on May 24th. We are now beginning the process of reviewing those comments that we have received. For those that may not be familiar, ISO 1345 is the standard that outlines the requirements for a quality management system for medical devices. This standard can be used by organizations involved in one or more stages of the life cycle of a medical device, including the design and development, the production, storage, distribution, installation, servicing, and final decommissioning and disposal of a medical device. FDA has had a long interest in the utilization of ISO 1345, and much of the rationale for incorporating by reference the 2016 version of ISO 1345 is highlighted here. We know that regulatory expectations for QMS have evolved since the publication of the current QS regulation over 25 years ago. ISO 1345 is used by many other regulatory authorities around the globe as a QMS requirement. Therefore, many global medical device manufacturers already have to meet the requirements of ISO 1345. Moving to ISO 1345 can benefit global medical device manufacturers to have a more globally harmonized QMS and, for the most part, comply with a single set of requirements. This could allow FDA the ability to work closely with regulatory authorities around the globe and facilitate regulatory convergence on QMS. In addition, FDA has always sought ways to harmonize utilizing ISO 1345 through programs such as the Pilot Multipurpose Audit Program, the ISO 1345 Voluntary Audit Report Submission Pilot Program, and finally, our current Medical Device Single Audit Program, or Mdsat. Through these programs, which utilize 1345, FDA learned many lessons which have demonstrated the feasibility for utilizing ISO 1345 as the basis for our QMS requirements. Also, through our analysis, we have determined that the requirements in the 2016 version of 1345 are substantively similar to the quality system regulation. The benefits of ISO 1345 as compared to the QS regulation is that the standard has more modernized QMS principles. It also has greater integration of risk management activities and stronger ties to ISO 14971. The QS regulation does not provide that level of specificity, as many of FDA's expectations are simply outlined in the preamble of the QS regulation. Finally, the requirements in the standard are globally harmonized and used by many other countries, including major markets such as Europe, Canada, Australia, Japan, and many others. The goals in developing the proposed Qmsr were to be able to simplify and streamline our QMS requirements. We also wanted to reduce burden on many manufacturers by aligning as much as we could FDA's QMS requirements with the globally harmonized QMS requirements outlined in ISO 1345. Therefore, we incorporated by reference the 2016 version of ISO 1345, while keeping country-specific requirements at a minimum. We really only focused on requirements that were deemed necessary to remain in alignment with the Federal Food, Drug, and Cosmetic Act. It was also important to maintain the same level of assurance in a firm's QMS and their ability to consistently manufacture safe and effective medical devices. As an overview, the proposed Qmsr withdraws most of the requirements of the current Part 820, but retains the scope and a number of the definitions. We incorporated by reference the 2016 version of ISO 1345, but we also ensured that we kept U.S.-specific requirements at a minimum. Where possible, we proposed to accept the incorporated requirement in ISO 1345 without any modification. In some cases, we did have to establish provisions in order to ensure consistency with other applicable FDA requirements. These areas include definitions, clarifying concepts, and in some cases even requirements. The proposed Qmsr also includes conforming edits to Part 4, the cGMPs for combination products. It is important to note that the conforming edits do not change the cGMP requirements for combination products. They only identify the corresponding ISO 1345 clauses and the called out provisions in Part 4. As you can see, the proposed Qmsr is quite streamlined. There are sections for the scope, definitions, incorporation by reference, the requirements for QMS, clarification of concepts, control of records, and device labeling and packaging controls. For definitions under 21 CFR 820.3, we have a number of categories applying to the definitions. There are some definitions that we have withdrawn, retained, modified, clarified, or superseded, and also added. For example, we have withdrawn the term established because it is already encompassed within the standard. As you can see here, we have also decided to retain a number of definitions from the current QS regulation to ensure adequate interpretation of the requirements in the proposed rule. There are also terms that we have decided to retain but modify portions of the definition. For example, the term rework has been revised to remove the reference to a device master record, since that term is not used in ISO 1345. In addition, to ensure that a product also includes services, we've decided to add the term service to the definition of product. There are 3 terms that we have also decided to clarify to ensure that they align to the FDA's legal and regulatory requirements. Those include medical device, labeling, and manufacturer. And finally, we've decided to add the terms customer and process agent to ensure correct interpretation of the standard. Looking in more detail at the requirements under 21 CFR 820.10, this is where we have the requirement for a manufacturer to document a QMS. We have also outlined how the requirements for a QMS link additional FDA requirements, such as medical device reporting, UDI, corrections and removals, and tracking, as well as the applicability of design and development activities. For traceability, we have clarified that this requirement in the standard under Clause 7.5.9.2 also includes devices that support or sustain life. The clarification of concepts under 21 CFR 820.15 identifies those terms in ISO 1345 to those of FDA. For example, for organization, we clarified the term to also include the meaning of manufacturer as defined by FDA. The standard uses the term safety and performance throughout, so when that term is used in the standard for FDA purposes, we would like it to mean safety and effectiveness. It's important to note that we are not making any assessment about the differences between safety and performance and safety and effectiveness. We are simply changing the term to align to our regulatory requirements. In addition, we've also clarified the term validation of processes to mean process validation for FDA's purposes. The control of record section under 21 CFR 820.35 supplements record-keeping activities outlined in ISO 1345, such as requirements for the signature and date, specific documentation that's required for areas like complaint handling and servicing, UDI, as well as the confidentiality of records. It is important to note that all of these requirements are in addition to those outlined in Clause 4.2.5 of ISO 13485. Given the number of recalls and post-market issues we have seen throughout the years, FDA has decided to supplement the requirements for device labeling and packaging in Clause 7.5.1e, as we believe that ISO 1345 lacks the specificity to ensure adequate control of these activities. Therefore, FDA has decided to retain the requirements for device labeling and packaging from the QS regulation, which are now outlined in 21 CFR 820.45. There are several key considerations with the proposed Qmsr. Importantly, the proposed Qmsr does not modify which establishments or products are subject to Part 820. It specifically incorporates and references the 2016 version of ISO 1345. We do recognize that while the standard historically does maintain stability for many years, it will eventually need to be revised. Any future changes to the standard will need to be evaluated to determine the impact of the rule, and if necessary, would need to be addressed through rulemaking. FDA is currently proposing a transition period of 1 year after publication of this final rule. We have also ensured that the standard is free and publicly available. It can be accessed through the ANSI Incorporated by Reference portal at the website listed on this slide. We would like to note that FDA inspections will not result in the issuance of a certificate of conformance to ISO 1345, and that manufacturers who have ISO 1345 certificates are not exempt from FDA inspections. There are also many FDA implementation activities that we have to undertake. For example, we will need to update our technology systems, train our FDA staff in the center and the field on the new requirements. We will need to replace the current quality system inspection technique with a new inspection model that is aligned with the requirements of the final rule. We also have to revise relevant regulations and other documents that may be impacted by this rulemaking. And finally, we need to ensure that we educate and communicate with our stakeholders. The first knowledge check. The proposed Qmsr replaces most of the existing regulation by incorporating, by reference, ISO 13485, 2016. True or false? True. Knowledge check 2. Identify the benefit of incorporating ISO 1345 by reference. 1. The requirements are very similar to the current Part 820, the QS regulation. 2. It is globally harmonized and widely in use. 3. It has modernized QMS principles. Or 4. All of the above. all of the above. In summary, FDA has issued a proposed rule to amend Part 820. That proposed rule was out for public consultation for a period of 90 days and just recently closed. This proposed rule will incorporate by reference the Quality Management Standard for Medical Devices, ISO 1345, and more specifically the 2016 version. As I explained, this change will further harmonize our global regulatory requirements for quality management system by being able to align ourselves with many other global regulatory authorities. And finally, the benefits of this change include the fact that it is more simplified and streamlined regulatory requirements and also has less burden while maintaining a similar level of assurance in a firm's quality management system. I would like to thank you for your time this morning.
Thank you, Melissa, for that very important and informational presentation. A lot of great information that was garnered there. And now we're going to move on to our question and answer portion of the session. And joining us for this is Keisha Thomas, who is also one of the leads with this modification to adopting this proposed rule to become the new quality management system regulation. So with that, Keisha Thomas is an Associate Director for Compliance and Quality in FDA's Cdrh's Office of Product Evaluation and Quality, and is helping lead this change. She's one of the center's quality systems experts, and prior to the current position, previously had worked in the Office of Compliance, has served as an AMI instructor, was part of the ISO 1345 voluntary program, for those who remember that program, about 10 years ago. So she has a lot of knowledge and a lot of good insight with regards to quality systems in general, and in particular, this change. So with that, I'm going to ask Keisha the first question that we received. So, how will future revisions to the ISO 1345 standard impact this new proposed quality management system regulation?
Good morning, Joe, and good morning, everyone. Fundamentally, any future revisions to the standard would be evaluated to determine the impact of the changes and whether the rule, when finalized, should be amended. If we deem that, revisions are necessary and appropriate, we'll update the final regulation in accordance with our Administrative Procedures Act. So we have considered this piece, and we've thought about what would happen if. It would really be dependent on the impact of the changes to the regulation itself, and then we would follow the appropriate steps depending on how that assessment determines the need for a change.
Okay, thank you. It's a great answer. And our next question, are manufacturers with Mdsat, Medical Device Single Audit Program Certificate of Conformance to ISO 1345-2016 exempt from FDA inspections? I know this is more of an Mdsat question than a proposed rule question, but this seems to be coming up quite a bit.
So they're exempt from routine surveillance inspections because they get their audits, on a specific schedule through the Mdsat program. However, that does not mean that the agency does not have the ability to go in and do directed or for cause inspections if there are signals or reasons that we identify a need to go in aside from the routine surveillance inspections. But fundamentally, for Mdsat participants, they are not held to FDA routine surveillance inspections.
Okay, thank you for that answer. It basically is clear. FDA holds its inspectional authority. Even if you're a participant of Mdsat, we still hold that inspectional authority for calls or when needed. So thank you for that answer. Our next question is, the standard has requirements for mandatory post-market surveillance more than just that of complaints. How is the agency thinking to incorporate that into the proposed update of the new rule?
Joe, can you repeat that? I had a portion where it clipped out for me.
Sure, no problem. So the standard has this is the way the question reads. The standard has requirements for mandatory post-market surveillance, more than that for just complaints. How is the agency thinking to incorporate that into the new proposed updated regulation and rule?
It's actually a really good question, and we're actually having conversations about that now as we're going through the comments and preparing for, the response and continued implementation and policy development. As of right now, we don't have an expectation, and I'm going to emphasize as of right now, because we've literally just started going through the comments and are working some of these policy decisions out. But what I can tell you is that's an area that we are paying close attention to and assessing to determine what we will do, if anything different in that area, whether if there is something different that's done, there would potentially be a need for guidance and further things, but we have not made a determination yet that we would be, to the extent of which we will be requiring the full set of requirements. And so that's really a to be determined. And when and if those expectations evolve, or increase, there will be communication, education, guidance, and time to prepare from the agency for stakeholders.
Okay, great answer. And yes, everyone in the audience needs to remember this is just a proposed rule at this point. They just closed the comment period about a month ago, so there's still going to be a lot of time to sort through those comments and come up with answers to a lot of the things with regards to these changes. So with that, let's go on to our next question. Does this mean the quality system inspection technique, QSIT, which is currently being used, will no longer be used for medical device inspections?
That is correct. A new inspectional paradigm and model is being developed to align with the requirements of the proposed rule and ultimately the final rule when that's completed. So yes, the QSIT will go away as we know it, and there is a replacement inspection model and paradigm in the works.
Okay, great. A lot of work that's going to need to be done all around. Our next question. Will the quality policy manual, as required under 1345, be audited by FDA? And is the expectation that the manual should be updated to include the special modifications or additions or clarifications that FDA implements in the final quality management system regulation? So this is kind of a two-part question. So the first one is the aspect of the quality policy manual that's always been required for ISO. Will that now be audited by FDA? And then the second part of the question is, will that manual need to be updated to include any of the additions and clarifications of the new proposed rule?
Thank you, Joe. So the first part of the question is, we're not planning to carve out any areas that we will not inspect or review, aside from what you currently expect to have reviewed when you get an ISO 13485 audit. So in the spirit of harmonization, we are fundamentally not trying to further have prohibitions or carve outs for what we will not look at when we come, and trying to be a bit more in alignment with everyone. So, I think during the presentation, you all heard, it's not our goal to have too many departures and carve outs. We really are trying to harmonize as best as possible. So where we can, we're anticipating a full alignment in the areas outside of those that you've already seen in the proposed rule for departures. So if your quality manuals are being assessed and evaluated currently during an ISO 13485 audit, it will be in a new inspection for FDA. As far as updating the quality manual, so, the scope of the quality management system, right, as defined in the quality manual is, going to be influenced by, the regulatory requirements and the roles in your organization. And so fundamentally, the activities that are undertaken by your organization should be identified in your quality management, in your quality manual. So yes, fundamentally, you will have to update your quality manual, just like you would now if there are exclusions to the activities that excuse me, to the quality management system activities that would be listed in your quality manual. Sorry, it's tough tongue warning right now. And so the same way that there would be exclusions carved out, the additions would need to also be included and updated in the quality manual. That would be the expectation.
Nope, and that's perfectly understandable. I mean, everyone's been working with a quality manual for the most part. I know it's not in the original quality system regulation, but I don't know too many firms that don't have quality manuals. And FDA generally looks at those manuals because those are the overarching aspect of your quality system. And it makes sense that any changes to it, whether it's, you know, regulatory or otherwise, would mean modification. So thank you for that answer. That definitely clarifies things. So our next question, could you explain more what is meant by, or clarify what is meant by, incorporating by reference?
Sure. So fundamentally, because the standard itself is a copywritten document that we don't own the license to, what incorporation by reference means is we will incorporate the requirements of the standard into our regulatory framework, and it will become a part of our regulation, but it will be incorporated by a reference to the standard and the ability to, you know, access the standard and its requirements at a centralized location. And so because in situations where we, you know, are including and incorporating standards that we do not own, the rights to, the incorporation by reference is the legal way in which the agency can incorporate that standard legally, but not violate the copyright limitations on it.
Thank you for that answer. That hopefully helps everyone understand what is meant by this incorporation by reference, because you'll hear that term and see that term quite a bit. So now our next question has to do with some aspects of harmonization, and I'm going to put a couple of these questions in a row, but I'm going to do them one at a time. So the first question is there any benefit for the FDA doing this harmonization?
Sure is. I think the agency as a whole has been engaged in and very interested in harmonization activities as long as I've been in the center, and it's been almost 20 years. Excuse me, just sinus drainage this morning. And I think there's value added not only for the agency, but also our stakeholders when we work as a global industry to align the requirements and harmonize where we can, so that we have stakeholders that are adhering to, if not the exact same set of requirements, a very similar and aligned set of requirements. Because we all fundamentally have the same goal in mind, right? And so there's huge value and benefit in continuing to move in the direction of harmonization where we can, whether that's post-market, whether it's regulatory, whether it's pre-market. I think there's a continued benefit in doing that, because we are very much in a global environment, especially in the medical device industry. Specifically, and that's not going to change. And so, you know, where we can and should harmonize, we will continue to look at, you know, appropriate ways to do that. That is not only beneficial to the agency as regulators, but also to our stakeholders and our patients as well.
Yes, I mean, absolutely. This has been something I think manufacturers and industry has been asking for, especially over the last 10 years. And as Keisha was saying, the whole concept of harmonization goes back to the 1990s with the original Global Harmonization Task Force. And I can remember with the ISO 9001 1994 standard, and then the 1996 medical device standard, there was initial discussions even back then of trying to have greater harmonization, because almost everyone that I know has one quality system, and they want that single quality system to be able to cover all of these different requirements, and harmonization is one of those efforts to help it do that. So with that, the next build-off question to this is, so by incorporating by reference much of ISO 1345 through this harmonization, will this complete FDA's global harmonization efforts for quality management systems?
Sure, Joe. So kind of as I alluded to just a minute ago, I don't know that we have like a finite end date or a finite set of activities that will fundamentally complete, harmonization activities. I think we will continue to assess across the medical device ecosystem as a whole for opportunities that are both advantageous and beneficial to all of the stakeholders involved. And so I don't know that I would say that, this effort is kind of, like the completion of our harmonization efforts. I think we will continually assess the utility and opportunity for global harmonization where the possibility is advantageous to our stakeholders and there's a benefit to public health.
No, thank you for that. It sounds like we get a little bit closer, but we're not there to full aspect yet. But we continue, it seems like every bit getting closer. Yep.
Right. And in full transparency, we're talking a little bit this morning about how, we got to this place because of the evolution, right, of quality management system practices and concepts. And those things, much like the medical device industry, will continue to evolve and things will continue to change. And so in keeping current and consistent and aligned, I think this will be a constant state of affairs as we all continue to kind of move along the evolution of, oversight and quality management systems for the regulation of medical devices. So I don't think there's like a finite stopping point. I think this is just the state of affairs and what will continue to, carry on through the future, but in a variety of different ways and manners.
Great point, because that's an absolute great point to point out that these things will continue to evolve and things will continue to change, which has kind of been a theme of this REdI program's devices track, is things will evolve over time and continue to do so. So with that, let's move to our next question. Will FDA accept ISO certificates? So we talked about Mdsat certificates. Now the question has come up about ISO certificates.
Yeah, we've gotten that question a lot, even prior to publishing the actual proposed rule. And right now, that is not in our plan. That's not to say that there could not be utilization for or recognition of firms that actually, have an ISO 13485 certification and have an established quality management system in place. The certificate itself, the certification itself, is not going to provide a pass on an FDA inspection because the organization has that. That's not an expectation that we have. But even currently, we do at times utilize, or review ISO 13485 quality management systems at times in lieu of, additional information. And so the best example that I can give is during the pandemic, right? When we were not able to get out to as many organizations as we would usually do or would need to. And we were information gathering and trying to get, a sense of, the state of affairs in a firm for a particular reason. And there have been times where we have utilized information from 13485 audits, from the Mdsat information that we have, to help and assist in decision making or getting a snapshot of what the state of affairs is in a firm. I think those activities will continue, but there is no expectation that the certification itself will, give a pass and there's no need for an inspection, or we will use that certification to say that a firm is in a state of control. There's no expectation at this juncture. But as I just mentioned, evolution occurs, and as we move into this space and implementation, and eventually get to a point of this is the law of the land, and we're operating in a space. There's always going to be consistent evaluation and assessment of the information, and the information that we're gathering, and the potential impact on our state of operations, and in the spirit of making good decisions for our stakeholders as well as ourselves. You never know what could happen. Don't take that as FDA said, eventually they'll do it. That's not what I'm saying, but I'm saying there'll be a constant assessment and evaluation as this evolves. But currently, right now, the certification is not going to likely impact FDA's activities or decisions.
Okay. Thank you. So FDA retains its inspectional authority for its regulations and rules. And it sounds like we don't know the future, so we'll see what happens. But the answer right now, FDA will maintain its own inspectional authority. And then the next question kind of builds off of that. So will this mean in the future, or will this mean that all medical device manufacturers in the US domestically will then be required to have ISO 1345? So this is kind of the flip of it, of will US FDA require manufacturers to have ISO 1345 certificates?
No, FDA is not going to include as a part of its requirements the need to have an ISO 1345 certification. What we will expect is that domestic and foreign manufacturers, following what will eventually be the final rule, they will adhere to the requirements right now, but there is not any plan to require an actual certification to 13485 in order to meet FDA's requirements and expectations. No.
Okay, thank you. And I think that alleviates some stress for some folks, because I know that they were worried about that, but nope, there doesn't sound like there's any need for worry there. So our next question is about follow-ups after inspections. So if a company is not in compliance with the aspects of the 1345 reference sections that are being absorbed into the new proposed rule, will warning letters still be issued? So I think this is more about would there be changes into the way that we do our current enforcement?
That's a good question. And fundamentally, Joe, I think your translation is correct. So yes, once there is a final rule that incorporates the requirements of 13485 into the revised version of 820, we will treat those requirements the same as we do now. So they will be the requirements for our quality, the fundamental requirements for our quality management system, and we will assess them the same way that we currently do for 820 requirements, and we'll still retain the ability to continue to levy whatever appropriate action the agency deems necessary. So yes, that won't change. We will still come out and do an inspection to the requirements and the process fundamentally for making an assessment for compliance and any potential enforcement action. We're not envisioning any changes to that process per se.
Okay, thank you. And then for our last minute here, I'm going to turn it back over to you for your final thoughts, Keisha, about everything that's going on in this space and what you would advise all of these stakeholders that are listening.
Thank you, Joe. I appreciate that. And so I know this is a significant change. It requires a lot. And I know the messaging about this is that the requirements are fundamentally or substantively the same. And that is true. That does not mean that the agency does not recognize that this is a heavy lift, because it's also a heavy lift for the agency. And so we're not at all dismissing or negating the fact that for companies and organizations that do not already, adhere to 13485, that this is not going to be a shift and a change. And so I want to say that first and foremost, because people are like, hey, the agency is making this seem like it's easy. It's not easy. It's not even easy for us. And so there is a lot to be done. inside of the agency. There's a lot of activity that has been going on for the last year and a half that will continue to go on in terms of not only responding to the comments, any evolution of policy and policy development that needs to happen, an assessment of all agency documents, guidances, other regulations that are potentially impacted by this change. We've talked a little bit about the inspection model that needs to be development that is in process as well. So it's a lot. It's just a heavy lift inside of the agency as it is. There's training and education and development that needs to go along with this. And so the agency is absolutely committed to communicating along the way of this change what our thinking is, and to put information out there, to partner with stakeholder organizations to try in any way we can to kind of, make the path towards the transition as at least informationful, to ease the burden or help with the transition in any way. As that additional information comes, we will communicate, whether that's in the form of webinars or guidance documents or a whole slew of other, mechanisms. We're committed to that. And with that said, even though the comment period is closed, we still have the ability for stakeholders to reach out to us and share their thoughts. DICE is a very good place to, funnel questions in through us. Our email addresses are all public. There are some email mailboxes that were associated and listed within the proposed rule publishing itself, where you can get questions specifically for this. And so what we would like is for you all to not only share with us, but, understand that we recognize the lift and the path that has to go forward, and we're going to try and do whatever we can do to help you all in this transition. And then the last piece of this, the biggest, most fundamental, glaring, change here is the enhancement of the risk management, or the more integrated, explicitly listed risk management activities for those of you that are not already using and adhering to 13485 of risk management activities throughout the clauses and throughout the quality management requirements, and the greater emphasis that will have for the agency as it's going through and assessing a quality management system. And so paying particular attention there will be helpful and useful. And with that, Joe, I will stop and you let me know how you want me to move forward.
Nope, that is it. And thank you so much, Keisha. And this, with her participation here and Melissa's presentation, this outlines the aspect of us trying to educate and provide as much information on this change. And I will also further note there will be more materials coming out. And yes, think about risk. Risk is something that's been going on for quite a while, and hopefully we're going to have some more educational stuff coming out about it as well soon. So thank you both, Keisha, and thanks to Melissa for the presentation. So with that, we are going to move to our next topic. So we're going to talk about purchase controls. Purchase controls are extremely important these days. They've been important going back to their incorporation into the 1996 quality system regulation. And to give us this presentation is Joseph Hillring. Joseph is a consumer safety officer in the post-marketing consumer branch in DICE in Cdrh's Office of Communication and Education. He focuses on working external stakeholders, locate and understand various regulatory requirements with a specialization in quality systems. Joseph knows a lot about QMS and QS in general, and answers a lot of the QS questions that come into us. So he's a great resource within our division. Prior to the career at FDA, Joseph worked for about 15 years working with different consulting firms on a spectrum of different medical devices. He has a bachelor's degree with a major in biology and a minor in public health from the University of South Florida, and a master's degree from Northeastern University majoring in regulatory affairs for drugs, biologics, and medical devices. So please give a warm welcome to Joseph as he talks and gives us a nice presentation on purchase controls at a glance.
Good morning. I am Joseph Hillring, and I'm a Consumer Safety Officer in the Division of Industry and Consumer Education's Post-Market and Consumer Branch. Today, I'm going to be talking about purchasing controls at a glance. We will be going over purchasing controls requirements in both the Quality System Regulation as well as the ISO 1345-2016 standard and comparing their similarities and differences. So why purchasing controls? I did a quick search of the recall database and found a recent Class 1 recall of ventilators as a good example. This establishment received a lot of non-conforming material manufactured by one of their suppliers, and when this incorrect material was used, it could result in certain chemicals being released into the patient during initial use of the ventilator. This cost the company a great deal of time in performing the recall, investigation, and correction, as well as a good amount of money to replace the product, all of which could have been avoided with proper purchasing controls in place. So, you have the Quality System Regulation 21 CFR 820 and the ISO 1345-2016 standard, and they are harmonized, including the requirements for purchasing controls. Here you can see just how closely harmonized these two sets of requirements are. If the slide was drawn to show the overlap between the regulation and the ISO 1345-2003 standard, the common pink area would be much smaller. These will be your learning objectives of this presentation. By the end of this presentation, you are going to be able to define the key terminology associated with purchasing control requirements. You will understand the importance and goals of purchasing controls. You are going to learn the specific requirements of purchasing controls for both the Quality System Regulation and the ISO 13485 standard. And finally, the similarities and differences between the regulation and the standard will become clear to you. Definitions. Understanding the terminology is very important in understanding the regulation and the standards requirements in their scope. The regulation defines product to include components, manufacturing materials, in-process devices, finished devices, and return devices. Many people think the word product means the product that is sent to their customers, but as you can see, the scope of the term product is much broader. Next, we have the definition of component, which means any raw material, substance, piece, part, software, firmware, labeling, or assembly which is intended to be included as part of the finished packaged and labeled device. A component is basically something that is needed in order to manufacture a finished, labeled, and packaged device ready to be shipped to the user. Next, manufacturing materials, which means any material or substance used in or used to facilitate the manufacturing process. A concomitant constituent or a byproduct constituent produced during the manufacturing process, which is present in or on the finished device as a residue or impurity not by design or intent of the manufacturer. It is important to recognize that manufacturing materials that include any substance that is used to facilitate the manufacturing process, such as cutting oils, mold release agents, and cleaning materials, are products that are subject to purchasing controls. Now, we will go over a couple important definitions of the standard. First, you have product, which the standard defines as the result of a process. This definition has two notes. The first breaks product into four categories: services, software, hardware, and process material. The second note defines service as the result of at least one activity necessarily performed at the interface between the supplier and customer, and is generally intangible. The definition of product in the standard includes all those items in the Quality System Regulation definition, but in addition, it specifically includes services. Next, the standard defines purchased product as product provided by a party outside the organization's quality management system. The standard also defines purchased product to include product that may or may not require a financial transaction. For example, product may be acquired from a sister company, but the company has separate and different quality management systems. The regulation in 820.50 states purchasing controls applies to all purchased product otherwise received, product, and services. So it is essentially the same. So why are purchasing controls important, and what are its goals? As I'm sure most of you know, the quality of the finished medical device depends on the quality of the raw materials, components, and services that went into it. Though the Federal Food, Drug, and Cosmetic Act gives FDA the legal authority to regulate medical device component manufacturers, the Quality System Regulation excludes component manufacturers. It only applies to finished medical device manufacturers. FDA is exercising enforcement discretion over component manufacturers and expects the finished device manufacturers to control their suppliers. As I'm sure many of you would agree, companies tend to outsource more and more to suppliers, I'm sure many of you watching do this yourself, and purchasing controls ensures the quality of those you outsource to. Quality cannot be ensured by product testing alone. Evaluation, selection, control, and monitoring of suppliers of product and services is also needed. Take a look at comment 99 in the preamble, which noted that the failure to implement adequate purchasing controls has resulted in a significant number of recalls due to component failures, mainly due to unacceptable components, which is why purchasing controls is necessary to provide the additional assurance that only acceptable components are used. I believe this certainly continues to be true today, maybe even more so. Goals. Here are the goals of purchasing controls. First, to establish and maintain requirements, including quality requirements that suppliers, contractors, and consultants must meet. Next, to select only suppliers, contractors, and consultants who have the capability to provide quality product and services. As we just mentioned, quality cannot be ensured by product testing alone. Selecting capable suppliers and then controlling these suppliers is also needed. We are designing quality into the process as opposed to just relying upon testing the device. Let's now take a look at the requirements in both the regulation as well as the standard. Here we have the requirements of 820. First, we need to establish procedures to ensure all purchased or otherwise received product and services conform to specified requirements. Second, we need to establish the requirements that must be met by suppliers. That includes contractors and consultants. These requirements are established prior to evaluating the supplier. In addition, we need to evaluate suppliers on the basis of their ability to meet specified requirements. We also need to define the type and extent of control to be exercised over the product, services, and suppliers, based on the evaluation results. Though not explicitly stated, there are preamble comments we will look at later in this presentation that make it clear risk is also used to determine the type and extent of control. Records of acceptable suppliers must be established. Many companies have what is commonly referred to as an approved supplier list, or ASL. The records of all other activities required by 820.50, such as evaluation records, are also required. We must establish and maintain purchasing data that describe or reference the specified requirements, approved in accordance with 820.40 document control. This would include product specifications and other controlled requirements documented in the manufacturer's device master record. In addition, we must obtain an agreement where possible from the supplier to provide a notification of changes in the product or service so that the manufacturer can determine the impact on the quality on a finished device. This is commonly referred to as a no-change agreement. Now we will discuss the requirements of the ISO 13485 2016 standard, beginning with Clause 7.4.1, the purchasing process. It is required to document procedures to ensure purchased product conforms to specified purchasing information. We must then establish criteria to evaluate and select suppliers. The standard is specific. This criteria must be based on the following: the supplier's ability to provide product that meets the requirements, the performance of the supplier, the effect of the purchased product on the quality of the medical device, and it must be proportionate with the risk associated with the medical device. These criteria are specifically stated in the standard if you pull it up. We are then required to plan the monitoring and reevaluation of suppliers, as well as monitor supplier performance and use the results as an input into the supplier reevaluation process. In addition, we must address supplier non-fulfillment of purchasing requirements proportionate to the risk associated with the purchased product. Records should be maintained for evaluation results, selection, monitoring, and reevaluation of suppliers, and any necessary actions. As you can see, the standard includes some more specific requirements, which I actually find quite helpful. We will take a look at the similarities and differences between the regulation and the standard later in this presentation. Now there is Clause 7.4.2, Purchasing Information. 7.4.2 requires that purchasing information shall describe or reference the product to be purchased, including as appropriate, product specifications, requirements for product acceptance, procedures, processes, and equipment, requirements for qualification of supplier personnel, as well as quality management system requirements. We are also required to include, as applicable, a written agreement that the supplier notify the organization of changes in the purchased product prior to implementation of any changes that affect the ability of the product to meet requirements. 7.4.2 requires us to maintain relevant purchasing information to the extent required for traceability given in clause 7.5.9. Again, we see more specific requirements in the standard. Traceability in clause 7.5.9 of the standard is explicitly required for components, materials, and conditions for the work environment if these could cause the medical device to not satisfy its safety and performance requirements. There are also traceability requirements for suppliers of distribution services. The last clause of purchasing requirements in the standard is 7.4.3, the verification of purchased product. This clause requires us to establish inspection or other activities to ensure the purchased product meets requirements. The extent of verification activities shall be based on the result of the supplier evaluation and proportionate to the risk associated with the purchased product. Then, we must determine whether any changes in the purchased product affect the product realization process or the medical device. The product realization process in the standard includes design and development, purchasing, production and service, and control of monitoring and measuring equipment. Continuing 7.4.3, when the organization intends to perform verification at the supplier's premises, the purchasing information shall be documented to include the intended verification activities and the method of product release. And, as always, we must maintain records of the verification. Now we are going to do some easy knowledge checks just to ensure you've been listening. So, do purchasing controls apply only to the purchasing of components for the finished medical device? True or false? False. It also applies to the suppliers of any product or quality management system related service, including consultants and contractors. Next, the type and extent of the control over suppliers is based on A, the evaluation results, B, commensurate with the risk of the supplied product, C, supplier quality historical data, or D, all of the above. And it is, of course, D, all of the above. Please refer to the definition of product in 820.3R, and remember, product is not just the final product that is distributed to the customers, it includes much more than that. Now, for what I am sure the majority of you are interested in, the similarities and differences between the standard and the regulation. Spoiler alert, they are harmonized and therefore very similar. First, we are going to discuss the similarities. Both the standard and the regulation require documented procedures for purchasing controls, including how suppliers will be evaluated and controlled. They also require that evaluations of suppliers be based on determining the supplier's ability to meet requirements. Both require the extent of control over suppliers to be based on the risk of the product or service to be received and the evaluation results. Risk is not explicitly addressed in the quality system regulation, but it is specifically addressed in the preamble comment 99, where it states that the degree of supplier control necessary to establish compliance may vary with the type and significance of the product or service purchased and the impact of that product or service on the quality of the finished device. That means risk. If the risk is low and you have determined that the supplier is highly capable, then less control would need to be required. Of course, as always, they both require records to be maintained. Both also require documented, specified requirements for purchased product and or services. And finally, they both require an agreement, where possible, with suppliers to provide notification of any changes to the product or service prior to implementations. Sometimes suppliers may not agree to a no-change agreement. Both the regulation and the standard recognize this limitation for some medical device manufacturers, hence the words, where possible. Companies should, however, document that they attempted to obtain a no-change agreement. And if a no-change agreement is not possible, then the company should consider additional controls, for example, certain receiving inspections. Please also note that manufacturers need to require their suppliers to notify them of changes before they implement the change, so that the manufacturer has time to assess the impact of the changes prior to its implementation. Now we will talk about the differences between the regulation and the standards requirements for purchasing controls. A more detailed comparison between the entire quality system regulation and the ISO 13485-2016 standard can be found in the AAMI white paper called The Comparison, as well as the AAMI TIR 102, published in 2019. You will see as we go through these slides where the standard includes much more specific requirements for purchasing controls, which is helpful. Much of this specific information is included in the preamble of the quality system regulation. One difference between the regulation and the standard is that 820 is specific to evaluate, select, and control contractors and consultants, where the standard does not point out contractors and consultants, but rather simply refers to purchased product from any supplier. Preamble Comment 104 discusses the reason FDA specifically included consultants within the scope of purchasing controls, because FDA had observed that a surprising number of firms hire consultants who have no particular expertise in the area in which the firm is seeking assistance. Another difference is that the regulation requires the evaluation of suppliers to be based on the supplier's ability to meet specified requirements, including quality requirements, while the standard requires the evaluation to be based on the supplier's ability to provide the product that meets the organization requirements, the performance of the supplier, the effect of the purchased product on the quality of the medical device, and it needs to be proportionate to the risk associated with the medical device. Here, you can see the standard is much more specific on the criteria of how suppliers should be evaluated, while the quality system regulation allows for manufacturers to set their own requirements. Again, though the regulation does not specifically state that risk shall be used in the making of decisions about the extent of supplier evaluations, Preamble Comment 103 states, the initial assessment or evaluation, depending on the type and potential effect on device quality of the product or service, should be a combination of assessment methods. Here, the regulation does not specify reevaluation of suppliers, but the standard requires a plan for monitoring and reevaluation of suppliers and requires companies to address non-fulfillment of purchasing requirements in the supplier based on risk. Monitoring and reevaluation of suppliers is included in the extent of control the regulation requires companies to define for the supplier. Comment 103 of the preamble states, the capability of the product or service suppliers should be reviewed at intervals consistent with the significance of the product or service provided. Monitoring of suppliers and taking corrective actions when necessary is also required to be addressed in companies' CAPA process. The regulation does not name specific purchasing data or information, whereas the standard is much more specific about the purchasing information, and it should include product specifications, requirements for product acceptance, procedures and process and equipment, requirements for qualification of supplier personnel, and quality management system requirements. Once again, we see the standard provide more specific information about requirements. The standard also emphasizes qualification of the supplier's personnel. Obviously, personnel can have a huge impact on product quality. There is no mention of traceability requirements in the purchasing requirements of the regulation. This is covered in 820.65 traceability of the regulation. The regulation does not specify verification of purchased product in purchasing controls, but the standard does in 7.4 purchasing. The regulation covers this in a different section in 820.80b. Lastly, the regulation does not specify the effect of changes where the standard requires us to determine the effect of changes to purchased product, as well as that purchasing information include on-site verification and product release activities. Again, the standard is much more specific. Verification of purchased product is covered in 820.80b, which is receiving acceptance activities. In addition, Preamble Comment 99 once again addresses the use of supplier evaluation results and risk in deciding upon the extent of verification activities. The effects of changes to the purchased product is covered in the regulations requirements for change controls under 820.30i, design changes, and 820.70b, production and process changes. On-site verification and product release activities are covered in the regulation requirements for purchasing data, just like any other supplier specified requirements. It is also covered in the regulations requirements for receiving activities under 820.80b. Here we have some of the resources I used during this presentation, such as the proposed rule for aligning with 1345 and the quality system regulation. There's also the link to the preamble, the quality system regulation, for Cdrh land module, the AIMI material I discussed, and a really good example of purchasing controls in the Global Harmonization Task Force guidance for purchasing controls. To summarize everything we've learned, Purchasing controls are an extremely important process within a company's quality management system to ensure the quality of their medical device. Purchasing controls ensure that medical device manufacturers select only suppliers who have the capability to provide quality product and services. Companies can outsource their device design and manufacturing activities, but there is one thing a company cannot outsource, and that is responsibility. Again, there are numerous recalls because of issues companies had with their suppliers, and it is always the finished device manufacturer who is responsible for the recall, not their supplier. Thank you so much for joining me in this presentation, and now let's take some time for your questions.
Okay, thank you for that great presentation, Joseph. Now we're going to move on to the question and answer session. So the first question that's already come in is, are distributors subject to purchase controls?
Thanks so much for that question, Joe. And excuse my background noise if you guys hear a little thunder. I guess the weather really wants the Lightning to win the hockey game tonight. But yeah, getting back to the question. So yeah, definitely distributors would be considered suppliers of services, and there really should be a quality agreement in place to establish the distributor to ensure your requirements are met. For example, keeping records of the customers to whom your devices are being shipped.
Okay, great answer. We'll move to our next question. Do we need to follow purchasing controls and issue a purchase order for consumables?
So, I mean, it depends on what the consumable is. I mean, usually it's a single-use device or maybe a cleaning agent used in the manufacturing process. But I mean, really in either case, it would still be subject to purchasing controls, and you should really adhere to your purchasing controls procedures, especially if it's being used in your quality management system or device.
Yep. So exactly. If it's something you're using and it's part of that system, you're spot on. You definitely have to make sure that it's being controlled and that those vendors are being controlled under Purchasing Controls 820.50. So next is, if I have a manufacturer and it's an internal supplier, does that internal supplier require a quality agreement?
Excuse me, I guess I was unmuted. But yeah, I mean, even an internal supplier would still be subject to purchase controls. I mean, it'd be all part of your purchasing procedures, and I would definitely probably still have a quality agreement in place just to ensure that everything's right. And if there's a change to that internal supplier, I mean, it's good to track what's going on exactly with your supplier, even if it is internal, whether or not.
Yep, good answer. We do get that question a lot in DICE when you have corporate and sister companies, and if you're dealing with a company that's outside of your quality management system, they're just like any other supplier. So spot on that you have to control them even if you consider them internal, because they're all part of the same corporate umbrella. The thing to look at is, are they part of your QMS or not? So with that, we'll move to our next question. Is our notified body and Mdsat auditing organization subject to purchasing controls?
Thanks, Joe. Yes, definitely. I mean, third-party auditors would definitely be considered suppliers and with whom you contract to perform services. So yeah, they would definitely still be subject to purchasing controls. And you definitely want to record that in your quality management records.
Okay, perfect. And our next question, our company performs audits to evaluate and qualify suppliers we consider high risk. So this links into that whole discussion of risk from earlier and then through your presentation. Is there a requirement to conduct annual re-audits of these suppliers? So our company performs audits to evaluate and qualify suppliers that are considered high risk. Is there a requirement to conduct annual re-audits of those suppliers?
Thanks, Joe. I mean, of course, if your procedures require an annual re-audit, then you have to follow your own procedures. That'd be the first thing. Other than that, there isn't really a requirement in either the regulation or the standard for annual re-audits, nor is there any specific method required to periodically reevaluate suppliers. I mean, that doesn't mean it's not a good idea. Both the regulation and the standard require manufacturers to monitor their suppliers' performance. That's in 820.50A2, and as well as in 820.100 TAPA, as well as in clause 7.4.1, and then clause 8.4 and 8.5 of the standard. So in addition, the standard in clause 7.4.1 specifically requires manufacturers to plan the monitoring and reevaluation of suppliers. But however, there's no explicit requirement that the reevaluation requires an annual re-audit. It's really the manufacturer's responsibility to determine what reevaluation activities are appropriate based on the supplier's performance in meeting requirements.
Great comprehensive answer. I'll move to the next question. Is there a requirement to notify FDA if there's changes in the supplier? Does it have to be agreed by FDA first to make a change to a supplier? So this sounds like there's two parts. First, is a requirement to notify FDA if there's a change in a supplier? And then the second part of that question, does FDA have to agree to that change?
So, I mean, in general, no, you wouldn't really want to notify FDA of a supplier. You aren't required to notify FDA of a supplier change. I mean, it probably depends on the extent of the change of what your supplier, such as if it really affects the intended use of the device or something like that, that would trip the limitations of that device or require a new 510K, then yes, you'd probably have to submit something for that change. But if it's something, but other than that, you'd really want to just have it follow your purchasing controls and, you know, evaluate your new supplier and record that within your quality management records.
Nope. Good answer. And it does depend. I mean, it depends on what it is that they're providing you. For example, if you think about sterilization, if you're changing sterilization suppliers, are you changing from same to same sterilization type? Or are you changing from ETO to, say, gamma radiation? So you'd have to depend and walk through that. But it's up to you to make those decisions as to what you do. And as for that second part, you pick your suppliers. You're on the hook for your suppliers. We don't pick your suppliers for you. So great answer. And let's move on to the next question. Is 21 CF, this is more of just a general question, is 21 CFR 820 referring to the current quality system regulation or to the proposed QMRS? So they're asking about the current 820 and what is the current 820 referring to?
So yeah, I would definitely say that it's the current regulation. You know, the proposed rule is that it's a proposed rule, so you're still subject to those current quality system regulation requirements. But other than that, I mean, is it a good idea to begin getting into compliance with the proposed rule? Absolutely, I would definitely say it's a good thing to start thinking about, especially here soon.
Nope, good answer. And just that, it's a proposed rule. You're currently on the hook for the regulatory requirements of 820 1996 quality system regulation. Okay, so our next question is, do we need to have purchasing controls, or actually this question was already generally asked, so I'm going to go to the next one above below it. What are acceptable ways to evaluate and qualify a consultant as a supplier?
Thanks, Joe. So, I mean, just like any other supplier, neither the regulation or the standard require any specific method to evaluating consultants. Commonly, I mean, in industry practice, it's common to obtain and review the consultant's resume for information relating to their qualifications for the particular service that you're hiring them for. For example, their education, background, skills, experience, or training. I mean, for some companies, they might also ask for consultant references, you know, to reach out to people that know the consultant and, you know, their background to make sure that they are qualified for what they're doing.
Okay, perfect. And our next question is, quality agreements only for raw material suppliers? What about service suppliers, such as those who perform maintenance activities?
So, I mean, yeah, quality, it's in your purchasing controls to have quality agreements with your suppliers. I mean, definitely those other, manufacturers, process suppliers or services like that, they all fall under that definition within the standard and the regulation as purchased product. So, I mean, yeah, you definitely would still want to have a quality agreement with them in place. I mean, that's one of my most common answers regarding purchasing controls in DICE when I get questions on that. How do I, do that? I always say it's always a good idea to have a quality agreement in place for that, between you and your suppliers of anything.
Yep, absolutely. Regardless if they're raw material or services, it's always a good idea to utilize those quality agreements because they help protect you. So our next question, what if a supplier is your customer and is providing free components?
I mean, whether or not the components are free doesn't really matter. You're still supplying components and they're still going to be your supplier. So, I mean, they're definitely still going to be subject to purchasing controls. And, I'd definitely still have a quality agreement with them in place, as well as, change agreements in place, and ensure all the components are meeting your specifications so they would not affect anything negatively.
Okay, perfect. And our next question, what happens if a supplier or a supplier of product refuses to sign a no change agreement? So this goes back to the questions about quality agreements and the no change aspect of it. What happens if a supplier or product refuses to sign that no change agreement?
So yeah, I mean, as I discussed in the presentation, both the regulation and the standard recognize this, and that it's not always possible for manufacturers to ensure their suppliers agree to a no change agreement. And I mean, really, in that case where they refuse that, I mean, the manufacturer should choose to identify a different supplier potentially. But I mean, if in the case where you don't have that option to find a new supplier, such as if there's no supplier of a particular product of raw material or something such as that, I mean, when that occurs, I mean, it's expected that the manufacturer will add additional controls in order to ensure that the requirements are met by the supplier, such as, I mean, when they receive that product, you really want to have some additional receiving inspection activities for that product. That's definitely what might be my advice. Have some additional controls in place for when a no change agreement is, so you would notice when a change comes for that product when you receive it.
Yep, absolutely. You're spot on. Those acceptance activities go up, and unfortunately, there's sometimes single source suppliers that they're the only person in town, and they're not going to assign agreements, in which case you got to do those extra acceptance activities as you pointed out. So next question, do my suppliers need to register?
Always registration questions. So I mean, it depends. Usually no, as most suppliers supply components or services of some kind. But if they are doing some other type of manufacturing there that would meet the definition of a finished medical device, then they would probably need to register with FDA, or they would definitely need to register with FDA. And I always recommend, we have a very handy website called the Who Must Register List and Pay the Fee webpage, and that just specifically defines how FDA defines certain establishments and whether or not they're required to register, list, and pay the fee, and identify the manufacturers if they're importer. So yeah, I definitely recommend going to that webpage and reviewing that. And if you still have questions, you can always call us or email us. We get a lot of these questions.
Okay, thank you for that. And our next question, when we use an outside establishment for doing calibration, so a calibration service on our manufacturing equipment, will we need to qualify them through purchase control since they're only calibrating our manufacturing equipment?
So yeah, definitely. I mean, again, the definition and the standard and regulation both include this as a service, so you would absolutely need purchasing controls for that service that they provide.
Okay, and this will have time for just one last question. I use the supplier to manufacture components for a device. The supplier has not validated their processes. However, we do 100% receiving inspection of those components. Is that acceptable?
Thanks, Joe. So, I mean, if the manufacturing process is one that requires process validation under the regulation in 820.75, or the standard in clause 7.5.6, and I mean, Tanya's going to talk about this coming up, so pay attention to the next one. Then, I mean, per those requirements in the regulation standard, then you really have to ensure that the supplier's process is validated. I mean, it should be evaluated as part of your qualification of that supplier, and the record should be available to you, because an FDA investigator or an ISO 13485 auditor may ask during an inspection of your facility. They may ask for those records.
Okay, thank you. And with that, I'm going to turn it back over to you for your last thoughts on purchasing controls.
Thanks again, Joe, and thanks everyone for listening. So, I mean, simply put, I mean, implement purchasing controls. I mean, whether you're purchasing components or hiring a consultant, you really want to ensure whatever you're doing meets your requirements and that they are acceptable. I mean, this is not only to avoid costly mistakes, but leads to better device quality. And really, you know, just use the resources available to you, including reading the preamble, the regulation, the standard, and really make use of all the resources that are available to you. There's a really handy global harmonization task force document on purchasing controls, as well as the AMI ISO documents, and this really makes it easier for yourself to ensure you comply with purchasing control requirements in both the regulation and the standard. Thanks again, everybody.
Thank you, Joseph, for that great presentation as well as Q&A session. With that, we are going to take a 20-minute break. So we're going to break for a short period of time and come back at 10:25. So 10:25, we'll see you back as we talk about process validation. And please don't forget, use those resources that I talked about earlier, as well as live webinars. So we'll talk more about the upcoming webinars. I think yesterday we had some placement slides that showed them. So please make sure to get on those webinars, and then right here some information with regards to the Cdrh mailing list. Cdrh New is a great mailing list to be on. So thank you, and see you after the break. welcome back from the break, and hopefully you had time to write down the different websites as it relates to communications coming from Cdrh and FDA. Those communication resources are fantastic tools to get information and to constantly have updates, so please utilize those. So next up in our morning of quality systems is process validation and process validation at a glance, which will be presented by Tanya Wilbon. Tanya Wilbon is a branch chief in the post-market and consumer branch in DICE in the Office of Communication and Education. Tanya leads DICE's efforts to educate and inform the medical device and radiological health industry on its regulatory requirements, as well as handles our consumer team and our consumer educational efforts. So Tanya does a lot with regards to the post-market staff and dealing in those post-market topics, be they both industry and consumer. Tanya has been with FDA for over 22 years and has more than 10 years of clinical laboratory experience on top of her FDA experience. She began her career at FDA as a microbiological scientific reviewer within Cdrh's Office of In Vitro Diagnostics. So prior to an OPEC, prior to an OHT7, there used to be OIR, where Tanya had worked, and was a quality systems specialist with OIR. Tanya also is an instructor for the Association for the Advancement of Medical Instrumentation, otherwise known as AMI, and she serves as FDA's Content Advisory Group and as an instructor for FDA to Basic Medical Devices course for new FDA investigators and staff. Tanya received her Bachelor's of Science degree in microbiology from Howard University and is also a certified microbiologist by the American Society of Clinical Pathology, ASCP. So with that, let's give a warm welcome to Tanya as she presents on process validation at a glance.
Thank you, Joe, for that kind introduction. And thank you, everyone, for joining this presentation titled Process Validation at a Glance. As Joe indicated, my name is Tanya Wilbon, and I am the branch chief for the Post-Market and Consumer Branch within the Division of Industry and Consumer Education. My presentation today will focus on process validation. Let me begin by asking, how do you ensure that your process consistently makes product that meet your specifications? Do you conduct 100% inspection of the finished product and process attributes, similar to how the investigator is conducting 100% inspection of the finished product in this photo, in order to ensure the product or process meet the predetermined specifications or requirements? Well, I hope not. 100% inspection of your product to ensure it meets specifications can require the use of a large number of resources and result in massive costs, yet still would not guarantee the desired results of the process. Validation, however, provides the documented evidence that a product or process meets predefined specifications and is fit for purpose. For example, meets quality, reliability, and customer requirements when operating within specified limits. Comment number 41 of the preamble to the final rule of the Quality System Regulation specifies that one of the principles on which the Quality System Regulation is based is that all processes require some degree of qualification, verification, or validation. It further clarifies that manufacturers should not rely solely on inspection and testing to ensure processes are adequate for their intended uses. So remember, 100% inspection does not equal a validated process and thus does not ensure the process was adequate and will result in products that meet predetermined specifications. Process validation is a necessary requirement in the development and manufacture of every medical device. It helps to assure product quality and reliability, process capability, and also confirms the efficacy of the delivered product. Implementing an efficient validation process does not need to be difficult, provided there is a clear understanding by all involved on how to implement such a process. As mentioned in the previous presentation, as a result of ongoing harmonization efforts, the FDA Quality System Regulation, entitled 21 of the Code of Federal Regulations, Part 820, hereafter referred to as QS regulation, has medical device requirements that are not consistent with the requirements in the ANSI/AMI ISO 13485-2016 standard that is titled Medical Devices Quality Management Systems Requirements for Regulatory Purposes, hereafter referred to as ISO 13485-2016. But rather, they are quite similar, and they also have a few differences. These harmonized requirements include those for conducting process validation activities. This slide depicts the harmonization of process validation requirements of the regulation and the standard as shown by the overlapping circles. Now, it is important to understand the similarities and differences of these process validation requirements, especially if you plan to participate in a medical device single audit program audit or an Mdsat audit, or even if you plan to simultaneously market devices in the United States, the U.S., and in a country outside of the U.S. that requires compliance to the standard. Harmonization of these requirements regarding process validation concepts makes it so much easier to navigate the regulatory requirements of many countries. This presentation will clarify those harmonized concepts and will focus on the similarities and differences of the requirements for conducting appropriate process validation activities. For this presentation, the learning objectives are to review basic process validation information, state the QS regulation and ISO 13485-2016 requirements for process validation, just as a review of those requirements, identify the similarities and differences between the regulation and the standard, and then finally review a process validation example demonstrating how to ensure you are meeting regulatory requirements of the QS regulation and the ISO standard for a particular process. So let's begin with some background information about process validation. Process validation is defined in 21 CFR 820.3Z1 as establishing by objective evidence that a process consistently produces a result or product meeting its predetermined specifications. Plain and simple, objective evidence is factual data demonstrating a truth. Process validation is an integral part of the production and process control subsystem, which is a key element of an effective quality system. It includes a series of qualifications based on established protocols that have been reviewed and approved. And all of these validation activities must be completed prior to the release of finished device for marketing. You should validate the process if clinical or destructive data testing is necessary to demonstrate that the process produced the desired result of a product. The Global Harmonization Task Force document, or the GHTF document, titled Quality Management System Process Validation Guidance, states that process validation is vitally important if the predetermined requirements for the product can only be assured by destructive testing. You should also validate the process if routine in-product testing does not examine all of the quality attributes of the finished product or does not have sufficient sensitivity to verify the desired safety and efficacy of the finished product. The requirements for conducting process validation activities, as indicated, are codified in Part 820 of the Quality System Regulation, specifically in 820.75 for process validation. They are also provided in the ISO 1345 standard in clauses 7.5.6 and 7.5.7. Clause 7.5.6 also include requirements for the validation of software used in the production and service provision. This presentation will not focus on the software validation requirements of this clause, which these requirements are also required in the QS regulation, but under 820.70I. Now keep in mind that the requirements in both the regulation and the standard are linked to other processes such as purchasing controls, design controls, and other quality system subsystems. So here is a list of a few examples of processes that specifically require validation. They include sterilization process, aseptic processing, injection molding, and welding process. These examples are indicated in both the preamble to the final rule of the QS regulation and in the GHTF process validation guidance document as those processes that require validation. As a reminder, the purpose of process validation is to ensure the process will consistently produce results or products that meet predetermined specifications when the process operates in specified limits. Deciding to validate a process has numerous benefits for the manufacturer. Those benefits include having more consistent quality products and an increase in output, the benefit of reducing inspection and testing, the benefit of having fewer complaints, fewer scrap and rework product, and being able to have predictable planning and scheduling. Another benefit is of having fewer recalls due to product not meeting predetermined specifications. So now let's review the regulatory requirements for process validation in the QS regulation and also in ISO 1345 2016 standard. The QS regulation 21 CFR 820.75 indicates that validation is required when the results of a process cannot be fully verified by subsequent inspection and testing, and shall be approved as defined in the validation procedures. Now, fully verified means being able to confirm all of the key quality attributes. If clinical or destructive testing is necessary to demonstrate the process produced product or results that meet specifications, then that process cannot be fully verified and thus shall be validated. The regulation requires that all validation activities and the results of the validation shall be documented. This includes the date and signature of the individual among the validation and, where appropriate, the major equipment validated. So again, this includes the date and signature of the individual approving the validation and, where appropriate, the major equipment validated. 820.75 of the QS regulation requires the manufacturer to establish and maintain procedures for monitoring and control of process parameters for the validated process and ensure that specified requirements continue to be met. It is important to note and to understand that although the monitoring can be accomplished at an interval and frequency determined by the manufacturer, FDA notes that the interval and frequency should be periodically evaluated for adequacy, especially during revalidation and any evaluation. The regulation requires that validated processes be performed by qualified individuals. It is again further clarified in the preamble comment that the validated process must not only be performed by personnel with the necessary education, background, training, and experience for their general jobs, but it must be performed by personnel qualified for those particular functions. For example, If an individual is performing welding validation activities, they must be qualified to perform the welding process. The regulation also requires that for these validated processes, the manufacturer is required to document the monitoring and control method and data, the date the validation activity was completed, and where appropriate, the individual performing the validation process or the major equipment that was used. And finally, the QS regulation requires the manufacturer to review and evaluate the process and perform revalidation where appropriate, and that's when changes or process deviations occur, as seen in the results obtained. An example of a process change could be a change to the order of operations for performing that particular process. Now, the manufacturer is also required to document the activities completed. The next few slides will focus on the ISO 13485-2016 requirements for validation of process for production and service provision. Clause 7.5.6 requires validation of any processes for production and service provision where the resulting output cannot be verified or is not verified by subsequent monitoring or measurement. This is very similar to the QS regulation requirement. The validation shall demonstrate the ability of the process to achieve planned results consistently. Again, very similar to the regulation. The standard requires the documentation of procedures for validation of processes and includes a specific list of activities to include in the procedures. That list includes the requirement for defining criteria for review and approval of processes, as well as the qualification of equipment and personnel. The procedures shall also include use of specific methods, procedures, and acceptance criteria, and the use of statistical techniques as appropriate. The rationale for sample sizes used shall also be included. Additionally, it shall include requirements for records, revalidation, and criteria for revalidation, and finally include approval of those changes to the process. The records shall be maintained according to clauses 4.2.4 and 4.2.5. Now, this is an example of how clause 7.5.6 is linked to other clauses in the standard. Clause 7.5.6 also includes requirements for software validation. Now, as mentioned earlier, this presentation will not focus on those requirements. Next, clause 7.5.7 requires documented procedures according to clause 4.2.4 for the validation of processes for sterilization and sterile barrier systems. These processes shall be validated prior to implementation and following product and process changes as appropriate. It requires the organization to maintain records of the results, conclusions, and any necessary action from the validation according to clause 4.2.4 and 4.2.5. Additional information can be found in ISO 11607-1 and ISO 11607-2. Before we continue, let's do a knowledge check. So, what are the benefits of validating processes? Is it A, for fewer recalls? Is it B, have more consistent quality? Is it C, both A and B? Or is it D, more scrap and rework? Well, the correct answer is C. It is both A and B. I listed both of these as a benefit for validating processes. It results in fewer recalls due to product meeting specifications or regulatory requirements, and validation provides a high degree of assurance that the process will consistently produce product or processes that meet specifications. When you validate your process, you will have less scrap or rework product, not more. So that's why option D was not correct. We've indicated that the process validation requirements of the Quality System Regulation and the ISO 13485-2016 standard are harmonized and that there are no conflicting requirements, but there are several similarities and differences. The next few slides will list those similarities and differences. The Quality System Regulation and the ISO 13485-2016 standard are generally similar regarding process validation concepts. Overall, they both require processes to be validated with a high degree of assurance where the results of a process cannot be fully verified by subsequent inspection and test. Examples of when a process cannot be fully verified were provided in the previous slides. They also both require approval of the validation process and require the validation results and activities or actions to be documented or maintained. Overall, the requirements are very similar. In addition, both the regulation and the standard require changes to the validation process to be addressed. The QS regulation specifically requires the review and evaluation of the process when changes or deviation occurs. The standard requires procedures to address approval of changes to the process. So they both require revalidation where appropriate based on process evaluation and the criteria for revalidation. So they both also address revalidation where appropriate. So although both the regulation and standard have similar process validation concepts and requirements, there are a few specific differences in the requirements, but only a minor or moderate effort would be needed to meet both sets of requirements. The QS regulation has explicit requirements for monitoring, controlling, and documenting validated processes during routine production to ensure specified requirements continue to be met. It also has explicit requirements for having qualified operators perform the validation. Although the standard does not explicitly state these same requirements, it does require similar concepts, such as requiring qualification of personnel and requiring criteria for revalidation. The standard differs from the regulation in that it requires a list of specific items that shall be contained within the validation procedure. For example, it requires you to list the defined criteria for review and approval of the processes. It requires you to list approval of changes to the processes and equipment and personnel qualifications. These all should be included in the process validation procedure. Although the QS regulation does not specifically list items to be included in the validation procedure, the regulation includes some of the activities listed to be performed and requires documentation of those activities completed. So therefore, a minor to moderate effort would be needed to comply with both the regulation and the standard. The standard differs from the regulation in that it specifically requires procedures for the validation of sterilization and sterile barrier systems, that they are validated prior to use and following product or process changes as appropriate. The standard also requires records of validation results, the validation conclusions, and any necessary actions. The regulation does not specifically require this. The preamble clarifies that sterilization is a process that requires validation, and therefore it would require only a minor to moderate effort to ensure both the regulation and standard could be met. Although there are differences among the regulation and standard, implementing the different requirements would need only a minor to moderate effort to comply with both. This is mainly due to the close similarity of the requirements and of the general process validation concepts, as well as the ongoing harmonization efforts. Let's do one final knowledge check. So what is one example of a similar process validation requirement of the QS regulation and the ISO 13485 standard? Is it A, list specific requirements in the validation process procedure? Or is it B, require validation of a process where results cannot be fully verified? Or is it C, both A and B? Well, the correct answer is B. They both require validation of a process where results cannot be fully verified. For example, when destructive testing is necessary to demonstrate the process produced product that meets specifications. Only the standard lists specific requirements to be included in the validation process procedure. Therefore, option A was not correct. I thought it would be helpful to do a quick walkthrough of a validation process when proceeding to ensure the requirements of both the regulation and the standard are being met, now that we've seen some of the similarities and the differences. So here's an example. Let's walk through this one. Company A manufactures blood culture bottles. The blood culture bottles are filled with a liquid solution. Company A has a validation procedure in place for using this filling process that meet the requirements of ISO 13485 2016 standard for clause 7.5.6 for validation of processes for production and service provisions. The first step you would do to determine what is needed to also comply with the QS regulation is to perform a gap analysis of your validation procedure for your filling process. You would analyze and compare the requirements of the standard and the regulation. If it was determined that there were requirements not specified in the filling validation procedure that are required to meet the QS regulation, then the next step would be to revise your current filling validation procedure to include those requirements. And at a minimum, you would need to include the following QS regulations that are not specifically required in the ISO standard and that we pointed out as a difference. So those requirements would be to include a requirement to include the date and signature of the individual approving the filling validation, the requirement to monitor and control the filling process parameters for the validated filling process at designated intervals and frequency. You would be required to include the requirement to have qualification requirements for the individual performing the validation. Include requirements for the monitoring and control method and the data for the validated filling process, as well as requirements to include the date and validation, the date the validation was performed. Finally, you would need to revise the current filling validation procedure to include requirements that include the individual performing the validation or the major equipment used, and requirements to document all of the validation activities. So as you can see, after you've revised your procedures, then you would train your appropriate employees on the revised filling validation procedures and thus implement that new procedure to revalidate your process as appropriate. So hopefully this example demonstrates how it's easy to navigate those differences. Here is a list of resources that were referenced during this presentation. This slide lists some additional references that were also referenced during the presentation. So in summary, process validation establishes objective evidence that the process will produce desired product or results when operating within specified limits. There are several benefits to validating processes, including having fewer recalls, reduced scrap, and rework. And process validation requirements of the Quality System Regulation and ISO 13485 2016 standard are harmonized, meaning there are no conflicting requirements, and while there are some similarities and differences, there are more similarities than there are differences, and thus the effort to comply with both the regulation and the standard is minor to moderate. Thank you for joining me for this presentation. Now, I will take some of your questions.
Thank you, Tanya, for that great presentation. We'll get right into your live Q&A. And the first question's already in. So how many samples, lots of devices should be tested during process validation activities?
Thank you, Joe, for that question that we oftentimes receive frequently. So the short answer is it depends. There is the regulatory requirement of 21 CFR 820.250, where the quality system regulation requires that when you are conducting sampling or using a sampling plan, that it should be based on a valid statistical rationale. So therefore, a statistical technique should be used when you are determining the number of production runs and sample sizes. So there's really no finite number or no specific number for whatever process you are validating. So when you're trying to determine what that number is, or what the number of lots that you should be validating, you want to make sure that it is statistically based. That requirement is also similar to what ISO requires in clause 7.5.6. It too indicates that your validation processes should include statistical techniques when you're deciding your sample size. So it should be statistically based.
No, thank you for that. And we at FDA have our own statisticians. I know I'm not a statistician. I know some statistics, but I know where to find statisticians. But yes, definitely look for a statistical based aspect. From that, it builds off to our next question. What is the most common standard? So they might be talking about statistical standard here. They don't say, but what is the most common standard for selecting an adequate number of samples during process validation?
Thanks, Joe. Another great question. Here, I can't give you a specific standard, but I can refer you to the GHTF process validation guidance document, where they list several statistical techniques. So it really depends on the criteria that's being used in your process that you're trying to validate. So there are several examples listed in the Global Harmonization Task Force process validation guidance document. That should be helpful.
Nope, and that's spot on, especially with what are you sampling for. So there's different ones that are noted in that guidance document. ANSI ASQ has some different sampling tables, but as Tanya pointed out, what is it that you're trying to sample for? So please look at those references and those resources that are available to you. So our next question. Are process validations requirements necessary to be met during non-clinical and preclinical activities?
So here again, if you want to ensure that the process that you are using provides the results and the product that you intend, I would highly suggest that. As far as the regulatory requirement, it's not required until you decide that you are going to move forward with marketing this particular product. So again, it's strongly recommended, and you want to make sure that the processes you use are producing product that meets the specifications.
Yep, and remember, this dovetails with your design control requirements, because if you're not using manufacturable process, you have to go back and show how what you are using during design validation meets those manufacturable criteria. So you'll make that decision, but remember how this also dovetails with your design control requirements. So our next question, if a process can be fully verified, is validation of this process required?
Okay, that's a pretty simple one, as both the regulation and the standard states. If the process can be fully verified, then validation is not required. But remember, there will be strict documentation that you will have to provide to demonstrate that, and you have to include all of your conclusions and the rationale for the conclusions that you have arrived. So yes, for both the regulation and the standard, if you can demonstrate that it can be fully verified, then validation is not required.
And our next question, are we required to use separate procedures for the QS regulation process validation requirements and the ISO 1345-2016 process validation requirements in our quality system?
Oh, thank you for that question. That's an excellent question, especially with the possibility of moving forward to revisions to the quality system regulation in referencing the ISO 13485 standard. Currently, there's no requirement that you have to have separate procedures. You can have one procedure that addresses all of the regulatory requirements for both the regulation and the standard, or whatever other regulatory documents that you are complying with. So the answer to that is no, you do not have to have separate procedures. I would, in fact, think it would be a great suggestion to have one set of procedures that address all of those requirements. That way, in my opinion, it will make it easier for the individuals or the qualified persons who are actually conducting those requirements. They don't have to go back and forth to different procedures to ensure that all of the requirements are being met. Thank you for that question.
And our next question, can you clarify the difference between process validation and process verification?
Oh, wow, another good question. We do get that one often. You know, what is the difference between process verification versus process validation? So I think of it like this: with verifying, you're trying to just make a quick check to make sure that all of your specifications were met. You know, did I build the product right? You know, did I make it blue as it was supposed to? Is the product 3 inches long? Those are the types of verification activities. Whereas process validation, you want to make sure that you made the right product. You know, was the right product built? Meaning, did it meet the specifications, which include the intended use of that particular device? So one, verification, you're just trying to make sure you built the product right. The other one, validation, you want to make sure you built the right product. Okay, I hope that little analogy helped you to differentiate those two different activities.
Okay, thank you. And our next question has to do with revalidation. How often are we required to revalidate our processes?
So perhaps many of you already know FDA does not tell you exactly what to do, and in this situation is no different. We do require that you assess, and you have to document when revalidation is required. There are some requirements that when you make changes to your process or any of the products or the materials that may be used in your process, then you need to evaluate to determine whether or not it requires revalidation. One scenario would be, you know, would I still be able to produce the results that I intend, that I set out based on my predefined or predetermined specifications? And those criteria for revalidation, you want to make sure that you draft and develop and document those in advance already. So when the time occurs or take place, you already have those decision points defined and determined. So no, we don't tell you. You actually have to determine when your particular process that's used for your intended use will require revalidation, as well as the frequency. Now, of course, we do review that to make sure that it is in line and will support what you intended to support.
Okay. With that, that's our last question of the session. I'm going to turn it back over to you, Tanya, for your final thoughts on process validation.
Oh, okay. Thank you, Joe. So what I would like to encourage you all, and my call to action would be, make sure that you have procedures in place and that you validate processes that are required to be validated. For instance, those that you cannot fully verify, as well as those that FDA and other regulatory authorities have indicated require validation, such as sterilization processes and welding processes. So I would highly encourage you to make sure you draft those procedures. And then the second thing I would encourage you to do is to begin with performing a gap analysis. If you decide that you want to comply with multiple regulatory requirements in different countries, in the FDA or other regulations, I would suggest you begin with a gap analysis and to determine what part of your procedure or process you are missing and which there is an actual gap, and then identify those and then make sure you make the decision as to whether or not you would need them to comply with the regulatory requirement that you are complying with. All right, and thank you all again for taking the time out to listen to the presentation. I hope it has been helpful. And again, please feel free to contact DICE at fda.hhs.gov if you have any additional questions. Thank you.
Okay, thank you, Tanya. And with that, we're going to move on to our last session of the morning here being Eastern Standard Time in the US. And Brittany Franklin's going to talk to us about remote regulatory assessment, or RRAs, a very important topic, something that we've seen a lot of since the pandemic's beginning, and something that seems like it's going to continue on as things kind of come back or are closer to normal. So Brittany Franklin is a consumer safety officer in the Medical Devices Radiological Health Operations, or OMDRHO Division 2 of the Office of Regulatory Affairs, or ORA. That's basically the field. Brittany joined FDA in 2014, working in the Chicago District Office as an import investigator. She then transitioned to solely conducting inspections related to medical devices and combination products and became a medical device investigator. Due to her expertise in the field, she has also become a member of the dedicated foreign cadre for conducting complex medical device inspections and international inspections, which is a pretty high level of inspectional skill, and also is training new investigators and sits on a panel to provide educational resources to industry to fulfill the agency's mission. And everyone knows the agency mission here is to promote and protect the public health. So with that, I'm going to turn it over to Brittany so she can present on RRAs. Thank you, Brittany.
Thank you so much for that introduction. Hello, everyone. My name is Brittany Franklin, and I'm a Consumer Safety Officer located in Chicago's District Office, which is considered Division 2 of the Office of Medical Devices and Radiological Health. Today's topic will be remote regulatory assessments for medical device facilities, also known as RRAs. So let's jump right on in. So as we all know, everyone had to adjust and adapt when COVID-19 hit. So let's take a moment in our brains to think about what that may have looked like. So maybe this is the first time you've ever worked remotely. Maybe you had to find a new sleep routine because binge watching shows became the new norm. Maybe you had to adjust to being at home with your roommate or spouse 24 hours a day, 7 days a week. Or maybe you just had the pleasure of learning third grade math to assist your child with e-learning. Some or all of these examples may resonate with you. So just like you all had to adapt to the changes that came with COVID-19, so did the agency. Would you like to know how FDA assessed facilities during the COVID-19 pandemic? I'll give you a moment to think on it. So if you answered yes to this question, then stay tuned because this presentation is for you. So FDA had to adapt its business operations to provide the necessary oversight of regulated products while mitigating the spread of the COVID-19 virus. One of the tools utilized are remote regulatory assessments, also known as RRAs. The agency piloted this program during the pandemic and deemed it a necessary tool to assist with our regulatory oversight. So it is here to stay. And since it's here to stay, I'll be giving you a little more insight on the topic. So today's learning objectives for the presentation will be to describe an RRA, identify FDA's jurisdictions to conduct an RRA, review the process of conducting an RRA, and list RRA benefits and additional resources. So the million-dollar question of the day is, what is a remote regulatory assessment? Essentially, it's an examination of an FDA-regulated establishment and their records. It is used to evaluate compliance with the laws and regulations administered by FDA, and you've guessed it, it is conducted completely remotely. So the purpose of remote regulatory assessments are to assess and verify information submitted. So for instance, if you have a pending pre-market application or license, such as a 510K or a biological license application, the agency may use this tool to review documents to ensure the application is reviewed in a timely manner. The agency may also use this as a way to follow up on compliance activities or to ensure that a firm is adhering to regulatory enforcement actions such as a consent decree or injunction. So because each commodity falls under different regulations, which has different record review requirements, you might hear these terms used interchangeably within the agency to describe remote record review. So you might hear remote interactive evaluations, you might hear remote regulatory reviews, and remote regulatory assessments, which is specifically for medical devices. Next, we will jump into the jurisdiction for the agency to conduct remote record review and some of the commodities and their requirements. Some record reviews are voluntary and some are statutorily authorized. Here we will discuss some of those examples. Section 805D of the Federal Food, Drug, and Cosmetic Act, also known as the Fdnc Act, statutorily authorizes that foreign supplier verification program participants, usually food importers, are required to participate and provide all requests for records or other information for imported food products. These requests will be made on official FDA forms. Section 704A4 of the Federal Food, Drug, and Cosmetic Act also statutorily authorizes that all manufacturers of drugs and drug combination products participate in and provide all records for request or other information applicable to drugs and drug combination products. Section 704A1 of the Fdnc Act governs requests for records as it relates to medical devices. Here, the regulation states that record review or request will be in conjunction with FDA's authority to conduct inspections, and therefore it is completely voluntary. Unlike the previous two examples, no forms for medical device record review requests will be made. So let's talk to a little why the agency would conduct an RRA. If FDA cannot physically conduct an inspection, an RRA may be conducted. So as we saw during the pandemic, no one was allowed to travel. So travel restrictions might inhibit us from conducting inspection, and an RRA might be an applicable tool. In the event that your firm has had an 483 in the past, we might use this tool to review any applicable documents sent in to the agency to determine the prioritization of an inspection. In the event that there is time-sensitive information that needs to be evaluated, an RRA might be a useful tool to ensure timely responses. So now that you know a little bit more about RRAs, I'm going to complete a quick knowledge check. Are medical device RRAs considered statutory or voluntary? Think about this question for a moment. If you guess voluntary, you're absolutely correct. So this means that you can decline or withdraw from participation at any time throughout the assessment. Now that we know the participation in the RRA process is completely voluntary, let's pretend your facility has been selected to participate. The agency will use a risk-based approach when determining the selected facility. This is very similar to how an inspection work plan is adopted. Let's review the remote regulatory assessment process. So the process of conducting an RRA starts with a simple request. An FDA staffer will contact the establishment's point of contact. So someone like a CSO or an investigator will make the initial request or invitation via email on official letterhead. At this point, the establishment agrees or disagrees to participate, and because this is voluntary, if you disagree, there is no penalization. It will just be noted in your establishment history file. So for the sake of the presentation, we're going to pretend you guys have agreed to participate in the RRA. So we would arrange virtual meetings and schedule interviews, and this would be important for foreign entities because time zones are a factor. We would need to establish a secure remote connection, which might be through Zoom or Teams. The CSO would then provide an RRA handout to give you any information that might be helpful to why we're conducting the assessment. And there will be no issuance of FDA Form 482, a notice of inspection. So at any point, FDA may terminate the RRA and instead perform an inspection or use other available tools if the virtual connection is not strong enough. So next, we will request records or other information. This might be reviewing SOPs, such as the CAPA procedure or design control procedure. We might review quality records and documents, such as lab records, validation records, maintenance records for your machinery. We might also identify and review source data. We would also review processes and/or electronic systems, such as a Salesforce, which might be used for complaint handling. So as we're requesting these files, we would offer you a transfer system that might be best suited for your facility. So emails might be applicable, which is completely acceptable during an RRA. Box.com might be used for larger files, and then Electronic Submission Gateway, which is a web trader, is also a useful tool. So at this point, the CSO will review requested records and other information. So they might be reviewing to determine compliance with current good manufacturing practices. They will be identifying if corrective actions have been mitigated from previous inspections. And they could provide updates to observations and other outstanding quality issues identified at your facility. So to finalize the RRA, the CSO would schedule a closeout meeting. This might mean inviting your upper management, such as your chief operating officer, such as your management representative. A CEO might even be present. This is where you would discuss record review with management and any results that came from the review. And at this point, no FDA Form 483 observation list will be issued. That's right. No FDA Form 483 will be issued during an RRA. So after the assessment is completed, there will be a written narrative report generated. The agency will determine a final outcome of the RRA, and at the end of their conclusion, they will make a redacted report available for public disclosure. At this point, no FMD 145 will be sent out, like with an inspection. In order to retrieve the report, you must put in a FOIA request. So this concludes the process of conducting an RRA. Hopefully gives you a general gist of how an RRA will be conducted. So next, I will identify some additional benefits and resources of participating in an RRA. So as mentioned before, this will allow an establishment to correct quality issues outside of an inspectional framework. So if we're looking at 43 points from a previous inspection, we can give you feedback in real time about that correction. This could potentially decrease future inspection time, And as mentioned, it will help us evaluate your adherence to regulatory enforcement actions. This could also assist with assessing pending pre-market applications to ensure efficient retrieval and review of associated documents. This can support the agency's oversight on a firm's compliance, and it can also help determine prioritization of inspections or follow-up activities for an establishment. And the biggest benefit of them all could be that it will help the agency continue to protect the public health by assuring the safety, effectiveness, and security of medical devices and radiation-emitting products. And just before we finish the presentation, here are some additional resources that might be helpful in the future. The Office of Medical Products and Tobacco Operations has an RRA fact sheet that will be provided. It is hyperlinked here for your reference. The Office of Medical Devices and Radiological Health Operations Form Remote Regulatory Assessment handout is also hyperlinked here. Lastly, the agency is in the process of drafting a guidance for remote regulatory assessments, and that will be available in the near future. So before we leave today, I have another quick knowledge check. Which FDA forms will be issued during involuntary RRA? If you've been listening at all, this should be pretty simple. Take a moment to think about it. So is it A, a notice of inspection, an FDA Form 482? Is it B, a list of observations, FDA Form 483? Is it C, both A and B, a notice of inspection and a list of observations? Or is it D, none of the above? So if you guess none of the above, you are absolutely correct. There will be no issuance of a 482 or a 483 during an RRA. So in summary, we've learned that medical device RRAs are completely voluntary. There will be no FDA Form 482 or 483 issued during an RRA. And one of the most beneficial reasons an RRA should be conducted or considered is that it gives FDA the oversight without having to physically be on site or present at your establishment. So thank you for joining me today, and if you have any questions, please feel free to ask them now.
Thank you, Brittany, for that great presentation on fairly new program that's going on. So definitely a very engaging and timely topic to be talking about. With that, we've already got our first question in, and actually several questions are very similar to that. And it goes, are remote regulatory assessments considered inspections? So are they inspections? And then there's a second part to this question once you answer that part.
Hello, everyone. So simple answer, no, RRAs are not inspections. They are tools used in conjunction with the traditional on-site inspection. And because no 482 will be issued, which is usually our official start of an inspection, we will just say the answer is no. These will not be considered inspections at all.
No, thank you for that answer. And if you can highlight a little bit more, I know you talked about this throughout the presentation, the differences between a remote assessment versus an on-site inspection.
So, good question. So some of the differences, so of course, an inspection will start with a 482, and if there are any observations, a 483 will be issued. However, for an assessment, a remote regulatory assessment, neither of the two will be issued. So a 482 will not be issued, and a 483 will not be issued. Some of the similarities are the request for records. Most records or all records are on limits or considered within our jurisdiction to ask for. However, as it relates to an inspection,
when you're looking at internal audit documents, we would not look at those in an inspection, and that would still apply for a remote regulatory assessment. Of course, it's remote, so a RRA would be remote and a physical inspection would be on site. But those are some of the key differences and similarities that come to mind.
Okay, thank you. And I'm going to bundle a couple of these questions together because they all seem to be the same sort of theme, which is RRAs are voluntary, so the remote regulatory assessments are voluntary. However, if something is found as part of the RRA, will that then make for a mandatory visit of FDA in an inspection? So you have the RRA, but if something happens, does that then trip, a mandatory inspection to take place?
Good question. So within an RA, there will be a follow-up, or there will be a closeout meeting. Let's go with that. And at that moment, the firm will have an opportunity to provide feedback to the to the agency within 15 business days, just as you would with an inspection. So that information will be reviewed along with the report, and compliance will then make a decision. If the agency finds that information or anything in the report egregious, or if they determine it's high risk, then that can absolutely trigger an on-site inspection. Did that answer the question?
Yes, it does. That's perfect.
Perfect.
So the next question that kind of builds off of that, if a manufacturer refuses, so you've noted these are voluntary, if someone refuses, would that trip in causing FDA then to perform an on-site inspection?
So because it is completely voluntary, it could lead to an inspection, but because it is completely voluntary, it will not be considered a refusal on the firm's behalf, and it will not be noted as requiring a disciplinary action because it was refused. However, this can lead to an inspection because it will now be on the work plan.
Thank you. And our next question more has to do with the means of the RRAs. So what are acceptable means of virtual connections for the RRAs?
So as mentioned during the presentation, the agency or the investigator will then, set up a meeting, and usually we'll use Teams or Zoom. And as mentioned, emails can be utilized to chat, or you may use the chat function within those systems. I know usually emails are not utilized during an inspection to provide documentation, but during an RRA it is completely acceptable, and we accept those type of transfers.
Okay. And the next question pertains to records. So I'm going to ask it because there are several of these I'm bundling again together. Will the FDA ever request minutes or reports of a firm's management review meetings, or a firm's internal or supplier quality audits, or are there any records that are off limits when participating in an RRA?
So as mentioned, there are no records generally that are off limits. There are limitations due to production records, or limitations to actually reviewing a production process because we're not physically on site. However, in lieu of that, some firms may provide a video for production activities. We might see those type of records being provided. However, we will not be reviewing internal audit records or management review results. But if they are in like a CAPA form, then they might be reviewed. However, it is not our usual practice to review internal audit results or management review results.
Okay, perfect. And next question is, can sponsors request an RRA, or is this determined by FDA? So I guess the question here is, if somebody was coming up on their routine inspection, could a sponsor request an RRA?
So the answer to that question is simply no. The agency determines based upon our work plan, the risk of the device, and things of that nature to decide who can be eligible for an RRA. We do not accept requests for RRA at this time.
Okay, good to know. Does the RRA report require a response? You talked about this a little in a previous portion. And if so, could a warning letter be issued for failing to address deficiencies identified during an RRA?
So for the first part, we do not necessarily require you to respond to any deficiencies or discrepancies noted in an EIR or any narrative. You will have the opportunity to respond to these discrepancies within 15 business days of receiving. Well, you won't receive a 43. So at the close of the assessment, that is when your 15 business days start. Very similar to an inspection. However, you are not required, and since you're not required, there won't be any information to support the EIR on your behalf. So it would be, of your best interest to respond. However, this may be reviewed to lead to a warning letter or lead to an untitled letter if anything egregious is found. However, that would be solely to the discretion of our compliance team.
Okay, thank you for that. The next question has to do with lead time. So there's currently the lead time for an inspection. What is the lead time for RRA notification? And is there a difference between domestic and foreign lead times?
So the common lead time is 5 days. So we will make the request within 5 days of the invitation to start. And that will be, as stated before, sent on a letterhead stating we are requesting, to invite you to participate in an RRA. And then at that point, you will, decide yay or nay, and we will then move on from there. So it is a general 5-day time frame prior to the start.
Okay, great information to know. And building off of the foreign piece of that, is ORA conducting RRAs for foreign medical device manufacturers?
So we have conducted some foreign RRAs. We don't see that as a common or usual practice for moving forward with foreign RRAs because we are hoping to physically visit, given that the time frame and the frequency is a little longer or a little, far and few between, if you will, for foreign entities. So we are utilizing them, but our hopes are to be at those facilities a little more frequently than utilizing the RRA tool. But it is not off limits for foreign facilities.
Okay, and to build off of that, are interpreters needed for foreign RRAs?
So we do expect that there is someone at the facility who can interpret documents into English, or if we have non-English speaking personnel or staff on the call, someone can facilitate translation while conducting the RRA.
Okay, thank you. And this builds off one last one with regards to the foreign piece of this. What enforcement actions, if any, and I'm going to read into this, import detention, can be taken following an RRA? So I'm a foreign manufacturer. Is there enforcement actions that can be taken because of the RRA, and what would those enforcement actions be?
That is a question that we're going to have to follow up on. I'm not particularly sure. I don't know at the moment. I will say that, and we will get back with you on that. We can provide that information.
Perfectly acceptable, as we're into some new territory here with some really
we
good
are.
questions. So, Next question. Can we consider using a remote regulatory assessment as a tool to prepare for an on-site inspection? So this is where it sounds like the firm's looking to use the RRA to their advantage to get ready for their on-site inspection.
I would say it would probably be a good, useful tool. If you've never been inspected, this may be in preparation to prepare for an on-site actual audit. However, please note, as stated before, that it is something that we will request, so you won't be able to request it from us. But if chosen, it could be a useful tool to prepare for an on-site audit. And I'm not sure if I mentioned this, but this will give you a chance to answer or ask any questions outside of a traditional inspectional framework, and you won't be penalized for asking questions during the assessment.
Okay, that is really good to know. And this has to do with the length of time. So we talked about how much lead time to go into one. Usually, how long does it take for one of these remote regulatory assessments? So how long do these assessments take if you're the manufacturer?
So we have allotted about 2 weeks time frame, so 14 days. That can be about 3 weeks if we're speaking business days. And it can be prolonged if, egregious discrepancies have been noted. And if that's the case, then the staff will be notified about the extension of time if it goes beyond that 14-day time frame, or 15-day time frame, I'm sorry.
Okay. And our next question, what if requested documents are not readily available? Can those be provided later, say on the next day?
Absolutely. So because this is a process that may take up to 2 to 3 weeks in business time, we have the full time frame to utilize the retrieval of these documents. But please note, it is a continuing effort to keep it moving forward. So as requested, please try to get them in as they become accessible or readily available.
Okay, and speaking of provided documents, the next couple of questions have to do with Freedom of Information Act. So first, are RRA documents FOIAble? So can people look through the Freedom of Information Act to get documents as they relate to the RRA or findings from the RRA or observations?
Yes, so that's a good question. So at the conclusion of the RA, the narrative report or EIR will be written, and the only way to retrieve that is via a FOIA request, because there is no FMD 145 or no correspondence from the agency to the firm. And so everything related to this assessment will have to be FOIA'd. And that is a usual, we say that the time frame to retrieve that is about 90 days for it to be available within our system. So yes, these documents can be FOIA'd. They will be redacted for all, personal information or any, proprietary information. It goes through our usual FOIA requested process.
Great, you answered the next two questions, which was somebody was asking about time frame, and then the secondary part that yes, it'll go through the regular process of redaction. So good information to know. Okay, and our next question, are these emails that are part of the RRA, are they considered records themselves?
Yes, they are considered records. So if you're submitting them and identifying, information within that email, they can for sure be utilized as records if necessary.
And our next question, what is the selection criteria? So what criteria for selection is FDA using for manufacturers to be selected to have an RRA versus an on-site inspection?
So currently we are looking into the lower risk firms and facilities. So that's usually like class 1s and class 2s. We are also taking a risk-based approach, looking at firm history, compliance history, all those things will be taken into consideration when being chosen. But we are currently looking for class 1, class 2s, and mainly surveillance routine assessments, if you will. So those who are less complex, so no class 3s, usually no like follow-up or compliance activities will be completed unless it's a regular like 43 review from a VAI inspection where we're reviewing old observations and working through them. But usually will be class 1s or class 2s.
Okay, thank you. So risk-based approach. Our next question is more of a comparison. How does the RRA compare to, say, QSIT, the Quality System Inspection Technique?
So that's a very good question. Essentially, we will be utilizing the same technique to perform an RRA. It may not be as in-depth because of the time frame and the channels used, but it will take the same approach to conducting the assessment as it would an inspection.
Okay. And this, I know this has come up before, but can an establishment receive regulatory action, such as a regulatory meeting warning letter for quality issues found during just the RRA? So there's no follow-up inspection. Can things go directly to regulatory action?
I don't think it will go directly to regulatory action. I do believe that there will be some assessment prior to going straight to regulatory action, maybe an on-site inspection first or an investigation if necessary, but I don't think it will go from an RRA directly to an untitled letter or warning letter. Compliance will have to do their review, and depending on how egregious, as stated before, there may need to be involvement of Cdrh and all that good jazz. But as of now, I don't think that there is a direct link from an RRA directly to regulatory action. There will be some steps in between.
Okay, thank you. Our next question is there any type of resources such as remote regulatory assessment checklists or other resources and tools available for manufacturers to be able to look at?
So at this moment, we are in the process of drafting, a guidance document with some Q&As that have come up. We have some handouts that are given out prior to each assessment. And we are also in current development of other tools for this particular process. It is fairly new, so we are still in the process of any policies and other additional tools that might be helpful moving forward.
Okay, that may actually answer the next question, but I'll let you answer it as well. So are there going to be updates to the compliance program guidance manual, the CPGMs, or the investigator's operation manual?
I believe so. So there will have to be some adjustments for this new tool. We are still in the process of working out all of the kinks, but presumably there will be some changes to some other documents that may be affected by this tool.
Okay, thank you. And it sounds like it sounds like from what you said earlier, the first thing to keep an eye out for is that guidance document. So
Correct.
That should be something on everybody's radar, is to keep an eye out for that. And then we have time for one last question. How many auditors or investigators participate in an RRA?
So because these are routine and probably lower risk devices, we would usually send one individual for an inspection, and that applies for remote regulatory assessments as well. It is usually one investigator or CSO conducting the assessment.
Okay. Thank you again for the very insightful presentation and this Q&A session. With that, I am going to turn it over to you to give your last thoughts on the topic of remote regulatory assessments. So thank you, Brittany.
Absolutely. So my call to action, or some things that you should look into or be prepared for, firstly, is to determine if the RA process is beneficial for your facility to participate in. Next, if you're going to participate, ensure you have a secure and remote connection and capabilities. I would tell you to gather appropriate staff and personnel to participate, like management, SMEs. I know we all probably are working from home, so calling them in or making sure they're available would be helpful. Ensuring documents are easily accessible electronically. So that means preparing your documents to be submitted via email or via the other channels like box.com. And then lastly, be on the lookout for the final guidance documents that will be released. And that is all that I have. And thank you for participating today, guys.
Thank you, Brittany. So we will be wrapping up our session on the medical devices track for REdI 2022. As we've talked about throughout the last day and a half, please complete the survey and evaluations. Your feedback is very, very important to us, and it helps to develop the future educational programs such as REdI. So if you want to see new topics for 2023, and we always think about REdI 2023 and eventually REdI 2024, please fill out that information in those surveys and evaluations. So thank you again for giving us your valuable time. We hope that this program was useful to you and that you learned something new. I know I did. In almost every session, I learned something new. These were great topics, very timely topics with a lot of great information. And with that, I want to thank all the presenters and the participants that made this happen. Their hard work and knowledge is much appreciated, because without them, we couldn't put on these programs. I have a certain level of knowledge, but not nearly the breadth and depth of the knowledge of the different experts we heard from over the last day and a half. Absolutely amazing. I don't think you could get this type of insight and information anywhere else. So definitely appreciated everything that the presenters and the participants did. Their hard work showed through completely. And also, I'd like to give a special thanks to Elias Malice and Brenda Stoddard, whose very key leadership is critical in making this annual program such a success. So there's a lot of planning and work that goes in, all the way from the plenaries to each of the tracks, to each of the sessions, to each of the presenters, and helping them with their materials and putting these presentations together. And I don't know how it would be done without Brenda and Elias. They do such a great job. Their knowledge of both the overall aspects of what they understand in the programs, to being able to speak and present themselves and to moderate sessions is amazing. So I definitely give a big thank you to both Elias and Brenda. And they make such a great success of this program. Okay. And last, please note that REdI 2022 is not over just yet. So come back after lunch for the start of the biologics track with presentations by the Center for Biologics Evaluation and Research, Cdrh. So we're not done just yet. We are finishing up for the medical devices track, but we also have another half a day and a full day tomorrow to talk about biologics. So we started with the plenary on Monday. We talked about drugs and Cdrh for a day and a half. Then we did the devices track, and next we're going to go into biologics. So enjoy the rest of this program, and I hope to see you all soon at our next DICE educational event, be it webinars. Webinars are going to be several of them scheduled throughout the month. We have a standing in vitro diagnostic COVID-19 webinar town hall that happens every other Wednesday. So please reach out and hit our different educational events, and I hope to hear and see you soon at them. And feel free, if you have any questions, to reach out to DICE. We are here to help you, so please contact us by phone or email, and then use those educational resources that we talked about throughout the last 2 days, Cdrh Learn and Device Advice, and link up with us and get communications and information directly from FDA by subscribing to our mailing list. Cdrh New is a great mailing list to be on. You'll get everything as it relates to the center. And visit us on our social media platforms, be they Twitter, Facebook, or LinkedIn, to get more information. Also, we have a YouTube channel. Some folks are listening in on YouTube right now. Please visit that YouTube channel. And there's actually a device playlist for those basic playlist videos that are on Cdrh Learn that are on YouTube. So please use all those resources that are available to you and do what you need to do to make sure that you have safe and effective devices that work as intended, so that we are all on the same page on promoting and protecting that public health. So thank you, and have a good lunch and a good rest of REdI.
Good afternoon to all of the participants and attendees of the Regulatory Education for Industry Annual Conference 2022. It is a great pleasure to welcome everyone to our third track of the conference, which will be filled with presentations from the Center for Biologics Evaluation and Research, or CBER. My name is Larissa Lapteva, and I'm the Associate Director in the Division of Clinical Evaluation, Pharmacology and Toxicology in the Office of Tissues and Advanced Therapies in CBER. Together with a team of moderators whom I will introduce in a few minutes, we will be hosting the CBER track, welcoming our speakers and faculty, moderating sessions, and fielding questions from the audience. As a reminder, the CBER track will run today and tomorrow before we conclude the conference at the end of the week. Many of you in the audience probably know that Center for Biologics regulates a large variety of biological and related products, including blood and plasma-derived products, vaccines, allergenics, tissue-based and tissue-engineered therapies, products intended for xenotransplantation, and, of course, cellular and gene therapies, as well as devices used with biologics. Together with the Center for Drugs and Center for Devices, CBER is delighted to again participate in this educational conference event organized by the Small Business Industry Assistance Program. In the beginning of the week, during the plenary session, you heard the recent developments related to COVID vaccines, treatments, and diagnostics, many of which were spearheaded by the CBER's Office of Vaccines Research and Review, which continued its tireless work to combat the pandemic with vaccine approvals, issuance of emergency use authorizations for the use of booster doses and expansion to pediatric age groups, and all of this while collaborating with stakeholders in the United States and other countries to ensure access to the life-saving vaccines and to continue evaluation of the scientific knowledge and clinical data accumulated with the use of the approved vaccines to ensure their safety and effectiveness. But besides the pandemic-related work, in the past year, Center for Biologics continued facilitating the much-needed research conducted for the development of many other biological products, including those intended for prevention and treatment of serious diseases and various medical conditions. In the Office of Tissues and Advanced Therapies, we have continued observing a steady influx of new development programs with regenerative medicine therapies, including cellular therapies, gene therapies, tissue-engineered products, and also plasma-derived therapeutics, and many others. And as our scientists review these multidimensional and complex development programs, they see that there are multiple issues which are often similar issues that more than one product sponsor may be encountering, and solutions to which may be applicable not to one but to multiple products of similar design or multiple programs with novel therapies. And as we continuously see emerging and common issues in biologics product development, the CBER speakers today will be highlighting these common problems and recommending potential approaches to enable forward movement for new therapies. All of our presenters in the CBER track participating this year based their presentations on numerous applications which they have reviewed and discussed with product developers. Today, you will hear presentations on a variety of issues common to the product quality and manufacturing, preclinical and clinical development of cellular and gene therapies, and we will be particularly focusing on products developed for the treatment of oncological conditions and rare diseases. We will also discuss best practices for interactions between CBER and product sponsors, because these and many other issues remain particularly relevant and important for smaller companies especially those that transition from academic research to product development and subsequent scale-up manufacturing. We will also cover a number of interesting topics tomorrow, which you will see here on the slide and which I will highlight at the end of today. It was our intention to make the agenda interesting and diverse in order to share CBER's experience and provide as much information as we can, including references to the available regulatory resources, which you can find on our website and which we will be showing you today and tomorrow. As you watch the presentations, please send us your questions through the chat box. As with the two previous tracks, we will have Q&A time at the end of each session. Let me now introduce our moderators for the CBER track. You see them here on the slide, and you will hear their voices throughout these 2 days in the beginning of each session and during the Q&A time with our speakers. Dr. Irina Tipper holds a PhD in molecular medicine. She is currently a team leader in the Division of Cell and Gene Therapies in the Office of Tissues and Advanced Therapies, where she supports regulatory review of regenerative medicine therapies, immunotherapies, and xenotransplantation products. Dr. Graham Price is a research microbiologist and product quality reviewer, also in the Division of Cell and Gene Therapies in the Office of Tissue and Advanced Therapies. He holds a PhD in biological sciences, and his expertise is in immunology and virology. In his regulatory role, he reviews investigational new drug applications and biologic license applications for human cell and gene therapy products, and his work is focused particularly on the chimeric antigen receptor, genetically modified T-cell-based products, and cancer vaccines. Ms. Jennifer Albert holds a Bachelor of Science degree in nursing. She is a registered nurse with experience in critical care and oncology, as well as clinical research in early trials involving stem cell transplantation and immunotherapies for hematological malignancies and metastatic solid tumors. Jen currently serves as the regulatory project manager in Audited, where she manages submissions for novel biologics and devices, and also, importantly, various communications with product sponsors. Dr. Peter Bross is a physician with background in oncology. He has a long-standing clinical and regulatory experience in working with both drugs and biological products, but primarily, of course, with biological products developed for the treatment of different types of cancers. Dr. Bross currently serves as the chief of the oncology branch in the Division of Clinical Evaluation, Pharmacology and Toxicology in the Office of Tissues and Advanced Therapies. So these are our moderators, but before we proceed with the program, I have a few announcements regarding continuing education credits for this activity. In addition to the Certificate of Attendance, which supports continuing education credits for the members of the Regulatory Affairs Professional Society, Society of Clinical Research Associates, Society of Quality Assurance, and Association of Clinical Research Professionals, this conference offers a total of 32.5 continuing education credit hours to physicians, pharmacists, and nurses. The CE credits are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend via Adobe. Unfortunately, we cannot offer CE to those participants who attend via YouTube or who will do post-event viewing. All speakers are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single company. Speakers attest that the content that they contributed to this conference is supported by the best available knowledge and evidence. All speakers are also reminded to disclose when products or procedures being discussed are off-label, meaning that they're not FDA approved, and if there are any limitations on the information that's being presented. CBER speakers who are presenting today have nothing to disclose except Dr. Das, who discloses her previous employment with Progenics Pharmaceuticals, for which all of the relevant financial relationships have been mitigated. Those in the audience who would like to receive continuing education credit, including physicians, pharmacists, nurses, and those claiming non-physician continuing education credit, you must attest to your attendance and complete the final activity evaluations through the CE portal. For multi-day activities, participants must attest to their attendance and complete the session evaluation each day. Final activity evaluations must be complete within 2 weeks or 14 days of the activity. For pharmacists and pharmacy technicians, please note that you will have to provide your correct NABP and date of birth information, and this has to be done in the required format in order to receive credit for this activity. Pharmacists are recommended to log into the CPE monitor 8 weeks after the last session of the activity to ensure that your credit is captured correctly. Each track, and the CBER track is no exception, will have its own survey and evaluation link. Each night, the Small Business Industry Assistance Program will send one email to the attendees of that individual event day. The email will have the information on the CE claim codes. It will also contain the links to the CE portal and the survey link. And I think at this point we are done with the administrative CE related part. And now, finally, let me introduce our first presenter for the CBER track, Dr. Wilson Bryan. Dr. Bryan is a physician and trained neurologist. Prior to joining FDA, he served as an investigator on clinical trials in cerebrovascular diseases and neuromuscular disorders. Dr. Bryan joined the Food and Drug Administration 22 years ago, and he currently serves as the director of the Office of Tissues and Advanced Therapies in CBER. Dr. Bryan will provide an overview and update on the office's regulatory activities and our most recent product approvals.
Good afternoon. My name is Wilson Bryan, and I'm director of the Office of Tissues and Advanced Therapies, or OTAD, at the Center for Biologics Evaluation and Research, or CBER, at the FDA. Now, this slide shows the number of investigational new drug applications, or INDs, that have come in to OTAD, or its predecessor offices, over the past decades. And the part of the graph that is in blue represents what I call research INDs. That is, these are INDs where sponsors are trying to gather evidence of safety and effectiveness to bring a product forward to the market to address unmet medical needs. The part of the graph that is in orange represent expanded access INDs, which are intended primarily to treat patients, not to gather research evidence of safety and effectiveness. The bubble in the first bubble on the left is for 2016 and shows that we received 223 new INDs that year. By 2020, 4 years later, that had increased by 199%. It almost tripled to 666, so a huge jump. And then with the pandemic, there has been a slight decrease from 2020 to 2021. Now, most of these INDs are for serious and life-threatening diseases. That is, over 90% are for serious and life-threatening diseases, and approximately 50% are for rare diseases. A few fundamentals about the office. First of all, the FDA itself has 7 centers, and the 3 centers on the left of this graph are the ones that regulate medical products for humans: the Center for Drug Evaluation and Research, or CDER, the Center for Devices and Radiological Health, or Cdrh, and the Center for Biologics Evaluation and Research, highlighted here in yellow, or CBER. And I also want to mention the Oncology Center of Excellence, or OCE, which coordinates oncology reviews across the centers. Now, in the Center for Biologics, the Center Director is Dr. Peter Marks, and Deputy Director Dr. Celia Witten, highlighted in yellow at the top of this graph. And at the bottom of the graph are the 3 product offices that regulate specific types of products. First, the Office of Vaccines, which has been extremely busy, of course, during the pandemic. The Office of Blood Research and Review, also extremely busy during the pandemic with convalescent plasma, and highlighted the Office of Tissues and Advanced Therapies, or OTAT. And OTAT regulates a wide variety of products, but our mission is really to promote the public health through collaborative, science-based regulation of medical products. I want to emphasize that all of our work has to be based in good science. I'll talk about that a little more in a second. But we're trying to facilitate drug development, ensure safety of individuals. Our regulatory decisions have to be data-driven, impartial, and compassionate. To emphasize our regulatory science, in OTAT we have 21 scientific laboratories that put out, in 2021, put out 51 publications and participated in 47 external conferences. The research is on a wide variety of mission-critical areas, including COVID-19. Our products, again, highly diverse. We regulate purified and recombinant proteins for hematology, such as coagulation factors. We regulate immunoglobulins, antivenoms, snake antivenom, spider antivenom, stem cell and stem cell-derived products in all types of stem cells, hematopoietic, mesenchymal, induced pluripotent stem cells, terminally differentiated cell therapies, such as pancreatic islets and myoblasts, therapeutic vaccines, and other antigen-specific active immunotherapies, cancer vaccines and immunotherapies, non-infectious disease therapeutic vaccines. So if you have a vaccine to treat Alzheimer's disease or a vaccine to treat opioid addiction, that would come to us. OTAT products include gene therapies, including genetically modified cells, and these gene therapies can be administered in a variety of ways through plasmids, viral vectors, or bacterial vectors. We regulate xenotransplantation products. There's been a lot of interest recently with the xenotransplantation of a heart from a pig to a human. Tissues and tissue-based products, and then some devices and combination products, particularly devices that have a cellular component, and selected devices for the manufacture or delivery of cells. I want to talk a little bit about our recent approvals, and the theme here is that our products are for serious and life-threatening disease. As an example of an in vivo gene therapy, Luxturna, approved in 2017. And this is a directly administered adeno-associated viral vector gene therapy that targets a disease caused by mutations in a specific gene, that is in the RPE65 gene that causes retinal dystrophy. These patients develop blindness over the first few decades of life. I want to show a video of a young boy who has this type of retinal dystrophy. And what you're going to see here is he's trying to walk through a maze. And the maze has some obstacles. It's got stop signs. And in this first video, he's using the untreated eye. So let's see if we can bring this up. And you'll see he's looking at the floor. There are arrows on the floor that he's trying to see to tell him which way to go. And he's struggling. He's needing some directions to figure out. He's really stalled here, and it's not going so well for him. Let's see how he does with the eye that was treated with Luxturna. You can see the arrows on the maze. He's got this. He's moving just so quickly through the maze. The huge dramatic difference that Luxturna has made for patients with this form of blindness. I want to talk a little bit about ex vivo gene therapy. And these 6 products on the right are all examples of chimeric antigen receptor T-cell products, or CAR T-cell products. What they do here is they take a patient that has a hematologic malignancy, and they take the blood out and they isolate the immune cells, the T cells, and then they genetically alter the T cells so that they express an antibody that will attach to the tumor. And then they put these genetically modified cells back into the patient, and they attack the tumor and they kill the tumor. And it works. I mean, this is almost like science fiction, but it's science. It's amazing. And these are life-saving drugs. All of these 6 products, each of them works for some form of hematologic malignancy, generally studied in patients that had exhausted all their treatment options. These were patients who were going to die. And so these are life-saving products. I want to say a little bit about allogeneic cell therapies. Stratigraft, approved in just last year in 2021, produced from 2 kinds of human skin cells, grown together to make a cellularized scaffold. And this is for treatment of adults with thermal burns, also referred to as deep partial thickness burns. And if you look at these photos here, look at the top row. This is starting on the left, a severe burn treated with stratigraft, and then by the last photo on the right in the top row, you can see the result a year later. It's a dramatic improvement. Again, a huge advance in therapy for burns. Purified plasma protein, not a cell therapy or gene therapy. Look at plasma protein products. This is Ribosome plasminogen, treatment of an extremely rare disorder, plasminogen deficiency type 1. And these patients have a disorder that can impair normal tissue and organ function and can lead to blindness. This is a photo on the left. You see the eyelid with what's called a ligneous lesion in a patient who has this plasminogen deficiency. And then with, I think the photo on the right is taken 4 weeks later, and the lesion has completely resolved. This is a life-changing drug, really making a difference for patients with rare diseases. And speaking of rare diseases, here's a tissue product, allogeneic processed thymus tissue. This was also approved last year in development for decades. trying to move forward to get this out there for patients. These patients have a rare congenital athymia. They don't have a thymus, and therefore their immune system doesn't work properly. And these kids, most of them are going to be dead by age 2 due to infections. And what they do here is they take some thymus tissue from a donor, and it's transplanted into the thigh muscle of these kids with congenital athymia. And it works. It allows these kids to develop an immune system, and there are patients who have lived for decades now. It's just a life-saving product. So what we're doing in OTAD is we're trying to facilitate development, and I want to look closer at sort of the activity. These are research INDs, the number of research INDs received each year for gene therapies. And going back again to 2016 as a reference, we received 67 new INDs for gene therapy. 4 years later, in 2020, that was up to 161. That's a 140% increase, more than a doubling in 4 years. And then with the pandemic, it's come down a little bit, about 5% to 153. Now, let's see what's happened with cell therapy. Cell therapies, the pattern has been a little bit different. 2016, there were 63. By 2019, it had grown to 91. That's a 44% increase. And then in 2020, there was a dramatic 66% increase in 1 year to 151 new cell therapy applications. And that's a reflection of the pandemic. There's a lot of interest in whether cell therapies or cellular-derived therapies might have anti-inflammatory properties that could help address COVID-19. In 2021, the number has come down some, from 151 to 112. That's a 26% decrease in 1 year. So looking at that, those two together, gene therapy products in the blue line here, you follow over at 2016, it just takes off and increases dramatically up to 2019. And then with the pandemic, it's basically plateaued. In contrast, you follow the red line for cell therapies. 2016, it starts to takes off dramatically in 2019, and then peaks in 2020 and has come down some in 2021. Now, I want to emphasize our efforts to facilitate the development of products, and particularly one way we do that is through a breakthrough designation and RMAT designation request. BTD stands for breakthrough designation, and these are products that have some preliminary clinical evidence of effectiveness, so they get extra attention. And we've been getting breakthrough designation requests since the program started in 2012, and we've granted now 38. I should say that this slide is updated through May 20th. And then RMAT designation, which started later in 2017, 197 requests and 74 granted. So we've now got 74 products and indications with RMAT designation, 38 with breakthrough designation. A few products have both designations, and so we've got right about 100 products that have one of these designations. In summary, the scientific advances have spurred recent growth in research and development of advanced therapies. OTAD is dedicated to facilitating the development of novel products to treat serious and life-threatening diseases. I want to acknowledge the folks who are going to give presentations this afternoon and tomorrow on behalf of the Center for Biologics. These include representatives from our Division of Cellular and Gene Therapies, our Division of Clinical Evaluation and Pharmacology Toxicology, our Division of Plasma Protein Therapeutics, Division of Regulatory Project Management, and the immediate office of the Director. Besides representatives of OTAT, from our Office of Compliance and Biological Quality, OCBQ, we have Dr. Yitmarek, and from our Office of Biostatistics and Pharmacovigilance, we have Drs. Al-Chandani and Obidi. From the Office of the Cdrh Center Director, Dr. Osorio. So really a broad spectrum of representatives for the Center for Biologics. The Planning Committee, I need to recognize the folks who put in all the time in planning these presentations from Cdrh, and particular guidance and analysis from Drs. Anatole, Laptiva, Rousey, and Wang, who helped a lot with my presentation. Now, this is the contact information for OTAT and how to reach us, as well as how to reach Cdrh. Now, one reason you might want to contact us is because we're trying to hire people. As you can tell, we are extremely busy, and we're looking for scientists and folks with clinical experience, physicians, nurses, other clinicians who might be able to help us in trying to move these exciting products forward into hopefully into the marketplace. So we're particularly looking right now for folks with clinical hematology experience, either hematologists or nurses, physician assistants, others who might have clinical hematology experience would help us to review the exciting products such as the CAR-T products and products to treat so-called benign hematology such as thalassemia, sickle cell, and hemophilia. So if you're interested in working for the FDA, this is my contact information. So if you're interested in working in OTAT, please let me know. But if you want to reach me for any reason, this is my contact information. It's pretty straightforward, wilson.bryan@fda.hhs.gov. I'm really looking forward to the presentations from Cdrh over the next day or so, and I thank you for your attention.
Thank you, Dr. Bryan, for a great introduction to OTAT's regulatory activities and the wide range of OTAT-regulated products. My name is Irina Tipper, and it is my pleasure to introduce the next two speakers from OTAT who will present on common product quality issues and preclinical challenges for cell and gene therapy products. Carrie Lorenzo joined the Division of Cellular and Gene Therapies, or DCGT, within OTAT in 2019 and serves as Associate Director for Regulatory Science and Review. In this role, she advises DCGT leadership on regulatory and scientific issues, provides input on development and evaluation of new policies, and on scientific and regulatory programs pertinent to the agency. Dr. Lorenzo obtained a BA in biology from Douglas College, Rutgers University, and a PhD in pharmacology from the George Washington University. She has over 25 years of experience in medical product development in the pharmaceutical industry and U.S. federal government, where she provided regulatory support and advice for numerous gene and cell therapy development projects, as well as projects for other biologics and drugs. Dr. Malek Sunay received her doctorate of philosophy in pathobiology from Johns Hopkins School of Medicine in 2015. She then completed a postdoctoral fellowship at the United States Army Medical Research Institute of Infectious Disease in 2020. In 2020, she joined the FDA as a pharmacology and toxicology reviewer in the Division of Clinical Evaluation and Pharmacology Toxicology, or DCET, within OTAT.
Thank you for the introduction. I would also like to thank the organizers for inviting me to speak today. Development of cell and gene therapy products can be very challenging, and this presentation is designed to highlight the challenges and potential solutions in order to help you successfully navigate quality issues. The learning objectives for my presentation today include recognizing the types of products regulated by the Division of Cellular and Gene Therapies in the Office of Tissues and Advanced Therapies in Cdrh, to recognize challenges associated with the chemistry, manufacturing, and control for cell and gene therapy products throughout product development, and to understand the advantages and challenges associated with expedited product development programs, and to be aware of key FDA guidance documents and other resources for the CMC issues in the development of cell and gene therapy products. The Office of Tissues and Advanced Therapies in Cdrh is responsible for the review of cell and gene therapies. The Division of Cellular and Gene Therapies and OTAT conducts review of the chemistry, manufacturing, and control information for these products. As you can see, cell and gene therapy products are a diverse group of complex biologics. They are often biologically and technologically complex, particularly with respect to how they are derived and manufactured. Examples of gene therapies include ex vivo modified cells, genome-edited cells, plasmids, and RNAs. Examples of cell therapies include many types of human cells, such as T cells, stem cells, and also xenotransplantation products. DCGT also regulates tissue-engineered medical products and some devices, which will not be part of this presentation today. When regulating cell and gene therapy products, OTAT uses a risk-based approach for regulatory decision-making, which includes evaluation of the patient population, disease, product and stage of development in this risk-benefit assessment. Even though precedent can help guide reviewers, a one-size-fits-all approach doesn't apply due to the uniqueness of each product development program. Science, medicine, policy, regulations, and other resources are assessed by FDA review teams when making data-driven decisions. Review at the FDA is highly product-dependent. Product considerations include lot size, manufacturing processes and technologies, product testing challenges, such as little test material or very short shelf life, risk due to source materials, such as using animal-derived materials, which could increase the risk, and product inherent variability, which present consistency challenges, such as products using autologous cells. As previously mentioned, DCGT conducts a CMC review of cell and gene therapy products. CMC includes practices and specifications that must be followed and met in order to ensure product safety and consistency between batches of specific products. During review, we look to see, are all the materials in the final product safe? What is the identity, strength, quality, and purity for your product? What is the manufacturing process? What are the controls and the test methods? CMC expectations increase from early stage development through commercialization. CMC information is required to be submitted to the FDA for investigational products and can be provided either in the original IND or through cross-reference to related INDs or master files. Adherence to GMP regulations assures the identity, strength, quality, and purity of the products by requiring that manufacturers adequately control manufacturing operations. CGMPs refers to current good manufacturing practice regulations enforced by the FDA that provide for systems that assure proper design, monitoring, and control of manufacturing process and facilities. This includes establishing strong quality systems, including identifying and investigating deviations, obtaining quality raw materials, establishing robust operating procedures, personnel training, and maintenance of reliable testing facilities. Flexibility in GMPs allow companies to best implement controls and to modernize. Please note that for phase 1 trials are exempt from the 211 GMP regulations. This allows for greater flexibility for phase 1 manufacturing to comply with the statute. However, manufacturing must still be well documented and well controlled. More info on phase 1 manufacturing can be found in the 2008 FDA guidance document, CGMP for phase 1 investigational drugs. In the IND, you must be able to demonstrate that you can consistently and reproducibly manufacture the product. Product safety issues are the primary concern for early stage clinical development. Some common product safety issues to consider include release assays. Is the methodology robust? Are controls and reference standards included? Methods for sterility and mycoplasm. Are they qualified? Are rapid tests used? Dose measurement assays. Is it the same assay for preclinical studies as for the clinical studies? Is it a qualified assay? Critical raw materials and reagents. What's the source? Is there documentation? Is there a certificate analysis for these products? In the delivery device, what is the regulatory status? Is it investigational? Is it compatible with the drug product? Please be aware that the amount of information to be submitted will depend on the phase and scope of the initial clinical investigation. So if your initial IND submission contains a clinical study whose results are intended to provide the primary evidence of efficacy for licensure, more detailed CMC information will be needed. So what would be the type of information for the later stages of development? For late-stage development, expectations include having a controlled manufacturing process, well-developed and qualified or validated analytical assays, and having sufficient experience to refine product acceptance criteria. Most of the challenges encountered in late-stage development stem from having limited manufacturing experience, including not many lots produced and multiple rounds of process changes. In the preceding slides, we have reviewed some CMC expectations for early and late-stage development, and these expectations can apply to all types of products. Now we will focus on CMC issues unique to cells and gene therapies. Many common CMC issues, such as material qualification, establishment and specification, and major manufacturing changes, can be very challenging during the development of cell and gene therapy products. However, cellular therapy products, whether or not gene-modified, present additional CMC challenges due to the autologous or allogeneic nature of the cells. For example, challenges for allogeneic cells-based product include donor eligibility requirements, cell bank qualification, and reproducibility of replacement cell banks. Challenges for autologous single product lots include product tracking and segregation, high variability product lots, and limited material or time for testing. When moving towards commercialization, there are also specific issues to consider if scaling out or scaling up. For scaling out, you'll need to establish a stringent change of identity, establish and document appropriate segregation, and we also recommend you consider performing a capacity study to identify bottlenecks and ensure that adequate resources are available. For scaling up, you will need to consider that not all processes scale up easily, and adjustments may need to be made. Cells can often be sensitive to cell density, and adherent cultures and suspension cultures may need to have different strategies. One of the paradigms in manufacturing of cell and gene products is the scale of the product lots, which dictates many aspects of CMC expectations for cell and gene therapy products. For conventional biologics, one product lot can often treat many patients. However, for cell and gene therapies, one product lot may treat only a few patients or a single patient. The limited patient use scenarios possess unique challenges for scaling product, comparability assessment, product distribution, and the impacts of manufacturing failures. For example, failure of a single patient lot may dramatically change the course of treatment and disease status for the patient in the case of a serious and life-threatening disease, such as CAR T cells for the treatment of cancer. I won't go into detail on this slide. I just want to point out that manufacturing is complex, and we understand there are a lot of challenges. Complex logistics for autologous products include collection, manufacturing, shipment, testing, handling, and treatment. Under usual circumstances, coordination of these activities is complex and becomes problematic when influenced by unanticipated events, such as a patient's disease status, improper product handling at the clinic, and transportation. Additionally, there is often the issue of multi-product manufacturing facilities which require appropriate segregation of materials, equipment, and products. For these and other cell and gene therapy products, you may also want to consider where you can add automation to the process. Automation has the potential to reduce manufacturing variability, the potential for more uniformity between the different facilities, and can be more of a closed system, which would have less risk of contamination of the product. In addition to CMC challenges for gene therapies, the increased interest in gene therapy product development has led to bottlenecks in manufacturing. The field is observing a lack of capacity to manufacturing vectors. Incorporation of advanced manufacturing technologies, including improved producer cells, modern bioreactors, and improved purification methods, may improve vector production and help alleviate the bottlenecks, but other solutions are sorely needed. Some of these issues for cell therapies were previously discussed, such as scale-up versus scale-out and the logistics for autologous cells. I want to emphasize here that in addition to the different sources of materials for autologous and allogeneic cell products, there are additional unique development consideration for these two types of products. For example, allogeneic cell lines may be preferred product type by sponsors due to the potential to treat more subjects and be scaled in comparison to autologous cells. However, there's an added risk for allogeneic cells due to the potential for rejection by the immune system. Here's a high-level overview of manufacturing challenges for cell and gene therapy products which have been discussed. These challenges relate to availability of resources, reagents and materials for manufacture, cell culture and testing. There's a huge scope for development in these areas. There are also a number of product-related challenges that arise from the nature of these products, such knowledge of the CPPs and the CQAs, assay development and manufacturing changes of comparability. To help sponsors of INDs understand FDA's expectations and to assist in product lifecycle management, FDA has developed many expedited programs that allow additional avenues of interaction with the FDA. We will discuss these programs and CNC challenges associated with accelerated development in more detail after the challenge question. So challenge question number 1. Take a few seconds to consider which of the following statements is not true. The statement that's not true is B. All CMC considerations for autologous and allogeneic cellular products are identical. For successful development, manufacturing should progress in parallel with clinical development, with full compliance with the regulations required for licensure. Due to the shortened timelines for accelerated clinical development programs, it's a big challenge for manufacturing to keep pace with clinical development. As demonstrated in this graph, CMC review concerns increase as product development advances, with safety being the primary concern at all stages. In accelerated development programs, where fewer and smaller size studies may be conducted, a well-defined manufacturing process should be established and assays should be qualified prior to initiating studies designed to provide the primary evidence of effectiveness to support a marketing application. CMC requirements for licensure are the same for products under expedited programs as for the traditional development paradigms. This schematic highlights how promising clinical results under accelerated development can give less time for product development, which necessitates an earlier focus on critical CMC issues. To meet development timelines, we encourage sponsors to qualify or validate their assays and processes and develop a potency assay early. So how can the FDA help CMC keep pace with clinical during accelerated development? The FDA has developed expedited programs which include breakthrough designation, regenerative medicine advanced therapy designation, fast track designation, accelerated approval, and priority review. Details about these expedited programs can be found in the FDA guidance documents listed on this page. These programs provide programmatic assistance to the sponsors based on the sponsor achieving product development milestones. While some of these expedited programs are applicable for all investigational product types, one is specific for CBER regulated products, and that's the RMAC designation. So the Regenerative Medicine Advanced Therapy, RMAT, was signed into law in 2016 and creates a pathway for designation as a regenerative medicine advanced therapy. The definition of regenerative medicine therapy includes cell therapy, therapeutic tissue engineered products, human cell and tissue products, or any combination products using such therapies or products. Combination products can be eligible for RMAT designation when the biological component provides the greatest contribution to the overall intended effects of the combination products. In addition, the FDA interpretation of the 21st Century Cures Act adds that gene therapies, including genetically modified cells that lead to a sustained effect on cells or tissues, can be considered regenerative medicine therapies. Listed here are the criteria for these 5 programs. These programs are applicable during various stages of development. For all 5 programs, the indication must be a serious condition. Fast Track, Breakthrough, and RMAT designations are distinct designation programs with different programmatic requirements. For Fast Track, non-clinical or clinical data can be used to demonstrate the potential to address unmet medical needs. For breakthrough therapy, preliminary clinical evidence means evidence that is sufficient to indicate that the drug may demonstrate substantial improvements in the effectiveness or safety over available therapies. FDA expects that such evidence generally would be derived from the phase 1 or 2 trials and should involve a sufficient number of patients to be considered credible. FDA encourages sponsors to obtain some preliminary comparative data, which can be compared to available therapy or placebo if no available therapy exists, or to standard of care. RMAT doesn't require evidence to indicate that the drug may offer a substantial improvement over available therapy, as is the case with breakthrough. However, the preliminary clinical evidence needs to be obtained from clinical investigations specifically conducted to assess the effect of the therapy on a serious condition. In regards to CMC, it's essential that the preliminary clinical evidence be generated using the product that the sponsor intends to use for clinical development, not an earlier form of the product, which will not be developed. This slide lists the benefits of these programs. Accelerated approval is an approval pathway, which can be a more rapid pathway to gain licensure. Even though treatment may get to the patient quicker, post-marketing confirmation trials have been required. Priority review provides a shorter clock for review for marketing applications, 6 months compared with the 10-month standard of review. This designation will be assigned at the time of the original BLA or efficacy supplement filing. For the next 3 designations, Fast Track, Breakthrough, and RMAT, frequent meetings with the FDA are the major benefit. Additional benefits are listed for these designations, with RMAT having the most health benefits listed. As previously mentioned, accelerated clinical development can reduce the timeline for product development. Importantly, CMC and GMP requirements and expectations are not changed during accelerated development. Plan accordingly and focus on the product issues early, including CQAs and CPPs, assay and process development and validation, raw material qualification, supply chain, and major manufacturing changes. During phase 1 and 2, you need to plan for commercial scale manufacturing and comparability studies if needed. To be successful, CMC development needs to be aligned with clinical development. The CMC approach towards licensures needs to focus on the essential goal, which is to ensure the availability of a quality product that can be consistently produced at the time of approval. Even though CMC and GMP requirements and expectations are not changed during accelerated development, FDA may exercise some flexibility on the type and extent of manufacturing information that is expected at the time of submission or approval for certain components to a certain degree. This is determined on a case-by-case basis. Areas of potential flexibility include validation strategies, manufacturing scale-up or scale-out strategies, use of post-marketing commitments, or post-marketing requirements. We recommend that sponsors interact with OTAT review staff early during product development. The draft guidance entitled Formal Meetings Between the FDA and Sponsors or Applicants of PDUFA Products describes standardized procedures for requesting, preparing, scheduling, conducting, and documenting formal meetings between the sponsors and the FDA. So why are these meetings so important? Gene and cell therapy products are novel products, and rapid timelines result in an increased need for feedback from the FDA during CMC development. Communication is especially useful throughout the product life cycle for topics that lack published guidance or special circumstances. OTAT provides advice to specific queries via teleconferences or written responses, and we issue written minutes for formal meetings. Okay, so now we're ready for challenge question number 2. Take a few seconds to consider which of the following statements is true. Okay, the correct statement is D. Features of expedited development designations, fast track breakthrough RMAT, vary but always include frequent meetings with the FDA. I'm going to conclude this presentation with a list of guidance documents and other resources for your reference. Listed here are some of the key cell and gene therapy guidance pertaining to CMC that have been published by the FDA. At the website listed at the bottom of this slide is a complete list of cell and gene therapy guidance documents. In addition, this year, Cdrh is planning to publish these 5 guidance documents pertaining to cell and gene therapies. Of particular interest for CMC is the manufacturing changes and comparability for human cellular and gene therapy products draft guidance for industry, which was recently announced. A complete list is located at the website listed at the bottom of this slide. To summarize, the paradigm is changing. Smaller studies, quickly completed, and early studies are supporting licensure. To accommodate this shift, CMC strategies need to be aligned. It is generally easier and less risky to make CMC changes early rather than later. In addition, to manufacturers, think about logistics, how product will be shipped, stored, and handled at the clinical sites. Investment of significant effort into understanding product attributes and analytical testing at all phases of clinical studies is crucial. Clinical development and product development are not always easy to keep aligned, but it's important to make sure that your CMC keeps pace and you don't put off important CMC work, which could result in stalled product development. For cell and gene therapy products, the traditional development life cycle is changing. With accelerated programs and effective products, the challenge for CMC is to keep pace with clinical development. Plan ahead and seek FDA advice. Here's the contact info for OTAT and additional useful websites. This concludes my presentation. Thank you for your interest in cell and gene therapy CMC issues. I'll be taking questions during the upcoming panel session.
Good afternoon, everyone, and I hope everyone's doing well this afternoon. Thank you, Irina, for your introduction. Today, I will be presenting common preclinical challenges in the development of cellular and gene therapy products. This slide gives the learning objectives for today's presentation. In today's talk, I will describe Cdrh OTAT regulated products, help you gain an understanding of preclinical considerations for assessing the safety of cell therapy and gene therapy products. I will introduce the opportunities for early interaction with Cdrh OTAT, and I will discuss common challenges with the preclinical elements of an IND submission. I will now describe the products that are regulated by the Office of Tissues and Advanced Therapies, or OTAT. OTAT is within the Center for Biologics Evaluation and Research, or Cdrh, which regulates a diverse portfolio of biologics products from blood and blood components to therapeutic vaccines to allergenic products. Cellular therapies and gene therapies fall under the regulation of OTAT. Gene therapy products include ex vivo genetically modified cells, non-viral vectors such as plasmids, replication-deficient viral vectors such as adenovirus, adeno-associated virus, and lentivirus, replication-competent viral vectors such as measles, adenovirus, and vaccinia, as well as microbial vectors such as Listeria and Salmonella. Cellular therapies include stem cells and stem cell-derived therapies, including adult stem cells from hemopoietic, neural, cardiac, adipose, and mesenchymal origins, perinatal stem cells from blood placenta or umbilical cord blood, fetal stem cells such as neural fetal stem cells, embryonic stem cells, as well as induced pluripotent stem cells. Cellular therapies also include functional mature differentiated cells such as retinal pigment epithelial cells, pancreatic islet cells, chondrocytes, and keratinocytes. OTAT also regulates other products as well that are described on this slide. This slide illustrates the growing number of gene therapy and cell therapy investigational drug applications, or INDs, that were received by OTAT over the past 2 decades. While the pandemic may have slowed things down just a bit, continued increases in the number of these applications are anticipated to coincide with continuing advancements in these exciting fields of study. Now I will discuss considerations for preclinical evaluation of cell therapy and gene therapy products. The FDA has generated guidance documents to communicate with investigators about the expectations for preclinical programs involving cell therapy and gene therapy products. Specifically, I would like to draw your attention to the guidance document published in 2013, Guidance for Industry, Preclinical Assessment of Investigational Cellular and Gene Therapy Products. This document provides recommendations regarding the substance and scope of preclinical information needed to support clinical studies for investigational cell therapy and gene therapy products. Specifically, the document outlines how OTAT evaluates safety and activity of cell therapy and gene therapy products and emphasizes critical elements to consider in the development of your preclinical program for a cellular or gene therapy product. When considering your preclinical program, it is important to consider the contribution of preclinical data to support your proposed clinical study plan. So how does preclinical data support the proposed clinical study plan? First, preclinical data provides justification for first-in-human clinical trials in subjects with a target disease or condition. Preclinical data can also be used to support patient eligibility criteria. Preclinical data is important in establishing the starting clinical dose level, dosing regimen, or route of administration of your intended clinical product. Preclinical data can also be used to establish the feasibility and reasonable safety of the product administration procedure. Importantly, preclinical data can also help to identify potential toxicities and physiologic parameters to help guide clinical monitoring. And finally, preclinical data can inform a balance of benefit versus risk profile in the context of the particular clinical indication under study. Therefore, a sufficient preclinical program should include objectives that address these aspects. Preclinical studies should be designed to address potential safety issues in a context that considers both the biology of the product, including the putative mechanism of action and intrinsic properties, and the proposed clinical indication. When animal studies are necessary, as they often are with these types of products, preclinical programs should provide rationale for the animal species or animal model selected for preclinical evaluation. Importantly, strategies should be implemented to ensure judicious use of animals and incorporate the 3 R's to reduce, refine, and replace animals in animal studies without compromising the quality of safety and activity data collected from these studies. Also, it's important to consider the quality and applicability of existing accessible preclinical and clinical data available for the proposed clinical product or similar products, subject population, and the proposed clinical route of administration during the development of your preclinical testing program. Finally, the diverse biology and clinical indications, along with the rapid state of evolving scientific research of cellular and gene therapy products, necessitate a flexible strategy for these products, as one-size-fits-all strategy does not apply. While flexible, preclinical testing programs should be science-based and data-driven, based on accumulated knowledge and experience, and also based on available technologies and methods. Early and ongoing communication with pharmacology toxicology staff and OTAT can help to ensure regulatory expectations related to preclinical safety, biologic activity, and understanding of the mechanism of action are met. OTAT provides regulatory and scientific input on your preclinical programs via formal and informal meetings based on the stage of development of your product. This slide shows a product development timeline in reference to the phase of clinical study, and importantly, at the bottom, highlights the opportunity for interaction with OTAT during each stage of development. Early interactions with pharmacology and toxicology staff in OTAT that occurs at the preclinical phase of product development can be critical in ensuring a successful IND submission to initiate a first-in-human clinical study. Today, I will focus on some key aspects and challenges or gaps that have been observed in interacts and pre-INDs and ways to mitigate these challenges. And I'll follow with a discussion of pharmacology and toxicology considerations for an IND submission. The first type of meeting that I will discuss is the Interact meeting, which stands for Initial Targeted Engagement for Regulatory Advice on Cdrh products, previously known as the pre-IND. I have provided the FDA website for Interact meetings in this slide for your information. The goal of an Interact meeting is to enable sponsors or investigators to obtain preliminary informal FDA feedback for innovative investigational products at an early stage of development on specific issues that are not yet at the pre-IND meeting phase. You should plan to request an Interact meeting when you have selected a specific investigational product to evaluate in a clinical study, established your preclinical study plan, and have generated preliminary preclinical proof of concept data and some safety data, but are not yet at a stage for conducting definitive preclinical studies. Examples of pharmacology toxicology content and topics for discussion should be focused on your preclinical development plan and may include the design of proof of concept or other pilot safety, biodistribution, or cell fate studies, the adequacy of the selected animal species or models used, the suitability of innovative preclinical testing strategies, products, or delivery modalities, or to obtain advice on modification of a preclinical program to ensure judicious use of animals. Now I would like to discuss some important points to keep in mind when preparing to request an Interact meeting, and also some things you should avoid in this process. First, in preparation for an Interact meeting, you will want to prepare specific questions that you would like to discuss. The scope of these questions from a pharmacology toxicology perspective can be based on your specific issues in the areas that I have discussed on the previous slide. Next, you should make sure that your package is comprehensive and includes all relevant information needed for the review team to understand your product, preclinical development strategy, and provide useful feedback on your specific questions. Next, while your meeting package should be comprehensive, you will also need to make it concise and succinct, and remember to adhere to the 50-page maximum page limit for the meeting package. Additionally, try to make your package as reader-friendly as possible to allow reviewers to easily navigate through your materials. Finally, please refer to the Interact SOPP provided at the website on the slide for details and procedures for requesting an Interact meeting. Next, some things you should avoid when preparing for an Interact meeting. First, don't forget that the full meeting package is due at the time of the meeting request. Requests that are missing a meeting package or contain incomplete materials may be denied. Importantly, don't forget to stick to the scope of an Interact meeting, which is early in preclinical development, and refrain from conducting definitive proof of concept and safety studies at this stage. Also, in your package, don't forget to include comprehensive summaries of your available preclinical data. These should include the animal model or test system used, detailed methodologies, and a summary of your preliminary results with your interpretation of the findings. Also, in addition to providing the animal model or test system, don't forget to provide your rationale with the relevant supporting data for the selection of a specific animal model or test system. And finally, don't forget to convey all issues and findings of concern that arise from your preclinical studies. Next, I will move on to pre-IND meetings. The goal of pre-IND meetings is to communicate your initial development plans with the core disciplines and other disciplines as necessary to facilitate an eventual IND submission. This should occur when your proof of concept and preliminary safety studies are completed and you're ready to conduct definitive safety studies. The preclinical section of your pre-IND meeting package should include a comprehensive summary of all completed preclinical studies. These can include both in vitro and in vivo studies. They should include a description of the animal model or species used or test system used, detailed study designs, resulting data, and your interpretation of the findings. Pre-IND meeting packages can also contain comprehensive protocols for proposed safety studies. These protocols, or detailed outlines, should include the animal species or models proposed to be used, proposed dose level, dosing regimen, route of administration, delivery procedure, including a device if applicable, study parameters, sacrifice intervals, and other information that may be important in helping the reviewer understand the proposed study. Now I would like to discuss some do's and don'ts from a pharmacology toxicology perspective for your consideration when preparing for your pre-IND submission. First, be sure to include specific questions that you would like to discuss relating to your preclinical program to ensure a focused response from the review team regarding these issues and questions. Next, specify similarities and differences between preclinical and clinical products, including manufacturing process differences, formulation differences, differences in major release criteria, or construct differences that occur between preclinical and clinical lots of your product that are relevant for your investigational product. Next, be sure to include the design and findings of your completed studies and protocols or detailed outlines for your proposed studies in your pre-IND meeting package. Next, provide the scientific rationale for the dose levels, dosing regimen, and duration of your completed and planned studies. If your clinical study will involve pediatric subjects, please provide a discussion of proof of concept data to support prospect of direct benefit. Finally, make your pre-IND meeting package as reader-friendly as possible to facilitate review of your pre-IND materials by the review team. Next, I would like to focus on things that you should avoid for pre-IND submissions. Very importantly, don't begin your definitive safety and distribution studies until you have obtained FDA feedback in your pre-IND setting, as the goal of the pre-IND meeting is to discuss elements of these important studies to prepare you for a submission of an IND. Next, don't forget to provide adequate justification and discussions of any limitations for your selected animal models or test systems. Next, don't forget to discuss issues and concerns that arise from your completed preclinical studies. Next, don't forget to provide a copy of key publications that are cited in your comprehensive summaries. And finally, don't forget to consider and incorporate as necessary FDA-provided comments from Interact meetings if one was held for your preclinical program. I will now discuss investigational new drug applications, or IND applications. Under 21 CFR 312, any use in the United States of a drug or biological product not previously authorized for marketing requires submission of an IND application to the FDA. Any human research study must be conducted under an IND application if the research involves a drug or biological product, is a clinical investigation, and is not exempt from IND requirements. IND should include all necessary information regarding product manufacturing, pharmacology, toxicology, and clinical trial design. INDs are also required to conduct a clinical trial using an approved product for a new indication, new route of administration, or new formulation. Now I will review the documents in an IND application that contain pharmacology, toxicology, or preclinical information. These documents include the investigational brochure as appropriate, the pharmacology toxicology overview, which provides a concise, comprehensive overview of the pharmacology toxicology studies that were completed in your preclinical development program. Pharmacology and toxicology study reports. This includes complete study reports for each preclinical study that was conducted to support your proposed clinical study. Letters of authorization allowing cross-reference to other regulatory files may also be necessary if you're using other regulatory files to support your pharmacology toxicology program. You should also include any applicable publications that support the development of your investigational product. Now I would like to discuss some do's and don'ts for the pharmacology toxicology section of IND applications. First, don't forget that you should include the investigational brochure when this is applicable. Also, ensure that the pharmacology toxicology section of the investigational brochure contains summaries that are not misleading and accurately and sufficiently reflect each conducted study in your preclinical program. Next, you should provide the rationale with supporting data for the selection of each animal model and test system used in your preclinical studies. Next, you should provide rationale and supporting calculation method for your dose level extrapolation from animal to human. Next, you should highlight data from preclinical studies to support elements of the clinical study design, including dose levels, dosing regimen, route of administration, and others. Include preclinical proof of concept data to support prospect of direct benefit when your first-in-human studies involve pediatric subjects. Next, include your proof of concept data to support a novel device if one was used in your preclinical studies. Next, don't forget to provide complete final study reports for all completed preclinical studies. And some things to avoid when submitting your PT section for INDs is don't forget to consider previous Interact and pre-IND meeting comments and incorporate them as necessary into your IND package. Next, don't forget to provide sufficient data to support the safety of the proposed starting dose level and dose escalation scheme. Next, don't forget to explain the similarities and differences between the preclinical product lots and your intended clinical product. Next, don't forget to conduct definitive safety studies per good laboratory practice, or GLP. Next, don't forget to discuss any toxicity signals observed in your animal studies. Also, don't include incomplete, unsigned study reports in your IND application. And finally, don't forget to respond to FDA requests for information within the FDA specified time interval to ensure a timely review of your IND application. I will now address some practical points for your consideration in developing your preclinical program for cellular therapy and gene therapy products and what is expected for pharmacology toxicology studies within these programs. First, your preclinical programs should use biologically responsive animal species and models or test systems, and you should also justify the model or test system used in these studies. Next, you should apply the 3 R's principle to reduce, refine, and replace animals as appropriate in animal studies. Next, you should administer the intended clinical product if feasible, and if you cannot administer the intended clinical product, you should provide a detailed summary of the similarities and differences between the preclinical and clinical products. Next, you should mimic the proposed clinical scenario in your preclinical studies as closely as possible with respect to route of administration, delivery device, delivery procedure, timing of product administration, and you should also ensure adequate study duration. Next, you should design your preclinical studies as controlled, masked, randomized studies with adequate group sizes and of sufficient duration to allow the assessment of the activity, safety, and distribution profiles of your investigational products. Next, you should remember to conduct comprehensive analysis of every unscheduled death that occurs in any of your preclinical studies. Next, you should remember to provide calculation and justification for the dose level extrapolation from animals to humans. Next, you should remember to conduct the toxicology studies in compliance with GLP. Next, you should submit complete study reports for all preclinical studies conducted. And these study reports should include your method of randomization and how your bias was controlled in your preclinical studies. Next, you should follow applicable guidance documents for the investigation of cellular and gene therapy products provided by the FDA. And finally, you should remember to approach CBER OTAT early in your product development program to ensure a smooth transition between early product development to the eventual first-in-human clinical study. Now I will discuss things to avoid in the development of your preclinical pharmacology and toxicology program for cellular and gene therapy products. First, don't assume that FDA personnel will design your preclinical studies. You should approach FDA for feedback at a stage when you have clearly defined preclinical programs for your investigational product. Next, don't assume that testing in one rodent and one non-rodent animal species or that non-human primates are required for preclinical testing for every investigational cell therapy and gene therapy product. Selection of an animal species should be based on the best suited animal model or species to obtain useful information pertaining to the activity, mechanism of action, and safety for your investigational product in your proposed population and your anticipated clinical indication. Next, don't conduct definitive preclinical studies without first obtaining appropriate proof of concept data, such as in vitro and in vivo data, that will be essential for guiding study designs for these definitive preclinical studies. Next, don't conduct non-GLP toxicology studies without the oversight by a quality assurance unit or person that is independent of the personnel responsible for the conduct of this study. Next, don't provide poorly written or organized or incomplete pre-IND or IND submissions, as this can delay the review process. Next, don't submit summary animal data without study reports in an IND. Next, don't assume the inclusion of publications alone in an IND is sufficient to support a clinical trial, as any publications that are included in an IND should contain a description of the publication along with the relevancy to support the clinical study. We will now move on to challenge questions based on the content of my talk. The first question is, what is the type of early interaction with CBER OTAT that can be requested to prepare for your IND submission? Is it an end of phase 1 meeting, a pre-IND meeting, an interact meeting, or both B and C? I'll give you a minute. If you were thinking both B and C, you would be correct. Both an interact and a pre-IND meeting are meeting types that are available to prepare for your IND submission based on the point at which you are in your product development. The next challenge question is a true or false question. True or false, you should include all complete study reports in your pre-IND package. Is this true or false? If you paid attention carefully, you would note that complete study reports, while they should be submitted in your INDs, should not be included in a pre-IND package, which should instead contain comprehensive summaries of your studies that you have completed, as well as any protocols for your planned definitive studies. The third challenge question is, what information for a completed pharmacology toxicology study should be included in an IND submission? Should it be an accurate summary in the IB, a letter of authorization allowing cross-reference to other regulatory files, final signed study reports, or D, all of the above? If you thought that the answer was D, all of the above, then you were right, and you're well on your way to being prepared for an IND submission. The final challenge question is, definitive toxicology studies should always be conducted in non-human primates. Is this true or false? If you guessed false, you would be correct. As I mentioned in my previous slides, that the selection of an animal model should be based on the ability to obtain useful information pertaining to the activity, mechanism of action, and safety of your investigational product for your proposed population and clinical indication. And this may or may not necessitate the use of non-human primates for cellular therapy and gene therapy products. So in summary, from today's talk, you should realize that OTAT regulates a diverse and complex group of products. And the complex biological properties and risks associated with OTAT products necessitate a case-by-case approach for the pharmacology and toxicology programs for these products. Preclinical data submitted in an IND should support the safety and biological activity of the product for the proposed clinical indication. And finally, early communication with CBER OTAT can mitigate potential issues with preclinical programs and help to ensure a successful IND submission. This slide includes some guidance documents for your consideration and includes the links specifically for the 2013 guidance that I mentioned, as well as some other guidance documents that you may find helpful in the development of cellular and gene therapy products. This slide provides my contact information in case there are any questions after my talk today, as well as ways to contact CBER OTAT and other relevant FDA contact information. This concludes my talk. Thank you for your attention, and I look forward to answering your questions and further discussion at our Q&A session. Thank you very much.
Thank you, Carrie and Malek, for your wonderful presentations, and thank you to the audience for your participation. We've received quite a few questions that are fascinating and will lead to a productive discussion. Therefore, let's dive in with the very first question for Carrie. What is different about cell and gene therapy that sponsors should be aware of if they venture into these types of therapies from more traditional biotechnology products?
Thank you for that great question, Irina. I think the main point for the audience to be aware is that gene and cell therapy products are quite complex, and they're often first-in-class types of products. So when you're defining the CPPs and establishing the CQAs, it can be really challenging. More traditional biotechnology products are really well characterized. So, a one size doesn't fit all approach is what's needed for cell and gene therapy products. So, some examples are like, cell therapies, they're living cells, some which may differentiate, expand in vivo. So, the determination of cell fate can be very challenging for those types of products. The logistics that we've discussed in the presentation for cell therapy products is quite complex, and these products often have a short shelf life. So getting your testing, your release testing done and getting the product to the patient in time, often when it has to travel long distances, can be, really quite a big problem. Some gene therapy vectors have the potential for insertional mutagenesis, and those require long-term follow-up. As a big part of my presentation talked about accelerated development and the difficulties with CMC keeping pace with clinical. So, the best advice would be to invest more in CMC earlier. You need to have qualified release assays prior to initiation of any studies intended to provide evidence of safety and efficacy to support licensure. So, plan ahead, be willing to look at different types of assays as you go through the development. You may need to drop some and pick up others. But if you start thinking about CMC early, you're better aligned to succeed than if you wait until the very end. Thanks for that question, Irina.
Yeah, thank you. And I definitely agree with your statement that planning ahead can certainly help with these products. Next up, we will have a question from Alec. What if there are no suitable animal models to evaluate the safety of proof of concept, for example, for CAR T cells? Will in vitro studies be sufficient to support a first-in-human clinical trial?
Hi, thank you for that great question. So as I mentioned in my talk, we recommend a flexible testing strategy and approach for each cell and gene therapy product. And the preclinical safety program should be designed to identify and characterize the potential safety concerns that are specific to the product and the intended clinical population. So using CAR T cells as an example, some safety considerations for CAR T cell products would be, characterization of on-target and off-target toxicities by evaluating the specificity to the target, evaluating the target's expression profile throughout the body, and by conducting tissue cross-reactivity assays. Also, you would like to highlight any previous clinical experience with the target or the antibody or the Fab that you're using, and you also want to evaluate the potential for uncontrolled proliferation of the CAR T cells. Also, you would like to evaluate the potential genotoxicities following insertions of the CAR into T cells. So you should provide adequate scientific justification for the proposed preclinical testing paradigm used to address these specific safety concerns for this type of product. And we understand that in some cases, animal models of the target disease or disorder may not be available. So in these cases, we do expect that the sponsor provides scientific justification for conducting the trial. And this evidence could come from in vitro studies, or perhaps in some cases from preclinical or clinical data from other indications. And so it's really the job of the sponsor to integrate these data, these different strands of data, and make a coherent case for moving their product forward to clinical trial. We also have a CAR T cell guidance for these specific products that you can find on the FDA website that I could refer you to. Thanks very much again for the great question.
Yeah, and thank you. And I would definitely agree with you that the website is a really wonderful resource for our guidance documents, which we have a plethora of. So let's move on to the next question for Carrie. Can you comment on CBER's thoughts on rapid sterility testing and if that can be used as opposed to standard sterility techniques?
So the standard sterility technique in USP 71, as we all know, has been around for quite some time, and it's used by a lot of our sponsors. But you're also permitted to use other types of sterility testing, but you need to make sure that it is comparable to the USP 71, and that you validate the sterility testing before you use it. So we do recognize that there's other methods out there, and it is permitted to be used, but you just need to make sure that it's up to par before you use it as a release criteria.
Thank you so much for that guidance. Next, Malek, is it acceptable to conduct the definitive study in a non-GLP environment?
So if the study was not conducted in compliance with GLP as per the 21 CFR Part 58, then the study report should specify any areas that deviate from prospectively designed protocol and the potential impact of these deviations on the study integrity. So we recommend strongly that the study be conducted according to a prospectively designed protocol, that it be performed as in a non-biased manner as possible, and that it have appropriate record keeping and documentation of all the data. And so you should include oversight. We would highly recommend that you include oversight of the conduct of all non-GLP toxicology studies and each resulting final study report by a quality assurance unit or person who is independent of the personnel responsible for the conduct of this study. And this oversight is important to assure study conduct according to sound procedures and to ensure the quality and integrity of the resulting data.
Thank you so much for that clear answer. Our next question is for Carrie. What are some of the strategies to address CMC issues during accelerated development?
Okay, thanks for that question. As I mentioned earlier, a lot of my presentation focused on accelerated development. Since we're getting a lot of products coming in with accelerated development programs, and I think it's really important to remember that CMC and GMP requirements and expectations are not changed during accelerated development. So where you would have had a longer period of time to conduct a lot of your assay development and qualifications and validations, when you do a traditional development timeline, this is all going to get reduced with the accelerated development. So it's important to remember that, I mentioned earlier that, cell and gene therapy products are complex biologics, and it's very difficult to know what you're going to need right up front. So it needs a lot of forethought. You need to plan for, trying out different kinds of assays and focus on the product issues early, your CQAs and CPPs, and your development and your raw materials, qualification and supply chain, and also major manufacturing changes. Because as we all know, one thing is consistent in almost every program is that your manufacturing you're going to have manufacturing changes throughout the development life cycle of the product. One of the biggest things that we should recognize early is that potency assays can be really challenging, and luckily, the regulations are very flexible with regard to the kind of assay that can be used, as long as it's measuring a meaningful biological parameter. So we have, in my presentation, I highlighted the 2011 potency guidance for cell and gene therapies. I'd highly recommend that folks take a look at that, where you can see some of the approaches that are used to deal with some challenging potency issues. And it's important to remember that a potency assay that measures a meaningful biological parameter is required for licensure. So the other thing is, during phase 1 and 2 for your product, you need to plan for commercial scale manufacturing and comparability studies if they're going to be needed. Traditionally, this may have been something you'd think about further along in development, but because of the shortened timeline, you need to start planning for those things early. And as I mentioned before, you need to make sure that your CMC development is aligned with clinical development. If you come in for requesting a late stage meeting and you still have a lot of outstanding CMC issues, it's going to be very difficult for you to move forward with your study if those issues aren't resolved. So I think, as everybody who's presented from the FDA has mentioned, is communication with the FDA is key. It's especially useful throughout product life cycle for products that are, may not have a published guidance, or there's maybe a special circumstance with your product. We provide advice to specific queries. We don't do face-to-face right now due to the pandemic, but we have teleconferences and written responses. And, for our meetings, we have formal written minutes. So that's a way for you to get advice from us, and that's probably one of the best things you can do if you have an accelerated development program.
Thank you, Carrie, for underscoring the importance of communication, because it's a perfect segue to our next question from Alec. Can pre-IND meetings occur even if a GLP toxicology study is already ongoing if there are questions about the early clinical development plan?
So yes, you can have a pre-IND meeting if the GLP tox study is ongoing. However, this is not ideal, as some of your safety signals that are going to be important in your clinical development may be gained from your GLP toxicology study. So you would really like to get our feedback on the study prior to beginning it, so that if you want to avoid a situation that we would ask for additional studies if you had submitted the study prior to conducting it, we could have avoided those potential the potential necessity for having to add additional studies and save animals in that case. There could also be product issues that may not be, you may not be aware of that could come up. And so those are also reasons why we would really like to see the GLP study design prior to the at the pre-IND meeting prior to conducting the study.
Thank you so much for that response. And we have just a little bit over a minute left, so let's see if we can squeeze in one more question for Carrie. Should 21 CFR 600 also be applied to cell and gene therapy products in addition to Part 211 mentioned earlier?
Well, I'll be able to answer this one quickly, Irina. So the answer to this is yes. Cell and gene therapy products are biologics, and 21 CFR 600 applies to biologics. So they do apply to cell and gene therapy products as well as the 200. So thanks.
Yes, and thank you both to Carrie and Malik for your participation, your wonderful Q&A session. Next, we will move on to the session on the clinical issues commonly encountered in regulatory submissions and clinical study design. This session is moderated by Dr. Peter Rose, and I will welcome him now.
Thank you so much for a fantastic presentation, Carrie, Malik, and Irina, for your valuable perspective, do's and don'ts for product and preclinical regulatory challenges and opportunities. My name is Peter Bross. I'm Chief of the Oncology Branch in Cdrh OTAT. I will introduce this clinical presentations. This session will focus on cellular and gene therapy product development from a clinical perspective with focus on issues commonly encountered in our regulatory submissions. First, Dr. Asha Das will be discussing cellular and gene therapy in oncology, common issues encountered in regulatory submissions. And Dr. Das is currently a team lead and clinical reviewer in our oncology branch, and she came to us from a variety of industry positions, and before that she was head of neuro-oncology at Cedars-Sinai, and she has held some other academic appointments, and her perspective in both academia and industry is invaluable. And next, Rosa Sharafat will give a discussion of the design considerations of clinical trials for rare diseases. And this will provide regulatory considerations for rare diseases to develop biologic therapies, including cellular, gene, and tissue-based therapies. And Dr. Sharafat is a board-certified pediatric endocrinologist currently serving as the acting team lead in Office of Tissues Advanced Therapies, and she was co-chair and speaker on the Cell, Tissue, and Gene Therapy Advisory Committee last September, having to do with adenovirus vector therapies, and she participated in a number of guidance revisions and has represented Cdrh in a number of public discussions, and she was associate professor of pediatrics at Georgetown before that. So Dr. Das' lecture will then proceed now.
Hello, everyone. My name is Asha Das. I'm a physician in the oncology branch in the Center for Biologics Evaluation and Research at the FDA. I'm here to discuss cellular and gene therapy in oncology, common issues encountered in regulatory submissions. My disclosures are here, and I have no financial relationships to disclose. The learning objectives of this session are to provide an overview of Cdrh's cellular and gene therapy product development programs for the treatment of cancer, highlight common issues encountered in regulatory submissions to Cdrh's oncology branch, and summarize OTAT-approved therapies for the treatment of cancer. FDA's regulation of oncology products is overseen by the Oncology Center of Excellence, with coordination among the three centers, CBER, Cdrh, and Cdrh. Much of the work that I will discuss today occurs in CBER, with collaboration with the other centers. CBER's product review offices are here, and my focus today will be the Office of Tissues and Advanced Therapies, OTAT. Examples of OTAT-regulated products are listed here. For the majority of today's discussion, I will focus on the cellular and gene therapies for the treatment of cancer. This graph illustrates submissions of investigational new drug applications, which you can see have grown substantially over the last 2 decades. Let's now discuss some unique considerations when designing cellular or gene therapy studies for the treatment of cancer, especially first-in-human studies. Unlike with conventional drugs, cellular and gene therapies often involve products and technologies for which there is comparatively little experience. Potential risks with these novel products may include secondary tumor formation, migration to non-target sites, an immune response to the vector or the transgene, and insertional mutagenesis. While many cellular and gene therapies are administered intravenously, the treatment may involve more invasive procedures. For example, intracranial administration may require general anesthesia, which poses an additional risk. Many therapies may need leukapheresis and lymphodepletion. And while a key benefit of cellular or gene therapies is that they may offer a long-term or even permanent cure, any problems with the treatment may also be more intense or longer lasting. There are also challenges to establishing a standardized approach for defining and capturing toxicities, such as cytokine release syndrome or CRS. Well-designed early-phase clinical studies are critical to the development of the product, and in these studies, safety is often a primary objective. Dose exploration will vary according to the different products. Historically, the primary objective of a phase 1 oncology trial was to define the maximum tolerated dose, which was then the dose used for phase 2 efficacy studies. More recently, emphasis has been placed on defining the recommended phase 2 dose of a new drug or combination. The recommended phase 2 dose may be lower than the maximum tolerated dose, because additional toxicities may emerge with chronic use. In these early phase studies, preliminary evidence of efficacy may be evaluated, whether it's biologic effects or preliminary clinical efficacy. Feasibility of manufacturing is often assessed. For example, for autologous CAR T cells, early phase studies may provide information on how long it will take to make the product and whether bridging therapies will be needed to control disease while trial participants wait for the CAR T cell treatment. For allogenic CAR T cells, early phase studies can also inform about the risks of graft versus host disease. A few points I'd like to emphasize. To mitigate risk from manufacturing failures, the protocol should be designed so that trial participants are not committed to receive high-risk lymphodepleting regimens until it's known that the product is available. Another point is with bridging therapies, which can make it difficult to determine whether any tumor response observed is due to the prior bridging therapy or due to the investigational product or to both. To help understand the impact of any bridging therapy on the study interpretation, we recommend that sponsors consider conducting separate pre-specified analysis for all trial participants, for participants who receive prior bridging therapy, and participants who did not receive prior bridging therapy. The information gained from these early phase studies can support the development of later phase clinical studies and may even expedite the clinical development program. Another unique regulatory situation is that sponsors may be interested in evaluating the safety and efficacy using multiple versions of a cellular or gene therapy product in a single clinical trial. Although multiple versions of a product can be studied together in a single clinical trial, each version is a distinct product that is generally submitted to FDA in a separate IND application. The objective of these early phase clinical studies is to guide which version or versions of the product to pursue for further development in later phase studies. These studies are not intended to provide primary evidence of effectiveness to support a marketing application, and they're generally not adequately powered to demonstrate a statistically significant difference in efficacy among the study arms. This guidance, studying multiple versions of a cellular or gene therapy product in an early phase clinical trial, provides recommendations for these studies that evaluate multiple versions of a cellular or gene therapy product, including how to organize and structure the INDs, submit new information, and report adverse events. This figure is intended to communicate that the primary and secondary INDs should be clearly identified. Typically, patients who have relapsed or are refractory to available therapies are recruited for these studies. Because of the toxicity concerns, the role of cell therapy in early stage disease is unclear. In terms of the patient population, one approach is to test safety and efficacy in a particular tumor type. Another approach is to target a specific antigen regardless of tumor type. This is a tissue agnostic approach, and these specific targets may require a companion diagnostic for an antigenic target or for HLA restriction. Companion diagnostic assays may require a study risk evaluation. Are trial participants foregoing standard of care or anticipated toxicities of the proposed regimen acceptable? And significant risk devices require an investigational device exemption. Because these studies generally enroll a refractory population, it may be difficult to conduct a randomized study. There may be single-arm trials, and in these cases, typical endpoints include safety, dose finding, objective response rate, duration of response, time to event analyses such as overall survival and progression-free survival will be difficult to interpret in the setting of a single-arm study, and historical controls may be unreliable. In randomized controlled trials, the primary endpoint is often a time to event analysis, overall survival, or progression-free survival. An appropriate control is required and may need discussion with FDA. We also need to take into account potential confounding impacts of concurrent treatments, such as lymphodepletion, the addition of checkpoint inhibitors, and as mentioned earlier, bridging therapies. The starting dose for these first-in-human studies may be based on toxicology data, prior human experience, and the dose should be based on transduced cells per unit weight or body surface area, particularly in pediatric patients, although flat dosing is also used. The dose escalation scheme should consider the anticipated cell expansion in vivo, anticipated toxicities. Dose escalation will usually be in half-log increments for biologic drugs. Log escalation is generally considered too aggressive. Dose escalations typically employ a 3x3 design with 3 participants per cohort. Continual reassessment escalation designs may be considered. Intrapatient dose escalation is not recommended, and enrollment is typically staggered between participants, especially in the first cohort and between dose cohorts. A sufficient interval between administration of treatment is important in identifying any acute adverse events, and it's important to provide justification for the plan and the starting dose based on the clinical or preclinical data. Dose-limiting toxicities allow us to protect trial participants and helps identify the recommended phase 2 dose. These dose-limiting toxicities may be confounded by toxicities of conditioning lymphodepletion regimens. The context, of course, is important. Some cytokine release syndrome may be expected. Severe CRS requiring ICU admissions is generally considered a dose-limiting toxicity. It's important to monitor for off-target toxicities. Sometimes we need to ask the sponsor to ensure that there are clear definitions. Grading of CRS is evolving, and CTC AEs may not be adequate. Instead, you may need to use the ASTCT consensus grading for CRS and neurologic toxicity associated with immune effector cells. Some examples of cancer cell therapy study dose-limiting toxicities that may be considered are listed here. Now, these are only examples from a recently published guidance titled Considerations for the Development of Chimeric Antigen Receptor CAR T-cell Products. What are some suggestions for your general approach to managing toxicities such as CRS? Well, for suspected CRS, include an algorithm for assessment and management. Rule out other causes of fever, such as sepsis and drug reactions. And management of toxicity should be outlined, such as using tocilizumab, which blocks the IL-6 receptor and is now approved to treat CRS. Steroids, which may potentially interfere with T-cell activity. A practical approach is to ensure that these drugs are available in your pharmacy. It's also helpful to provide specific indications for supportive care, fluids, ICU stays, vasopressor, and specify the cytokine sampling requirements. If participants are discharged to outpatient care, they should remain in reasonable proximity to the treating institution in case of delayed toxicities. While the major toxicities, CRS, ICANS, infection, B-cell aplasia, and hematologic toxicities have been well characterized and standardized grading tools exist, toxicity management is dynamic and will continue to evolve. In your protocols, we suggest that you include study stopping rules. These rules allow a temporary pause in enrollment and the treatment of additional participants in order to limit the number of trial participants being exposed to excess risk, which may include death or an increased incidence of unexpected toxicity. It's helpful to specify conditions such as the type and number of adverse events leading to a temporary hold in enrollment and dosing until a safety assessment can be completed. Based on the outcome of your safety assessment, a protocol revision may be warranted. You may decide to modify your eligibility criteria, the dose, or the safety monitoring plan. I'd like to clarify, these study stopping rules are not intended to terminate a study. Their purpose is to control the number of trial participants put at risk in the event that the early experience uncovers important safety problems. These rules may specify a number of serious adverse events or any death that occurs within 30 days of drug administration and will result in temporary suspension of enrollment, which may be a short time until the situation is assessed. The duration of monitoring of adverse events should be sufficient to cover the expected duration of effects. This duration will depend on information from preclinical studies and experience with related products. Long-term follow-up may be required for certain cellular and gene therapies. For example, for integrating viral vector-based products, 15 years of follow-up is required. Your clinical development program can continue while the long-term follow-up is ongoing, and the guidance is referenced here. Sometimes sponsors wish to simultaneously evaluate more than one investigational drug or more than one cancer type within the same overall trial structure. These types of protocols are called master protocols. In other words, a master protocol is one that's designed to test a single investigational product or drug combination in different populations, which in our case is defined by different cancers. This type of trial is commonly called a basket trial and is shown in the first figure. A basket trial evaluating an investigational drug combination may include a dose finding or safety lead-in component. The sub-studies within basket trials are usually designed as single-arm activity estimating trials with an overall response rate as the primary endpoint. A strong response signal in a sub-study may allow for expansion of the sub-study, and each sub-study should include specific objectives, the scientific rationale for inclusion of each population, and a detailed statistical plan that includes sample size justification and stopping rules for futility or efficacy. Another type of master protocol is one designed to evaluate multiple investigational drugs administered as single drugs or drug combinations in a single disease population, and is commonly referred to as an umbrella trial, shown in the second figure. Because of the complexity of these master protocols, sponsors should submit each master protocol as part of a new IND to FDA, meaning that the master protocol should be the only trial that's conducted under the IND. Here are some OTAT-approved therapies for cancer. All the data is from the prescribing information. We're looking at some of the selected highlights for each product, including the indication, number of trial participants, primary endpoint, approval pathway, year of approval, and if there is a risk evaluation and mitigation strategy or REMS program in place. Suppucell was first approved in 2010 based on a randomized trial. The CAR T cells were first approved in 2017, and there have been subsequent approvals with the most recent in 2022. All the approved CAR T cell therapies are either CD19 or BCMA directed. Most are approved on the basis of single-arm studies. You'll note that the objective tumor response rates are of large magnitude, and although not shown here, they're also durable. We're now beginning to see a time to event data as well. The OTAT approved therapies for cancer continue on this slide. And another point I'd like to make is that most are approved under regular approval, although a couple have been approved under accelerated approval pathway. The CAR T cells are generally only available through a risk evaluation and mitigation strategy or REMS program. What is a REMS program? Here's one example. Because of the risk of CRS and neurologic toxicities, these CAR Ts are available only through a restricted program. And the aspects of the REMS are outlined here. They're healthcare facilities that dispense and administer the product, and these certified healthcare facilities must have immediate access to tocilizumab. They must ensure that a minimum of 2 doses are available for each patient for infusion, and these certified healthcare facilities must also ensure their healthcare providers are trained in the management of CRS and neurologic toxicities. As your product enters or continues through development, there are several opportunities to approach FDA for product development discussions. At the Interact meetings, which stand for Initial Targeted Engagement for Regulatory Advice on CBER products, at the CBER Advanced Technologies Team meeting, at pre-IND meetings, which are highly encouraged. Following the IND submission, there are several opportunities at the end of phase 1 or 2 or pre-BLA. So to summarize then, cellular and gene therapies show promise for cancer therapy. These products are moving rapidly from the lab to the clinic. Toxicity is a concern. The products are complex, and often there are many subcomponents to them. There are many regulatory challenges, especially with these novel technologies and therapeutic combinations. Interaction and collaboration is crucial for the future success of these treatments in cancer. There are frequent opportunities to obtain regulatory advice at the CAP meetings, the Interact meetings, pre-IND and IND meetings, and we encourage you to take full advantage of these opportunities. As our discussion is coming to a close, let's take a couple moments to look at a couple of questions. So this is poll question 1. Which type of meeting would not occur prior to an IND submission? Interact meeting, pre-BLA meeting, pre-IND meeting, or a CAP meeting? Take a few seconds to consider the correct answer, which is a pre-BLA meeting. Second question, which primary endpoints are appropriate for a first-in-human study? Overall survival, safety, feasibility, dose finding, progression-free survival, patient-reported outcomes, or quality of life? And the correct answer is safety, feasibility, and dose finding. I'd like to end this presentation by just sharing with you some useful FDA information with the links provided and my contact information. Thank you for your attention.
Thank you for the nice introduction. The topic of my talk today is design considerations for clinical trials in rare diseases. The objectives are to describe the general design considerations for efficient clinical development of cellular and gene therapy products for rare diseases. Number 2, to discuss the use of natural history studies in such trials. And number 3, to provide an overview of the regulatory requirements to provide substantial evidence of effectiveness and safety of cellular and gene therapy products. At the end, I will review the regulatory approval of Zolgensma as an example of a gene therapy product developed for a rare disease. Let's start with the efficient development of cellular and gene therapy products. Rare disease is a disorder or condition that affects less than 200,000 individuals in the United States. Drug development for the approximately 7,000 rare diseases and conditions can be complex for many reasons. There can be challenges with using well-established trial designs, such as endpoint selection can become complex if there is a limited understanding of the natural history of the disease, or a small patient population can also make it difficult to conduct and interpret rare disease clinical trials data. Despite an increase in approvals for novel drugs to treat rare diseases and conditions, there's still a tremendous unmet need for FDA-approved treatments for these conditions. Congress passed the Orphan Drug Act of 1983 to stimulate the development of drugs for rare disease through orphan product development grants, protocol assistance, tax credits equal to the qualified clinical testing expenses, waiver of Prescription Drug User Fee Act or PDUFA marketing application fee, and granting 7-year marketing exclusivity once the drug is approved by FDA. An efficient clinical development program begins with early collaboration between basic scientists and clinicians in the research and development stage. When preclinical studies are beginning, the draft design of future clinical trials and design and conduct of natural history studies with banking of DNA samples should be considered. Collaboration between all stakeholders, including the regulators, is a key aspect of efficient and successful clinical development. We emphasize early and regular communications with the FDA Product Office, including formal regulatory meetings such as Interact, pre-IND, and end-of-phase meetings. For the rare diseases, cell and gene therapy products, investigators should design even the first in human clinical trials such that to be able to provide evidence of effectiveness. Thus, including randomization and concurrent controls are recommended at this early stage. Another crucial aspect of early clinical development for cell and gene therapy is that the manufacturing issues should be addressed and resolved as much as possible before initiating the first in human clinical trial. Early phase trials for rare disease product development are multi-purpose trials that aim at exploration of dosing regimen, potential biomarker finding, and preliminary efficacy, in addition to investigating safety and feasibility of the product. In serious diseases with no existing treatment, the use of placebo as a comparator should be considered if the natural history is not well known or if the disease is heterogeneous and interpretation of data may be challenging without a concurrent control. Therefore, every effort should be made to randomize patients from the beginning of the therapeutic testing phase. We encourage the sponsors to design early phase trials such that all trial participants receive the standard of care. In circumstances that there is an effective therapy available for the particular disease, no study participant should be denied effective treatments in order to be randomized to a placebo-only arm. In general, we recommend add-on designs, which means all subjects will receive treatment previously shown to be effective, and then will be randomized to the added gene therapy product or to the added placebo arm. The pathophysiology of many rare diseases is poorly understood, and natural history data often is variable or is poorly characterized. Especially for rare diseases with limited valuable patient resources, conducting randomized double-blinded trials with concurrent placebo or sham procedure-controlled arms may be the most efficient means of obtaining persuasive evidence of effectiveness. Again, noting that in the rare disease space, even early phase trials may ultimately provide sufficient evidence of effectiveness to support a marketing application. In early phase studies, we recommend a dose-ranging study in which the initial dose is supported by preclinical studies or available clinical information. The dose should be reasonably safe and have therapeutic potential. We encourage investigators to do substantial dose exploration throughout clinical development to identify a safe and effective dose, and this is particularly important for gene therapy products that are currently considered a one-time only treatment, and a repeat dose is not generally advised. In addition, a lot of gene therapy products may be delivered directly into the eyes or brain parenchyma using invasive surgical procedures. To protect patient safety, we recommend a stage approach, meaning that for the first several patients enrolled in such trials, delivery of the product should be only into one side. If the product is deemed as safe, then it can be delivered bilaterally. We also recommend that in the clinical protocol, the investigators include a detailed description of the delivery device, and in addition to standard safety monitoring, there are additional risks associated with gene therapy products, which include immune response to both the vector and the transgene protein. For some gene therapy products, monitoring of a potential insertional carcinogenesis is also necessary. The duration of monitoring should be sufficient to cover expected duration of the effect, which is guided by the information from preclinical studies and experience with related products. Since the effects of cellular and gene therapy products could be permanent, and while it may seem burdensome, long-term monitoring may be appropriate for these products. FDA long-term follow-up guidance for patients who receive integrating viral vector-based products such as CAR-T products that contain lentiviral vectors recommend investigators to continue to monitor clinical trial participants for up to 15 years. However, the clinical development program may continue while long-term follow-up of early phase trial participants is still ongoing. In early phase trials, we encourage the sponsors to explore a wide spectrum of endpoints, keeping in mind that whether there is the need to develop a new assessment tool or to modify an existing one for use as a clinical trial endpoint. Primary efficacy endpoints in trials supporting a marketing application should be clinically meaningful endpoints which either directly measure how patients feel, function, or survive, or can be surrogate endpoints which are reasonably likely to predict a clinical benefit. Next, I will discuss use of natural history studies in product development for rare. When knowledge about the disease is insufficient to guide a clinical development program, or a well-designed natural history may help in designing an efficient drug development program by defining the target population, describing the full range of clinical manifestations of the disease, and by helping in implementation of the critical elements of the clinical study design, such as the study duration and a choice of subpopulations. Natural history studies also can help in developing and selecting outcome measures that are more specific or sensitive to the changes in the disease, or more quickly demonstrate efficacy than existing measures. They may help develop new biomarkers that they may provide proof of concepts, or guide the dose selection, or allow early recognition of safety concerns, or provide supportive evidence of efficacy. These studies should be of sufficient duration to capture clinically meaningful outcomes and determine variability in the disease course. In general, use of natural history data as external control may be appropriate if all of the following criteria are met. When there is an unmet medical need and use of concurrent control is not practical or ethical, the disease course is homogeneous and well documented, study population and historical controls are suitably comparable, and the magnitude of the expected effect is large and self-evident. However, many rare diseases have a heterogeneous course, or due to inconsistency in historical control data collection, interpretation of the treatment effect can be challenging. In general, we do not encourage the use of external historical controls in place of concurrent comparators for rare disease product development. Next, we will discuss substantial evidence of effectiveness for rare disease cell and gene therapy products. The regulatory requirements for approval of all drugs and biologics is substantial evidence of effectiveness and sufficient evidence of safety. Evidence of effectiveness should be obtained from adequate and well-controlled studies. However, we understand that certain aspects of the drug development that are feasible for common diseases may not be feasible for rare diseases, and our regulations provide flexibility in applying regulatory standards. Our regulatory flexibility allows us to exercise scientific judgment in determining the kind and quantity of the data that is required from early phase of the clinical development until the design of adequate and well-controlled studies to demonstrate safety and effectiveness to support a marketing approval. The number of trial participants that is needed to establish effectiveness and safety of a treatment for a rare disease is determined on a case-by-case basis, and there is no specific minimum number of participants to determine adequacy of the sample size. The case-by-case determination of adequacy of sample size is based on the quality and persuasiveness of data, nature of the clinical benefit, duration of exposure, target patient population to be treated after approval, and potential risks associated with the treatment. We generally require more than one adequate and well-controlled clinical trial to provide the substantial evidence of effectiveness necessary to support a future marketing approval. The consistency of the results across two adequate and well-controlled trials greatly reduces the possibility that a biased, chance, site-specific, or fraudulent result will lead to an erroneous conclusion that a product is effective. However, our regulatory flexibility may allow us to rely on a single, adequate, and well-controlled trial in situations that a trial has demonstrated a clinically meaningful effect on mortality, irreversible morbidity, or prevention of a disease with potentially serious outcome, and confirmation of the result in a second trial would be practically or ethically impossible. There are several important considerations: persuasiveness of the single trial, robustness of the confirmatory evidence, seriousness of the disease, size of the patient population, and whether it is ethical and practical to conduct more than one adequate and well-controlled clinical investigation. Next, I will briefly review clinical development of Zolgensma, or onasemnogene abeparvovec, an AAV vector-based gene therapy product for treatment of spinal muscular atrophy, or SMA, which is an example of the review team using our regulatory flexibility and scientific judgment in approval of a gene therapy product for a rare disease. SMA is a rare autosomal recessive neurodegenerative disorder caused by irreversible loss of lower motor neurons in the brainstem and spinal cord, which results from mutations in the survival motor neuron 1 gene, or SMN1. Onasemnogene abeparvovec received FDA approval in 2019 for treatment of infants and children younger than age 2 years with SMA. The product was approved while the phase 3 trial was ongoing, which later on completed enrollment in 2021. BLA approval was based on data from the ongoing phase 3 trial and then completed phase 1 trial in patients with infantile onset SMA, or SMA1. All children enrolled in these two trials experienced onset of clinical symptoms consistent with SMA before 6 months of age, and in both trials, Zolgensma was delivered as a single dose via IV infusion. The ongoing phase 3 trial, which provided the primary evidence of effectiveness, was an open-label single-arm trial which used natural history data as comparator. All 21 participants received a single IV dose of 1.1 times 10 to the 14 vector genome per kilogram of body weight. Phase 3 trials' co-primary efficacy endpoints were the proportion of children achieving the milestone of sitting without support for at least 30 seconds at 18 months of age, And number 2, survival at 14 months of age defined by evidence of the combined endpoint of either death or permanent ventilation. And secondary efficacy endpoint was the proportion of participants who were independent of ventilatory support at 18 months of age. Some unique aspects of SMA clinical trials included relatively homogeneous population, all had onset of symptoms prior to age 6 months, and all had genetically confirmed by allelic deletions of SMN1 gene and carried 2 copies of SMN2 gene. As noted, well-defined natural history data was used as the comparator, and based on the natural history data, no patients meeting the study entry criteria would be expected to attain the ability to sit without support at 18 months of age, and approximately 25% were expected to remain alive without permanent ventilation beyond 14 months of age. At the time of BLA approval, 67% of phase 3 trial participants met the study predefined criteria of survival at 14 months of age, versus only 25% in natural history cohort met that criteria. At 18 months of age, 47% of children in the clinical trial were able to sit without support for more than 30 seconds. In natural history cohort, none of the children achieved the milestone of sitting independently at 18 months of age. For the BLA review, data from the completed phase 1 trial provided supportive evidence of effectiveness. Phase 1 trial also had an open-label, single-arm ascending dose design and enrolled 3 participants in the lower dose cohort and 12 participants in the higher dose cohort. While there was a clear dose response between the 2 cohorts in the trial, due to unresolved manufacturing issues at the time of the trial, there was an uncertainty regarding the administered dose in this early phase trial. The level of uncertainty and the single-arm non-controlled design only allowed for the use of the data from this trial as supportive evidence of effectiveness. However, the efficacy results in the high-dose cohort were compelling. At 20 months of age, 100% of trial participants were alive and ventilator-free, versus only 8% in natural history cohort were alive and ventilator-free. Additionally, 75% of trial participants at 18 months of age achieved the milestone of sitting without support for more than 30 seconds, and 17% were able to stand and walk. In comparison, none of the 3 participants in the low-dose cohort or children who did not participate in the trial achieved any of these 3 milestones. In summary, the clinical development of Zolgensma included several non-traditional elements of a study design for rare disease, which include open-label design of both trials, use of dose escalation cohort in early phase trial, demonstrating a clear dose response, and use of natural history as comparator in the late phase trial. And due to the unique nature of SMA, these features made this condition appropriate for the use of natural history controls, which included a well-characterized and homogeneous disease course, comparability of the trial participants and the children in the natural history cohort, and more importantly, robust treatment effect on an important aspect of the disease, such as mortality and morbidity, which implicated that the use of concurrent control may not have been practical or ethical in the late phase trial. So overall, the benefit-risk profile for this gene therapy product was deemed as favorable, and Zolgensma was approved in 2019. We end the session with 2 challenge questions. Question 1, Which of the following statement is not true? A. FDA requires sponsors to conduct natural history studies in early stages of clinical development to help in designing an efficient drug development program for rare diseases. B. Well-designed natural history studies can help the investigators and sponsors design more efficient drug development programs. C. FDA encourages the sponsors to resolve the product's manufacturing issues as much as possible before initiating the first-in-human clinical trial. Or D. Use of external historical controls in place of a concurrent comparator group is not recommended. I'll give you 5 seconds to decide which one is not true. And it looks like the majority of you chose answer A, which is the correct answer. FDA encourages sponsors to conduct natural history studies in early stages of clinical development. Challenge question number 2. Which of the following statements is true? A. In rare, serious, and life-threatening diseases with unmet medical need, the use of placebo as a comparator should not be considered. B. Randomized placebo-controlled clinical trials may be the most efficient means to demonstrate product effectiveness. C. FDA must not exercise any scientific judgment in determining the kind and quantity of data a sponsor is required to provide for individual drug development programs. Or D. A concurrently controlled randomized first-in-human early-phase trial for rare diseases is not necessary or recommended because the objective of such a study is to assess safety. I'll give you 5 seconds to decide which statement is true. And it looks like the majority chose option B. Randomized placebo-controlled clinical trials may be the most efficient means to demonstrate product effectiveness. So in summary, our regulatory standards require that approval of drugs and biologics be based on substantial evidence of effectiveness and sufficient evidence of safety. Evidence of effectiveness should be obtained from adequate and well-controlled clinical trials, and certain aspects of product development that are feasible for common diseases may not be feasible for rare diseases, and our regulators provide flexibility to apply into the regulatory standards. The next 2 slides will provide links to several guidance documents related to the cellular and gene therapy product development for the rare diseases. And these are CBER and OTAT contact information and resources. I would like to thank my colleagues who helped me prepare these slides, and thank you for your attention, and I will be happy to take your questions in the Q&A session.
All right, thank you so much, Rosa, for your enlightening presentation on some really exciting products that we have. And we had a couple of basic clinical trial questions that we wanted you to address. Wondered if you could elaborate on your discussion of development of products for rare diseases and explain how FDA is allowing some regulatory flexibility for addressing these unmet medical needs. For example, when might FDA consider single-arm studies as an appropriate strategy to support licensing applications?
Thank you, Dr. Bross, for this important question. And apologies for the glitches with the technical glitches with the presentation. So just to summarize what was presented as an example for the clinical development of gene therapy for SMA, under certain circumstances, trials using external historical controls for comparison rather than a concurrent randomized comparator may be considered appropriate design to provide substantial evidence of effectiveness and support the licensing applications. However, generally we consider those circumstances when all the certain criteria are met. So number one, when the gene therapy product is intended to treat a rare and serious disease for which there is an unmet medical need, and inclusion of the concurrent control is considered not practical and unethical. And the disease course is well documented and highly predictable, and can be objectively measured and verified, such as like high mortality, and that was the case in the SMA trial. Also, the study population and the population in the historical controls should be suitably comparable. And most importantly, the expected treatment effect should be large and self-evident and closely associated temporally with the treatment. And when all these criteria exist, we may allow for the regulatory flexibility to consider the external controls in those situations.
All right, thank you. And there's a related question, which is when is it feasible and reasonable, and how does the FDA view the use of real-world data in the development of biological products, especially for the treatment of rare and severe diseases?
Yes, sure. So in situations that the external historical comparator arm is being considered, that external arm data can be driven from prior clinical trial data, which could be one single trial, or pooled data from multiple trials, or the external data may come from observational real-world data. Which could be from registries, EHRs, electronic health records, or medical or pharmacy claims. However, it's important to note that the real-world data source should be temporally and clinically relevant to the investigational arm of the clinical trial to minimize the bias. Also, use of the real-world data necessitates sufficient individual patient-level data to ensure that a well-powered comparison can be made. And even when all these circumstances are met, use of external controls, it could be sometimes inadequate. For example, if important prognostic covariates either are unknown or were not recorded in the real-world data. So as a result, in general, we encourage use of concurrent randomized controls as comparator.
All right, thank you. And Dr. Das, I wondered if you could just elaborate a little bit on the challenges and opportunities for dosing with our products, and maybe you could compare with the Cdrh products, because we know that there's a Project Optimus going on in Cdrh, and how do our products fall into the Project Optimus field?
Sure, thank you, Dr. Bronx. Just kind of to start with some of the dosing challenges that we have with the CAR T cells and with the other engineered cell products. This is a topic that's frequently raised in discussions with our sponsors. And we generally recommend that the CAR T-cell products are dosed based on the percentage of CAR-positive cells. So why do we recommend this approach? Well, because there may be inconsistent control of the transduction. So this is a really important point to emphasize, is that because the transduction efficacy can vary, we generally recommend that you use CAR-positive cells for dosing. And then the dosing can be based either on body surface area or on weight. We frequently see it being based on weight. Also with the starting dose, we like to see a justification of the starting dose based on either the preclinical or the clinical data. And Dr. Bross, I'll turn it back over to you.
All right. Thanks very much. There was a question about a CAT meeting, but I think we're going to discuss that in a later session. And also there were some interesting questions about some of the CAR-T products for hematologic malignancies. There was a question about Yescarta and why some approvals were accelerated approvals and some were regular approvals. I believe I was not a reviewer on these hematologic BLAs, and so my understanding is it had to do with the duration of response. And also I would remind everyone that we do our reviews with close collaboration with the Oncology Center for Excellence, those experts. And so we have been reviewing products. We always have a OCE representative on our licensing review teams. Also, there was a note that there was a randomized clinical study, which I believe was the Zuma 7 study, which is really exciting for follicular lymphoma, and that was an earlier phase indication. And so I think that was a very exciting study because it really validated this whole approach of the CAR-T products for the treatment of hematologic malignancies. But I'm going to, we had a number of questions about orphan products, but I wonder if I could ask Dr. Sharifat if you could say anything about long-term follow-up. There was a question about why did we ask for 15 years on the follow-up studies and not 10 or 30 years or 100 years. And I wonder if you could have any comments on long-term follow-up.
Sure. Thank you, Peter. Yes. So we have the long-term, the updated guidance for a long-term follow-up that first of all, in general, we consider the long-term follow-up for each product based on the risk-benefit profile that is considered for that specific product. As general guidance, we expect a longer durations of follow-up for the gene therapy products that have a vector integrating vector-based and such as lentiviruses, gamma viruses. So those are the ones that 15 years have been proposed, and for other products such as AAV vector-based products as well, we consider the duration of follow-up based on the profile of the non-clinical data and clinical data that is available for that class of products at the time of the the review, and it could be up to 5 years, but for some products, it's been proposed even longer durations. So this is something that, as our knowledge of the toxicity profiles for these products evolves, that also can be revised and modified. But the general rule is based on the risk benefit profile for each category. We decide on the duration of the follow-up.
All right, thank you so much. And I know there's an advisory committee discussion going on today and tomorrow with some interesting findings about long-term follow-up. Dr. Das, we had a couple of questions about the multiple INDs and multiple products, and this is a complicated issue, and maybe we've already touched on this, but I wonder if you had anything to add to help sponsors navigate the challenges of these multiple INDs. I think there was a question about how we can minimize the number of different IND submissions that sponsors would have to deal with, and this is a challenge. I don't know, do you have anything to guide the sponsors in terms of to minimize the burdensome nature of these multiple submissions? All right, maybe that wasn't a fair question, but I think that we're working out the details of this issue, but the basic concept is that we require a separate IND for each product, each variation of the product. Asha, did you want to say anything about that?
Yeah. So just with the primary and secondary INDs, generally we ask that the protocols that are submitted are submitted to the primary IND, and then the additional CMC information to the secondary INDs. And I think this helps with the review because then the protocol is just sitting in the primary IND. I think there was a similar, there was a sort of a related question related to master protocols. These, of course, are submitted under their own IND. So that would be the only protocol under the master protocol IND.
Yeah, thank you. And the one thing I would add is that it's really helpful when sponsors clearly indicate which is their primary master protocol and which are the secondary protocols that just have a variation on the product. And we had a lot of interesting questions, but some of them were related to each other, but I think there were two questions, Rosa, about what situations in which we could use a smaller, relatively small sample size. And I wonder if you had any guidance for sponsors as to how to determine what indications might be appropriate for a smaller sample size for a clinical development plan.
Sure, thank you, Peter. There is, in general, there is no set number of minimum number of participants that is required for a clinical trial for a rare disease. Our general regulatory standard is the substantial evidence of effectiveness and sufficient evidence of safety for these products. So as long as the protocol design, including the number of participants in each arm, is sufficient to provide that regulatory standard, there is not really such a thing as a small number, and it's basically the evidence of effectiveness and safety that needs to be that standard needs to be met.
Okay, great. And I think we're almost at the end. I just saw we had a lot of really interesting questions. There was one really interesting question about whether a master protocol could have investigational products under Cdrh and another under Cbr. And is it feasible to run a study with a master protocol? And the answer is yes. I think we do this all the time. And I think that is the end of our time, and I'd like to thank everyone for answering these questions and for dealing with the occasional glitch in our applications. And so we'll now conclude the question and go on break until I think it's 4:05. And after the break, the next session will be about communication with OTAT. And this important topic will be presented by Eden Chain and moderated by Jen Albert, both from our regulatory management staff.
Hello, everyone. Welcome back from break. Let's continue to dive into our CBER track of 2022's REdI conference. My name is Jennifer Albert, and I will be moderating this next session on communication best practices interacting with regulatory project managers in CBER OTAT. Our speaker is Eden Chane, who is a regulatory project manager in the Office of Tissues and Advanced Therapies, as you all know for short as OTAT. She received her Bachelor's of Science degree in Cell and Molecular Biology from Towson University and a Master's degree in Biotechnology with a concentration in Drug Design and Discovery from Georgetown University. Prior to her time at the FDA, Eden worked in regulatory affairs in the pharmaceutical industry in supporting companies in their ANDAs, or abbreviated new drug applications, INDs, investigational new drug applications, and also NDAs, new drug applications, for both brand and generic drug products. She also has experience in the industry as a scientist, supporting quality control testing for drug products. She comes with great knowledge, and I have the great honor of introducing my colleague and our next speaker, Eden Chane.
Thank you for the introduction, Jennifer. Good afternoon, everyone. My name is Eden Chane. I'm the Regulatory Project Manager in the Division of Regulatory Project Management in the Office of Tissues and Advanced Therapies at the Center for Biologics, Evaluation and Research at the FDA. Today, we will discuss the best practices for communicating with your regulatory project managers for your submissions in OTAT. The Division of Regulatory Project Management, or DRPM, is within the Office of Tissues and Advanced Therapies, or OTAT, in the Center for Biologics, Evaluations and Research. OTAT oversees and reviews a diverse range of products, such as gene and cell therapies, tumor vaccines, immunoglobulins, hemostatic agents, anticoagulants, human tissue products, combination products, bioengineered tissues, and medical devices. Here's an overview of what we will learn today. I will walk you through some of the best practices that may improve your interactions with your RPM, who is the primary point of contact for all regulatory submissions in OTAT, while also increasing your familiarity with the Division of Regulatory Project Management, or DRPM. I'll also be highlighting the best communication practices with RPMs as a primary point of contact for all your regulatory submissions managed by OTAT CBER. So who should you contact if you have any questions about your regulatory application submitted to OTAT? The answer is the OTAT RPM. RPMs hold a critical role in OTAT as the primary liaison between the sponsor and the review team. All communication should be directed to the RPM, who is assigned to the submission under review. The RPM also serves as a regulatory expert and a project manager for all submissions related to INDs, IDEs, BLAs, NDAs, 510Ks, PMAs, HDEs, and related submissions, all pre- and post-submission communications, and internal and the sponsor or applicant meetings. Now, we will discuss about RPM's responsibilities in OTAT CBER. RPMs hold a critical role in CBER OTAT. They handle all application and submission related communications with industry and routinely apply their knowledge and regulatory law, guidance documents, and CBER review policies and processes while managing submissions in their portfolio. Project management, communication, and meeting management are additional skills that RPMs carry in their toolkit. RPMs remain up to date in all of these areas to ensure efficient project management and compliance with all regulatory requirements. When communicating with RPMs, include as much detail as possible. Provide your first and last name as well as your company name. If you're communicating on behalf of a company, include that information as well. Inform the RPM of the application type and number along with the specific reason for the communication. Inform the RPM if there's any time sensitivity related to the request. And if you call the RPM, consider following up with email to ensure the RPM has a clear understanding of the request. When initiating emails to the RPM, the sponsor or applicant should always include the topic and the STN on the email subject line. Because of the RPM's large portfolio, providing clear and detailed communications will assist the RPM with prioritization. Developing and maintaining a good working relationship with the RPMs is critical for successful interaction between sponsors or applicant and FDA during the regulatory review process. This relationship can be established by keeping each other informed, communicating clearly, and following through on action items. Consider establishing a mutual agreeable communication strategy with the OTAT RPM to streamline communications. In addition to the identified authorized representative, provide an alternate authorized contacts, including their full contact information and any respective submission management role they may hold. If the authorized representative has a planned absence, notify the RPM of the dates and alternative authorized contacts who will be covering. During application and submission review, RPMs may send numerous information requests. Acknowledge receipt of these requests and keep RPMs informed of any delays in the response. Timely response promotes a more efficient review. We also encourage sponsors to seek answers to scientific and regulatory questions from the resources found on the FDA websites, such as regulation, guidance documents, and Cdrh SOPPs prior to formulating inquiries to FDA. FDA policy positions are typically documented in FDA guidance documents and SOPPs. Sponsor or applicant can also work with consultants for assistance with strategic product development and regulatory planning. This allows both sponsors or applicant and FDA to focus their respective resources to address more complex and challenging product development and regulatory science issues. In order to protect your proprietary and other corporate confidential information, it's required to establish a secure email account with FDA before initiating interactions with OTAT. Cdrh staff can only send outgoing emails containing regulatory information through secure email. If secure email has not been established upon receipt of an application, this may cause delays in communication. For any questions, contact the FDA Secure Email team. When communicating with the RPM by email, keep in mind the following best practices. To ensure identification of important emails, include the topic and application details in the subject line. When responding to RPM's email, retain the subject line for easy identification. Only include information pertaining to the referenced application or a related submission. If you're sending an email for a new topic or new application, do not reply to previous email string. The submission cover letter should provide all critical information summarizing the purpose and any inquiries that are contained within the submission. A well-written cover letter helps the RPM with the triage and quick routing of the submission. On this slide, we will summarize quick points to consider for best practices when communicating with your RPM. Always provide a clear cover letter that outlines what the submission includes and where it could be found. This information can include cross-referencing information, authorized representatives with accurate contact information, sponsor's name and address, product title and proposed indication, information on any previous OTAT interactions regarding your product. Please don't ask us to pre-review a proposed submission before you submit it just to check. All communications with OTAT should go through the RPMs. Ensure that a secure email is established prior to the first submission. If there are any questions regarding secure email, please send them to the email address listed on the slide. It's also a good practice to always acknowledge receipt of communications from the RPM on the file. Have 1 or 2 backup authorized contacts to ensure a representative is always available during the review process. FDA will not discuss any information regarding your submission with non-authorized contact. In conclusion, I would like to highlight a few important communication resources in this slide. Best practices for communication between IND sponsors and FDA during drug development and formal meeting guidances, and I also encourage you to look at the OTAT webinars. And reviewing these resources and following the best practices outlined on the previous slides will help you to elevate a number of avoidable issues as a sponsor pursues their product development program. Now, we will go over the challenge questions. Challenge question 1. Which one of the following statements is not true? A. RPMs are your main points of contact in OTAT. B. Cdrh staff can only send outgoing emails containing regulatory information through a secure email. C. You should always contact the reviewers directly when you have questions about your submission. D. Be specific and clear in all communication and submissions. The answer is C. Challenge question number 2. Division of Regulatory Project Management, or DRPM, for OTAT is located under A. Center for Biologic Evaluation and Research, or CBER. B. Center for Drug Evaluation and Research, or Cdrh. C. A and B. D. None of the above. And the answer is A. If you have any further questions, please reach out to the email address provided on the slide. Thank you for listening to this presentation. We look forward to communicating with you soon.
Thank you, Eden, for your presentation and for your time to take the following questions from the audience. The first question is a very hot topic. On average, how many applications are in a single RPM's portfolio at a given time?
Thank you, Jen, for that question. So RPM's portfolio varies, but in minimum, we get 150 to 175 applications.
Yes, that sounds like a lot, and it does sound pretty accurate. We are pretty busy over here at Cdrh OTAT. So the next question is another good one. What is the expected time frame for a response when emailing my RPM?
Due to the RPM's large portfolio, the RPMs will prioritize and provide response based on your inquiry's time sensitivity.
That sounds like accurate also, that we do have a large portfolio. So the next question I'd like to bring it on to is can a sponsor request a meeting with reviewers through the RPM assigned? And as you respond, could you mind speaking up just a tad bit?
Yes. So we recommend during the review period to contact the RPM assigned for rapid communication. For after the initial 30-day review period for INDs, we request that there will be a formal amendment and request for a meeting.
Great, thank you so much. Where can, let me read this again. Where can we add an authorized contact to my file?
If additional authorized contacts to be added beyond the person listed on your 1571 or 356H forms, it should be clearly stated in your cover letter.
Thank you. And on that topic of authorized contacts, a member of our audience has asked, can a consultant be an authorized representative?
A consultant can be an authorized representative for the sponsor, but please clearly indicate the consultant contact information in the form 1571 and also 356H form and the cover letter.
Thank you so much. The next question coming in is, will the FDA discuss any information regarding a submission with a non-authorized contact?
No, FDA will not discuss any information regarding your submission with non-authorized contact.
Wonderful. Now let's keep going with these questions. Is secure email necessary prior to sending in a submission?
Yes, it's required to establish a secure email account with FDA before initiating interaction with OTAT.
Thank you so much. Moving on to questions about the cover letter in communication. Should a cover letter submitted to an IND or other application be addressed to the office or the department, the center head, or to the RPM assigned?
The cover letter should be addressed to the office director and in attention to the RPM.
Yes, that's very important. At least it'll identify where and who they're targeting that email communication. It is important. There is a question regarding important things to include when communicating with the RPM. I'm assuming this would be in that email.
Yes, include the name and the company, application type and number, reason for call or email, and also the time sensitivity of your inquiries.
Okay. Moving on to the email side, it will be this next question. Who should you contact if you have questions about secure email, given that we expect most to be a secure email partner with us? Could you answer that one?
Yes, questions can be sent to secure email at fda.hhs.gov.
Wonderful. I have a question regarding what should the subject line contain when emailing the RPM on my file? Would this also apply to all types of files?
Yes, so the subject line should contain the application number and also the reason for the email or inquiry.
Very good. Next is a question of, is it usually communicated to the sponsor when an RPM changes? If so, is the sponsor assigned to a single RPM that they must always correspond with?
The new RPM will usually send email to the sponsor introducing themselves and that they will be managing the file.
Wonderful, thank you so much. Now I have a question going back to authorized. I guess I could read this one. Can we establish a mail group to ensure the authorized group receives the information and facilitates a timely response? I feel like that this question might be a sponsor end question. Do you mind answering that one?
Can you repeat the question one more time, Jen? Sorry.
No, not a problem. The question is if they establish a mail group to ensure that the authorized group receives the information. I'm going to jump in and just say that if you have a group mail address, that should be registered as a secure email through our secure email.fed.hhs.gov. So I'll just leave that there. Sorry about that, Eden. Now, there are some questions regarding types of submissions managed at OTAT. Do you mind commenting on that?
We get several different submissions throughout the time.
We do have another question regarding how do you find out who an RPM is for a particular IND? If one is coming into a sponsor authorized representative several years ago and they can't find that in their files, what would be the best practice for someone to figure out who their RPM is?
Most of the time it's included in the acknowledgement letter, who the RPM will be. So they will get it once they submitted the application, they will see it in the acknowledgement letter.
Right, thank you so much. We have a few questions regarding meeting requests. Does the sponsor need to inform the RPM via email before submitting this person specifically says type C request, but I'm just going to generally ask you if any meeting request?
All meeting requests should be formally submitted to the agency. Jen, I also want to add that if they want to submit a meeting request, they should submit it as an amendment to their application or IND.
Great, thank you so much. I would like to circle back to a question that keeps coming up, is how do we find our RPM? Yes, we have a email address, otatrpms.fda.hhs.gov, that people can also email if they are unsure who their RPM is. We have a few more questions regarding can a sponsor communicate directly to a reviewer if they know their reviewer is assigned to their IND?
no, all communications should be by the RPM.
Right, that's a great answer. It is good to kind of funnel all of that, but it also makes sure that the reviewer is the correct reviewer still working on those types of files. We have a few more questions regarding if a sponsor has multiple applications planned, is it possible to request a specific RPM if a good relationship has been established? That's a tough one to answer, so I'm going to let you do that one.
Yes, so they can request a specific RPM, but it depends on the workload that the RPM have at that given time.
So true, very true. As the times are constantly changing, that is trying to go with the flow. Now, one last question so that we can kind of end a little earlier, maybe, is are these communication practices that you just presented and have been commenting on these questions recommended across all of CBER beyond OTAT?
Yes, these are best practices to follow.
Thank you, Eden. Thank you again so much. And that is about time we should have for questions. Thank you for the rapid fire. But again, thank you, Eden, for your time and expertise. And I will hand it off to Dr. Lepteva for our CBER Day 1 closing remarks. Thank you all.
Thank you, Jen. Good afternoon, everyone here on the East Coast and in other parts of the United States. And warm greetings to all conference attendees from different regions of the world who may be in other time zones. We have concluded the 4th day of the conference and the first day of CBER Track and heard presentations and discussions about common issues in the preclinical and clinical development of cellular and gene therapies. Unfortunately, we had a few technical issues along the way for which we apologize and hope we will have a better connection tomorrow. Now, this has been one of those combined days with talks from both Center for Devices and Center for Biologics. Therefore, concluding it, I would like to thank all of our presenters and speakers and panelists and moderators from both centers, as well as the audience for your continued participation and active engagement. We have one more conference day tomorrow dedicated entirely to presentations from the Center for Biologics Evaluation and Research. So tomorrow we will start the day with a session highlighting CBER's approach to post-marketing surveillance, safety signal detection and evaluation, and safe use of approved biological products, including CAR T cell-based therapies and other CBER-regulated products. We will then move on to an entire session dedicated to the discussion of the regulatory policies and approaches to product development in the field of xenotransplantation. Just to remind everyone, xenotransplantation involves the use of live animal cells, tissues, and organs in the treatment or mitigation of human disease. And while developments in this field have been moving slowly in the past decades, the ongoing advances in genetic engineering and better understanding of the immunological responses and biology of tissue engraftment and rejection have brought some new light to the field of xenotransplantation as a potentially promising means to treat a wide range of human disorders. So you will hear more about it tomorrow. And also tomorrow afternoon, we will have a very interesting panel of experts who will be talking about CBER's initiatives and existing processes for engaging and interacting with a wide variety of our stakeholders from patients and patient advocacy groups to developers of new technologies used in novel product design and manufacturing. And one of the presentations will highlight our communications with other regulators. But that's not all, because at the end of the day, you will hear our product quality experts' recommendations on the development of analytical methods for assay validation, safety evaluation of plasma-derived therapeutics, particularly with regard to product characterization and controlling product impurities. And we will also give an update on CBER-regulated devices. So with all of these topics in mind and in the agenda, we invite you to join us tomorrow for more CBER presentations. Today, however, please do not forget to complete your final evaluations for the day. We greatly appreciate and value your feedback and recommendations. And with that, let me conclude our conference day number 4, wishing everybody a great rest of the day, and we look forward to interacting with you on the conference platform tomorrow. Thank you.